brilliant minds from the field of immunology. We are excited to have you with us for what promises to be an inspiring, informative, and enriching event. Before we begin, I would like to take a moment to extend our heartfelt thanks to the many individuals who have worked tirelessly to make this event possible, including our distinguished speakers, participants, sponsors, and of course, all of you for being here today. Throughout the day, we will delve into thought-provoking sessions, each carefully curated to highlight the latest advancements and research in the field of immunology. We are honored to have a lineup of incredible experts who will guide us through this knowledge journey. A humble request for everyone to kindly put your phones to silent mode. We request all the speakers of the next session to hand over their presentations to the technical team near the stage. To start off, may I please request Professor Dipankar Nandi, convener, IMMUNOCON 2024, to come up on the dais and welcome our esteemed guests for the inauguration. We now request our revered guests to come up on stage. First is Professor P.S. Anil Kumar, Dean, ANF, IISc. Professor Utpal Tatu, Chair, Department of Biochemistry, IISc.
Thanks a lot.
Dr. Taruna Madan, President, Indian Immunology Society.
Thank you very much.
Dr. Amit Awasthi, President Incoming, Indian Immunology Society. And Professor K. Balakrishnan, Secretary, Indian Immunology Society. We are privileged to have you all here. I now request Professor P.S. Anil Kumar to welcome all the delegates on behalf of the Institute.
Good morning, all of you. Dignitaries on the dais, off the dais, faculty members, students from various institutions. On behalf of Indian Institute of Science, I heartily welcome you to Indian Institute of Science for this IMMUNOCON 2024 conference. IISc is very happy to host you all here. And in the next couple of days, you can have very intense discussions and deliberations. And it is apt that this conference is happening in IISc, one of the most leading institutions in the country. And when I have a request to the students that when you are here for the conference, please take some time to see different parts of the campus. It's a very green, nice campus with a lot of facilities. I also request you to go to different departments if time permits and see the kind of research that is going on at IISc and the facilities that we have at IISc. So with this deliberations, I hope that the one aspect which all of us are really worried about is the immunology part. So I hope that this conference will take these aspects to the next heights, and especially in IISc, where we are very strong in the biological sciences aspects, and very soon we will also have a medical school with a lot of collaboration expected between the Faculty of Science, Engineering, and Medicine, and that can take us to the next level for India as far as the healthcare is concerned. So it's very apt that this conference is in this campus at this point of time. And I once again welcome all of you on behalf of Indian Institute of Science to this conference. Thank you.
Thank you, Professor P.S. Anil Kumar. I now request Professor Utpal Tatu to welcome the delegates on behalf of Department of Biochemistry, which is the nodal department organizing this committee.
Thank you. On behalf of all the students, faculty, staff of Department of Biochemistry, Indian Institute of Science, I extend to you a warm welcome, all the delegates, speakers, students, everyone coming from around India and around the world. What a wonderful momentous event it is for us to host this very important conference. So happy to see Professor Talwar, the person who conceived this whole idea about the Immunological Forum, and his active involvement in this conference. And our mentor, Professor Padmanabhan, has found time to come and grace this occasion. Thank you very much, both of you, for being here. Just a couple of words. I think Professor Dipankar Nandi has taken a big leap forward in extending this wonderful area of immunology, taking it forward in the institute. And this is one important step in that direction, having this big meeting. A few words about the Department of Biochemistry. At the moment, I'm the chair, and reflecting back on what's gone on in 103 years of history that Department of Biochemistry enjoys. We've had some very important steps in our growth in various different directions, and as you all know, almost any research in today's world is connected with immunology. You can't be a biologist and not have something to do with immunology. But having said that, I can recount 3 important contributions in the department, which I think stand tall in taking forward the concepts of immunology. In early 80s, we had Professor Subbarao, who introduced allergy and immunology field ahead of his time in many ways at the Department of Biochemistry and at Indian Institute of Science. Late 80s, early 90s, it was Professor Manjunath who took forward and pursued viral immunology in this department and made important contributions. And it was in late 90s and in mid 90s or late 90s that Professor Dipankar Nandi ushered the department into molecular immunology, cellular immunology, with rich traditions that he brought from Nobel laureate Jim Allison's lab in UC Berkeley. And we haven't looked back since then. Teaching graduate students and then, of course, active research area have been hallmarks in the Department of Biochemistry, and we are making good progress in this field. Professor Dipankar Nandi has brought in some outstanding speakers from within and outside for this meeting, and I will end by saying that from the way the audience looks today in this auditorium, I think it's going to be an exciting couple of days for young minds, for students and researchers. I wish you all a great exchange in this exciting, important area of science, and hope you'll have a comfortable, productive stay in the next couple of days. Thank you again. Thank you.
Thank you, Professor Utpal Tatu. I now request Dr. Taruna Madan to tell us a little bit about Indian Immunology Society.
A very good morning to you all. I'm so happy to be here at such a historic place which has contributed so much to Indian science. Respected dignitaries, Padma Bhushan, Professor G.P. Talwar sir, a revered innovator and founder of our society. Padma Bhushan and Padma Shri Professor G. Padmanabhan, veteran biochemist and biotechnologist. Padma Bhushan Professor N.K. Ganguly sir, former Director General of ICMR. He could not join us today, but we definitely remember him. Professor N.K. Mehra sir, Vice President, International Affairs of the prestigious Indian National Science Academy and recently elected Fellow of the International Science Council. Thank you so much, Professor Mehra sir, for bringing this laurel to the country. Professor Santosh Kar sir, an eminent scientist, educationist, and entrepreneur. Our former president, Professor D.N. Rao sir, Professor Anil Kumar sir, Dean, Indian Institute of Science, Professor Utpal Tatu sir, Chair, Department of Biochemistry. Our election commissioners for 2024, Professor Pavan Sharma, Dr. Nibriti ma'am, and Professor Satyan Das sir. Our newly elected president, Professor Amit Avasthi, and the newly elected executive committee members. Our esteemed international and national speakers, fellow immunologists, and delegates. On behalf of the Executive Committee of Indian Immunology Society, we extend a very warm welcome to you all to the much-awaited IMMUNOCON 51, our 51st annual scientific extravaganza at the iconic J.N. Tata Auditorium complex of prestigious Indian Institute of Science, Bengaluru, the science and innovation capital of India. And this is the second time in last 51 years after the first one in 1993. And this being organized by Professor Dipankar Nandi, organizing secretary and convener. Thank you very much, Professor Dipankar, for all the efforts that you have done. We also thank and express our gratitude to the co-conveners, Professor Uday Kumar Ranga, as well as Professor Shripad Patil from NIMHANS, Bangalore. Professor Uday Kumar Ranga is from JNCSR. Thank you, Uday, and thank you, Shripad sir. Very happy to share that India has indeed made a mark in the translational immunology by the regulatory approval of our indigenous CAR T-cell therapy by a startup, ImmunoAct, by none other than Dr. Rahul Purwar, an esteemed member of our society from IIT Bombay, and at 1/10 the cost of global CAR T-cell therapy. So we Indians will now be benefiting from the CAR T-cell therapy, and the cancer patients have really a reason to jubilate. Indian immunologists rose to the occasion during the pandemic and contributed immensely to various vaccines against SARS-CoV-2 using different vaccine platforms, including the famed, most affordable Covaxin, and Dr. Pragya Yadav, our esteemed member, will be sharing the experience during the meeting. Being at the ICMR headquarters and being a part of Indian Immunology Society, it is indeed a moment of pride to be an integral part of these groundbreaking medical technologies of the country. We are indeed indebted to the organizing team of IMMUNOCON 2024, all those whom you will see as volunteers all across and some behind the stage, for selecting a very apt theme of the conference, Newer Beginning in Immunological Munificence. And this was definitely very apt post celebrations of our Golden Jubilee, and befitting the mandate, aims, and objectives of the society to promote and advance the science of immunology with all its subspecialties and allied disciplines, with emphasis on improving human health by encouraging teaching, training, and learning of immunology, by developing structured modules of courses, and promote state-of-the-art knowledge in the subject, with supporting fellowships and scholarships, by developing liaison with other international organizations such as IIIS and FOSIS, having similar objectives to promote scientific interactions and collaborations. For the benefit of young and budding immunologists, a continuing medical education workshop was held in NIMHANS, Bengaluru on 14th and 15th October, and a hands-on workshop on flow cytometry conducted by the tech experts on 16th October. And this was really indeed a great start to the conference. We have several plenary lectures by distinguished international and national faculty on various aspects of immunology, a scientific treat indeed. We will have several rising stars, with many of them speaking and presenting for the first time in an IMMUNOCON, presenting their novel research findings. I urge every one of you to please visit their posters and listen to them, what they have to share with us, and of course, guide them with your critique. Panel discussion on contemporary roles of immunology teaching and research in academia and industries will definitely be a cherry on the top. A special session dedicated to commemorate the translational contributions of late Professor Mohan Ramachandra Vani, Director, National Center for Cell Science, whom we lost early this year in April, to the field of bone and cartilage immunology, organized by his worthy student, Dr. Rupesh Srivastava. Thank you, Rupesh, for this initiative. We will have presentations of newer research tools by our esteemed sponsors, and of course, much awaited award sessions of IIS Senior Scientist Award, IIS Professor G.P. Talwar Mid-Career Scientist Award, and Professor G.P. Talwar Young Scientist Awards, including the 5 new initiated in last 2 years, Academia Industry Partnership Award, Polyphenolics Best Immunology Teacher Award, Golden Jubilee Oration Award, Golden Jubilee Rising Star Award, and Professor N.K. Mehra Lifetime Achievement Award. Our award committees with esteemed immunologists on board have selected the best in each category. The organizers have meticulously planned for the social events, and like the magnificent mixer event we all enjoyed yesterday, this is all to create opportunities for delegates to intermingle, exchange ideas, develop collaborations, and of course, long-term relationships. The Executive Committee 22-24 expresses their immense gratitude to all the contributors who shared and enriched our exciting Golden Jubilee years. Golden Jubilee Year was initiated with an online lecture by Professor Clive Gray, who is here with us again today, who launched our upgraded website with an immense support from Dr. Geeta Rai, our Vice President North. An online Golden Jubilee lecture series was initiated with lectures by IIS stalwarts, and the recorded lectures are available on YouTube for everyone. A record of 50 World Immunology Day celebrations across the country reflected the enthusiasm of all our members towards our 50 years of existence. Felicitations to our revered former presidents of Indian Immunology Society to acknowledge their immense contributions to immunology research and society in the grand celebrations at our 50th IMMUNOCON on the theme Frontiers in Immunology, a Gateway to Healthcare, organized by Dr. Uma Kanga, organizing secretary and convener, and Professor Sujata Mohanty, who is there with us today, co-convener, and the team IMMUNOCON 50 under the astute leadership and guidance of Professor N.K. Mehra sir at the All India Institute of Medical Sciences, New Delhi, the Institute of Genesis of the Indian Immunology Society in 1972. For the first time, dedicated IIS sessions were organized at IUIS, and we thank Professor Clive Gray again, American Society of Reproductive Immunology, and a big thank you to Dr. Savita Kulkarni, who's not here, and Dr. Pramod Gupta, who is here with us, for supporting the third IIS Forces event. We are grateful to Professor Shubhada Chiplunkar, former director, ACTREC, for organizing an IIS Advanced Immunology course in RGCB Thiruvananthapuram. And importantly, initiation of Immune Connect, our newsletter. Second issue will be unveiled today, and would like to express our sincere gratitude to all the contributors who have contributed to this issue, and editorial team of Dr. Rupesh Srivastava and Dr. Chaman Sen. Thank you so much. We assure you that our IMMUNOCON 50 team has left no stone unturned to rise to the grandeur of this historic event. And with more than 600 delegates, the conference is progressing towards a grand success. Our best wishes to the organizing team and all the delegates for a pleasant learning and connecting extravaganza. Thank you. Namaste.
Thank you, Dr. Taruna Madan. I now request Dr. Amit Awasthi to say a few words.
Thank you. First of all, I welcome you all. Dignitaries on the dais, Professor Nandi, Professor Anil Kumar, Professor Tatu, Dr. Taruna Madan, outgoing president, Dr. Bala, secretary outgoing, and esteemed colleague, especially Dr. Padmanabhan, Dr. Talwar, Professor Mehra, Professor Santosh Kar, Professor Sharma, and all. This is an opportunity which comes with the big responsibilities. And I am here, I'm not going to take too much time. Professor Taruna Madan has already given a very extended, report card of what we are doing and why we are in the business. So our whole idea is actually to take the immunology to the level of colleges. And this is my only two agenda during my term. First, I will like to take immunology to the colleges, to the students, where the students like to learn new things, because immunology, if you see, every month is changing very quietly and very significantly, right? She mentioned about the CAR T-cell therapy. We have first startup, which really Rahul has done a wonderful job. And now that actually opens the era of cell therapy, as well as checkpoint therapy in combination for different diseases, not only for cancer, but autoimmune diseases. So I think now we have to train our students to take on to these new areas where they can really contribute in the growth of India. That's what we are all looking for, Viksit Bharat, and that's a dream of our Prime Minister Narendra Modi ji, as well as Secretary DBT, Secretary DST. Everybody is working with the one agenda, is the Viksit Bharat, and immunology has to contribute so much into it because immunology is in the center of health science. The second agenda, what I would like to do in my term is women's in immunology. This is something which I personally like to take a lead on it and like to recognize the women who contributed so significantly in the field of immunology. And we would like to have a series, lecture series for young women in the immunology, the established ones, so that, the hand holding is required for these things. With these two things, I think if I do these two things successfully, I will be happy that I feel that I contributed. And I think Professor Nandi has done this wonderful job. If I see the list and if I go every lecture, I mean, this is amazing. You have to attend all these lectures. The people who are coming from international as well as the national speakers, some of these talks is really, really good. And I really encourage the students to really attend these talks. And that's how you will realize, how we can contribute to the landscape of immunology in India. With these words, thank you very, very much. And I love to meet you all, and let's work together for the betterment of the health and betterment for immunology for India. Thank you.
Thank you, Dr. Amit Awasthi. I now request Professor Dipankar Nandi to tell us a little bit about this meeting.
All right. A very good morning to you all. Thank you so much for coming. It's great to see a packed JN Tata Auditorium post the rains and the cyclone. We were really worried about it, but we are so glad that all of you are here today. So yesterday we had the flow workshop, and I thought the flow workshop would, there'd be a lot of people that would not come, but we exceeded those numbers. So that told me that we would have a full house today. That was a sort of a predictive. I'm so glad that you all have come. Thank you so much. What I'll do today is briefly give you an idea about the meeting and the thoughts behind the meeting so that, everybody is sort of aware on how this came about. Okay. So let me just mention that we are having this meeting after a long time. The last time IISc held a meeting here was in January 1993, a long time. And the last time Bangalore had it was in 2009 by Dr. Shripad Patil. And as mentioned by Taruna, the meeting last year in AIIMS was the 50th meeting of the Indian Immunology Society. It was, a golden jubilee moment, right? So this is the 51st meeting. So I thought that this would be a beginning of a new era. And that is very important because that's, the thinking behind it. So that led us to the use of this theme, which is newer beginnings in immunological munificence. Munificence means bountiful. But I got into a little bit of trouble with the word munificence because a lot of people had to Google it up. And they felt that, I was trying to ape Shashi Tharoor. And that was certainly not my intention, but anyway, so at least it got people to look up, words and the meaning of words. Okay, let me also mention a little bit about the logo. The logo, of course, is an antibody molecule, but there was some confusion because it's not a simple antibody molecule. It's a bispecific. You can see the antigen binding domains are somewhat different. And of course, the main building of the institute is over there to signify that IISc is holding it. So there's some artistic license that is taken. When I send this out to, some of my friends, the feedback that I got from one of them who works with bispecific antibodies is that you should have a knob and a hole because it gives structural integrity to the bispecific. So that was also added. Okay. So my original idea was to have a single session in the faculty hall because, single sessions are always nice, but then there are lesser number of people. But like many of my ideas, it doesn't work out, right? So we had to change, but the one that really changed me was this one, and this is the one that I'm showing of. This was a meeting with students of the student volunteers on Republic Day 2024, and the students felt that the faculty hall would be a small, would accommodate lesser number of people. And so we needed a bigger, space. And so when students said it, they are usually more clued in to the ground realities. So we had to revise our plans, and that's how the JN Tata came in. But bigger places mean increase in cost also. So anyway, we had to take that into account. The program here was as follows: 14th and 15th, there was a CME by Dr. Patil in NIMHANS. 16th was the flow cytometry workshop, which Keshav organized. Keshav and his team organized really well. And then we have the 17th to 19th over here. And then on 20th, we move to the faculty hall. Okay, so we are having, a lot of sessions and a lot of sessions are common. But to me, some of the important ones were, or some of the newer ones were, we have ones on systems immunology, one on comparative and evolutionary immunology. These are not ones that are very common. So then you have ocular immunology, very specialized. You also have one on skin disorders, again, very specialized. So it's good that we have both broad as well as very specialized areas, and there are sufficient number of people to have sessions on this, which is, again, very important. One of my favorites is that we have been able to slip in 2 historical talks. Yesterday, the first one was given by Dr. Shamal Roy, a wonderful story on a drug for leishmaniasis, which most people are sort of familiar with, and the contributions of UN Brahmachari on this particular one. And the other one we'll be having is on discovery of the SARS-CoV-2 virus, which led to Covaxin by Pragya Yadav. So, really important over here. So it's academically, it's going to be very heavy, but we also have other functions to sort of lighten it up. We have Yakshagana, which is tomorrow, and I encourage you all to attend the program. And then we have also dances and musical programs by IISc students, Rhythmika and so on. 20th October is primarily student talks in the faculty hall. Okay, the IMMUNOCON events tend to be large, and yeah, as you know, seen by yesterday's mixer. So it's usually in somewhat fancy places, and it's a little bit different from the usual ones, because we try to project ourselves as modest. But as I said, large events need large funds. So we've got a lot of support from people regarding this. And we are thankful to a lot of people who have helped us in this. Let me mention a little bit. We are thankful to all the granting agencies and industry. But let me mention one particular grant that we got, which is from the Company of Biologists. The Company of Biologists is also, the publisher of Journal of Cell Science. Most of you might be familiar with that. And one of the things that they wanted was proper representation of speakers, which is obvious, as you would see. The other part was on environmental awareness. So what they have done is they have given a grant for train travel for students. So all the students who came by train and applied for the grant before have all got it. They've got, or they will be getting fully reimbursed, right? That's something that people you need to keep in mind. We have also taken up the queue, and we are trying to reduce the amount of plastics. Most of the registered participants would have got water bottles. Please use the water. There's a lot of water around. Please use drinking water from there, and try and avoid the use of plastic bottles. The gift bags are also made of jute, and the gifts to all the distinguished people on the dais include succulents also, because they need less water. So we tried to be environmental friendly as much as possible. So the registrations have been, overwhelming. And so right now we have 623 registrations, and this is, after the cancellations. We had about 20 odd cancellations or so. And if you see the number of people over here, you know that the numbers are real. So the poster presentations, again, we have about 359, and we have, a lot of oral presentations also. So there will be poster awards, and we are about 10 to 12 that the committee will have to choose from 359. So it's a tough job for the committee, but I'm sure they will be all doing a very thorough job about it. The top 4 posters, Feb's Journal has decided to give cash awards to students directly for these. So thanks to Feb's Journals for sponsoring this. All the student speakers on 20th October will get gifts from the local organizing committee, as well as T-shirts from the American Society of Microbiology. So thanks to all agencies for sort of helping us in these initiatives. At times like this, it's also good to think about the past, because the past is often a guide of the future. And so let me just tell you a little bit about people who have worked in the area of immunology in IISc. Professor Modgul was one of the first people and endocrinologist, but he was also responsible for the first flow cytometer in IISc. And then we had Professor Subba Rao, who worked on allergies. In fact, he ran a clinic. He had a day where people would come in and test what are the allergens that they're allergic to. Importantly, he was also convener of the IMMUNOCON 1993 at IISc. Then we had Professor Rabi Naik and Professor Shaila. One was an immunologist, the other was a virologist, and they were, it was a combo of work, and so it's, we need to appreciate their contributions. Then, as Utpal mentioned, you had Professor Manjunath, who worked on immune responses in JV, and Professor Anjali Karande, who worked a lot on monoclonal antibodies. So it's important to recount their contributions as teachers, because even though they may or may not be here, their students are here, and in some cases, even their grand students are over here. And they're contributing either as speakers as chairs, members of the local organizing committee, and so on. So I do want to appreciate their contributions, and we need to recall their contributions in times like this. So we have about more than 20 international speakers, more than 100 national speakers. This is what is there on the website. But in reality, this is what we have finally. So, you know, the program has evolved, and so we have more numbers, and you can see the fair amount of representation that is reflected over here. I have used images only to make the point. So since there are a lot of talks, I really request all the speakers to keep to time. I think it's very important we keep to time. And the second request that I have for speakers is to spend time with the students during the poster presentations. One of the common complaints is that not enough time is given for poster presentations, and not enough faculty meet them. So please make sure that A, you keep to time during the talks, and B, you spend some time in the poster presentation with the students. Do spend some time at the industry halls also, because they have contributed a lot, and they need to feel that they also belong in this meeting. For students, I urge you all to attend the talks and learn. You have a plethora of talks, so huge display, and you can choose in some cases. Do ask questions, overcome your hesitancy. Interact with scientists is a great opportunity. Attend the poster sessions as well as the company stalls. Enjoy the meeting, explore the beautiful campus of IISc, as Anil has mentioned. The final point is, remember, balance is important. Immunology teaches us that balance is important. If you lack the immune system, you're immunodeficient, but too much of an immune response can lead you into allergies and other problems. So balance is very important. So have a good time, but also balance it out. Lots of people to thank, but I'll wait for the final day for this. I wish you all the best for the meeting. And finally, thank you so much for coming. Do enjoy, but be safe. Thank you.
Thank you, Professor Dipankar Nandi. I now request our distinguished guests to reveal the IMMUNOCON 2024 abstract book and Immune Connect issue 2. I now request Professor K. Balakrishnan to propose the vote of thanks.
Good morning, one and all. At the outset, I thank the authorities of Indian Institute of Science, Bengaluru, for the permission to happen this mega event with such an extravaganza. I thank Professor P.S. Anil Kumar, Dean, IISc, and Professor Utpal Datta, Chair, Biological Chemistry, Indian Institute of Science, for their warm welcome address as well as nice words. I thank Dr. Thermo Madan, President IIS, and Dr. Amit Abasti, President-elect and incoming President, for their wonderful and exciting words about the Indian Immunology Society. From the bottom of heart, I extend my heartfelt thanks to Professor Dipankar Nandi, convener IMMUNOCON 2024, for his brainstorming presentation about the meeting and his event and grace the occasion. I thank you very much from the bottom of my heart, sir. I thank once again all the senior immunologists of this country, EC members of Indian Immunology Society, and EC members of Indian Immunology Society elected to act for another 2 years, and young and energetic young minds of Indian Immunology Society, Indian Institute of Bangalore, and immunologists across the country who are here to raise the occasion and make the event grand success. And I thank from the bottom of my heart with immense gratitude the whole organizing team which is working meticulously behind Dipankar Nandi, the young and energetic students starting from last few months. And I thank one and all once again. Thank you very much for the chance given to me. Thank you very much.
Thank you, esteemed dignitaries. We will now proceed to our first plenary session. I would request our team to clear the stage, please. This one? Okay. This is one. Cuban. Please. Sorry for the delay. We will now proceed to our first plenary session, which will feature a recorded interaction with Nobel laureate Professor James P. Allison, Department of Immunology, MD Anderson Cancer Center.
Welcome to IMMUNOCON 2024, Jim. It's such a pleasure for us to have you here, as this is the annual celebration of immunology research in India. It's hosted by the Indian Immunology Society, which is more than 50 years old. This year, the meeting is organized in the Indian Institute of Science, Bangalore, a premier place for scientific research and education in India. There are about 500 participants in this meeting with students, faculty, and industry representatives, and we are all looking forward to your talk to kickstart this meeting. Time permitting, we will really appreciate if you can come and enjoy the beautiful that led to anti-tumor strategies. In particular, his basic studies on the use of anti-CTLA-4 in preclinical models of cancer were followed up with the first immune checkpoint blockade therapy approved by the FDA. In addition, Jim is also known for his studies on the biochemical elucidation of the T-cell receptor, which he did when he was an assistant professor. Subsequently, he moved to UC Berkeley and then did his studies on thymic subsets and the roles of CD28 on T-cell co-stimulation. Currently, Professor Allison is the Regental Professor and Chair in the Department of Immunology, MD Anderson Cancer Center in Texas, USA. As most of you are aware, Jim, along with Tasuku Honjo, received the 2018 Nobel Prize in Physiology and Medicine. We are greatly appreciative of his time and talk to enthuse the next generation of immunologists from India. Jim, please begin your talk, which will be followed by questions by Arushi Kaul, a UG student here, and then Anantika Chandra, a graduate.
Also show some mechanistic studies that we've done yesterday that's showing that the situation in tumors is really a lot more complicated than we thought. And myeloid cells we're discovering play a very important role in what's going on. And I'll just close with some where I think the field needs to go in the future. These are my disclosures. I don't think they're relevant. I'm going to start off by thanking people who did this work. Of course, Max Krummel is a graduate student who, as a graduate student, showed that CTLA-4 was a negative regulator of T-cell activation, flying in the face of papers saying it was a positive one, which was actually in Charlie Janeway's first edition of his immunology textbook. But we looked at that, found some holes in the experiments, did the experiments to straighten it out, and now it's commonly accepted as an inhibitor. I had a lot of trouble convincing pharma companies to move this to the clinic, and finally got help from an old friend, Alan Corman, who helped me team up with a little company called Metarex, make the first antibody to human CTLA, clinical grade antibody to human CTLA-4, which now is known as ipilimumab. And a lot of the work that I'm going to show a little bit today, but it's my colleague and partner, Pam Sharma. And a little bit of the work I'm going to show is from current postdocs in the lab, Naveen Sharma and Betty Shayuk. So in the early 80s, this was what we knew about T-cell activation. And it had become clear that the engagement of the antigen receptor wasn't enough to turn a T-cell on. You had to have a second co-stimulatory signal through the CD28 molecule in order to turn T cells on. And when you got those contemporaneously, you turned on all this stuff to get cell cycle initiated, cell cycle progression initiated, turned off the things that inhibited it, turned on anti-apoptotic factor, and hence allowed the T cells to expand. This is very critical in the immune response. I'm sure all your immunologists know. You know, you've got, what, 100 million different, probably closer to 1,000 billion. Nobody knows the real answer, but different clones in your body, and you've got to expand them from just a few to many hundreds of thousands if you're going to eliminate, a good virus infection or a tumor. So they've got to start proliferating. But while we were doing this work, we came across a gene that had been cloned. Nobody knew the function of it, called CTLA-4. So we made an antibody to it and showed that it, as I said, Max Crumble showed that it was an inhibitory molecule. And the way it works, we believe, is it has about a 4-log higher avidity for the ligands of CD28, the B7 molecules. So we think it accumulates in a cell when it reaches a certain level to shut off the TCR, co-stimulation and TCR signaling, but mostly co-stimulation. It prevents T cells from proliferating anymore. You have to have that, or your immune system will just keep running. FAST and all these other pathways that you hear about that terminate a response really aren't enough to do it. If you eliminate CTLA-4 in mice, they die in 3 weeks because everything just fills up with T cells. So a critical checkpoint. But what does this have to do with cancer? Well, one of the things that we had known since we started working on CD28 was that tumor cells, solid tumor cells at least, do not express the ligands for CD28, the B7 molecules. They have plenty of antigen on them. We showed that by putting B7 into tumors, and they get rejected immediately. They won't even grow in a mouse. No tumor we ever found would grow if you put B7. So the reason it's able to grow initially is because it's invisible to the immune system because, like I said, it may engage the TCR, but you're not going to get that second signal. So the only way you get that is for tumor cell death. Maybe the tumor gets too big or outgrows its oxygen supply or nutrients or whatever. Anyway, starts dying. That gives us an inflammatory signal to the innate immune system. And these antigen-presenting cells, dendritic cells, in particular CDC1 cells, come in and phagocytize the tumor bits, redisplay them in the context of B7, and you start the T cell response. But then, of course, you also start the clock. That happens in every immune response, tumor or not. And the CTLA-4 comes up and shuts it off. At least that's the idea I had. So I just thought, well, let's just disable the brakes, let the car, keep rolling. We've got the ignition switch turned on now. We pushed on the gas pedal. So let's just take the brakes off and let them go as long as we want to. The antibodies will go away and everything will come back to normal. But that was the idea. And so I could spend a lot of time on this, but this is actually a nice photo of cells coming together. This is a T cell. It's been previously activated. You can see the CTLA-4 little vesicles in the back of the cell. When it comes in contact with an antigen-presenting cell, those vesicles are dragged through the cytoplasm to the interface between the cells and literally fuse. Those vesicles fuse with the membrane and spray CTLA-4 right in the synapse, right in the region where TCR and CD28 are doing their thing. Everything just pushes CD28 out of the way. Anyway, in mice, this worked, and this shows a typical experiment. A tumor grows. If you put in a blocking antibody to CD28, the tumor grows slightly faster, showing you that the immune system's trying to get something done, but it got a late start. Just couldn't take it out. But when we gave antibodies to CTLA-4, the tumors still grew for a little while they're developing the army. And then the tumors were rejected. The mice are permanently immune for the rest of their lives to re-challenge with the tumor. So we tried this in many, many tumor types. The ones that were considered strongly immunogenic, particularly chemically induced fibrosarcomas and colon carcinomas and things like that, were readily rejected, just monotherapy with CTLA-4. Some other tumors, like the B16 melanoma, and some others are somewhat resistant to monotherapy. But if you add chemotherapy or a little radiation, or as we showed later, PD-1 when it came on, we never found a tumor that we couldn't cure in mice with this. So despite that, the drug companies weren't very interested. Finally, as I said, with Alec Corman's help and Medirex, we made the antibody. Bristol Myers Squibb got involved and then started after a lot of just small trials, opened a phase 3 registration trial. And in 2011, in 2000, 2005, when I was at Memorial Sloan Kettering, trying to keep an eye on these trials, I met this woman named Sharon Bellman, who was a patient there with, she was 22 years old, just graduated from college, just got engaged, had some trouble breathing, was diagnosed with metastatic melanoma. She had 31 nodules in her lungs. You can see some of them here. She also had, her liver was riddled with tumors, and she had a brain met, a 1.5 centimeter brain met. Also, she came to see Jed Wolchok, colleague of mine, and he treated her with 3 different chemotherapies with radiation, targeted radiation for the brain met. None of it worked. And he told her, you're hospice bound now. There's nothing else I can do, but we got this new drug. And of course, she said she'd been under these other treatments for over 1.5 years and suffering through all of them and all the side effects. But anyway, so she got IPI, and her tumors completely disappeared. She came in for a checkup after getting IPI, came back to see Jed, and she had no tumors anywhere in her body. About three months or so after treatment, Jed called me, and because it was the first patient we'd seen like that, but her tumors completely vanished. Anyway, that was in 2005. She started early 2004. This was late 2005. This is her in 2016 with her kids. I don't have a recent photo, but they're starting to, think about going to college now. So quite a success. And today, as I'll show you in a while, more than 50% of metastatic melanoma patients can respond, not only respond to immunotherapy, in this case, anti-CTLA-4 plus PD-1, but they are in remission. This randomized trial of that combination, about 55% of the patients are alive 9 years after treatment, after a round of treatment. So, Sharon's an example of it. But now we can cure the majority of patients. I don't have time to show you, but we're actually in neoadjuvant trials up to about 70% of patients with durable responses. So this drug, well, anyway, I'll get to that. Sorry, get ahead here. Anyway, this is Sharon. In 2018, we became friends after that, and I asked her to go to Stockholm with me for the Nobel Prize ceremony. There we are celebrating at this grand hotel there. So real success. And it's of note that she was able to have children afterwards, because typically after cancer therapies, people are sterile, but not with these immunotherapies. Anyway, this is the trial, that trial that she was in. It was 800 patients, and you can see the median, this is the placebo control. Placebo control was not unethical in those days because there was no standard of care. No drug had ever prolonged survival in a clinical trial, but it moved it over about 4 months. That would have been enough for approval. But as you can see here, this top line, which is the IPI only, flattens out about 2 to 3 years and stayed at 20%. Shortly after that, there were a lot of patients been treated, and in 2014 there was analysis of over 5,000 patients, of whom 10 years data was available on, and monotherapy, cured 10-year survival. I would say is a cure for this disease with just a single round of treatment with ipilimumab. Whereas before, as you can see here, the median survival was about 7 months. And almost nobody was alive after 5 years. So we went from a disease that was almost invariably lethal with no treatment to 20% here. And then I'll show you how it progressed. As we were publishing the early work on this, Tusker Hanjo reported that a molecule that he'd been working on for some time, and initially thought played a role in negative selection of thymus, was actually another checkpoint. It differs from CTLA-4. It doesn't work on cells during priming at the early stages. It works on fully differentiated effector cells, and its ligand is induced by gamma interferon. So if you have a tumor-specific T cell that gets near a tumor, recognizes an antigen, makes gamma interferon, the tumor upregulates the ligand for PD-1 and turns it off functionally, and so stops it in its tracks. So this track cells that have already been primed. So very different mechanism. Anyway, as I mentioned, when you put these two together, you get up over 50%. And this is a not quite up-to-date slide of that randomized phase 3 trial that resulted in approval of this combination in 2014. So far, I've just talked about melanoma. That was the real target, first target, because there was no drug that ever helped anybody. But it's not just melanoma. This just shows some of the approvals, not all of them. It's not just in melanoma. I know it's hard to read this, but this is IPI plus Nivo, a PD-1, Pembro, a PD-1. And then you go out here and you start seeing non-small cell lung cancer, colon cancer, head and neck cancer, some forms of leukemia. Anyway, many different kinds of cancer that are treated. These are mostly PD-1 antibodies. There are a lot of companies jumped in by making different PD-1 antibodies. There are 12 to 15 in clinical development now. 8 of them that are really used in a big way, and also ligands to PD-L1 now that are coming online. But one of the disappointing things with this, while it shows that this can work in many cancers, and the response rates differ a lot. In non-small cell lung cancer, bladder cancer, as just monotherapy, it's about 30 to 40% response rate. Others like glioblastoma and pancreatic cancer, we don't have much luck at all. Yeah, and if you just normalize, what became apparent very quickly is the response rates were associated with high mutational rates. This was because the targets we showed in others early on are neoantigens generated by mutations. But a real clear difference between these targets and the ones that that are targeted in so-called precision medicine with genomic testing is you're only targeting those mutations which generate drivers, and in most tumor there's more than one driver in the tumor mass, so those are not curative because you just wipe out all the cells that got one driver and the rest come up. The immune system can attack all of them. The immune system is completely agnostic as to what the function of these things are, because if they're made in the cell, there will be peptides presented on the cell surface if they fit the binding motifs of MHC, as you know. And the more of those, the more shots on goal. And so things like melanoma and lung cancer, which were very difficult to treat because of that genomic instability, are sitting ducks for immunotherapy. They've got so many targets. But anyway, this shows the prevalence of different kinds of cancer in the US at least. And we're only reaching about 20 to 30, well, I think it's closer to 30% now. Millions of people have been treated. Only about of total cancer patients with all types of cancer. So we need to do better. And, I don't think we're doing a very good job of that. We'll get back to that later. But anyway, one thing about the immune system, of course, is that it's accepted now as the fifth pillar, along with surgery, radiation, chemotherapy, and these precision genomically targeted therapies. It's accepted as a pillar of cancer therapy, but it can be used with all of those. Excuse me. All you have to do is kill some tumor cells enough to prime an immune response with whatever other technique you're using, and then stop it and use immunotherapy to take advantage of the priming of T cells that's happened. Let the immune system do the elimination of the last tumor cell. Unless you kill that last tumor cell with radiation, chemo, precision therapy, or remove it by surgery, if you don't get them all, it's going to recur sooner or later. Immunotherapy has the potential of eliminating all of them, perhaps, or maybe just reaching some kind of equilibrium, as Bob Schreiber talks about, where there's a constant war going on. We really don't know which it is, but anyway, the thing is that you can mix all of these, and we did a lot of this in animal models, but now it's moved to the clinic, and there's quite a bit of activity. Well, this is what we're doing here in the Allison Institute, led by Pam Sharma, again, is to assemble teams and do small trials, 18, 24 patients, something like that. Get tissue early on, 3 weeks, a month into the therapies, get biopsies, take them back to the lab, see what you got. You have a new molecule. If you do, go to a mouse and see if there's equivalent. Do the mouse experiments and see if it works. See some success there, then that can give you a strong indication that maybe you ought to try it. So you add it to whatever you gave first, and then do another trial. And you don't wait for the clinical response. Again, you get tissues from the tumor after the treatment, reanalyze them and see if anything new popped up, or does it look good now? Where you have those tetrads, you have everything under control, and then and only then do you go to a trial where you look for clinical endpoints and use 100 patients. So I think this is the sort of thing that has to be done if we're going to really, get this work to its ultimate conclusion, which I'll come to in a minute. But there's still a lot of work to do back at this end, a lot of mouse work, a lot of basic fundamental immunology, looking at not just T cells now, but the intricacies and the epigenetic changes that are associated with myeloid cell development, and how we can exploit that. So this is what we're trying to do. Standard therapies, chemo, the targeted therapies, whatever, they move the median survival over a few months, and you call victory. That's been the old way of doing it. We know now from IPI that you can not only move it over, this is kind of exaggerated here, but remember with IPI, melanoma moved it over about 4 months, but it bottomed out at about 20%. And we're about here with melanoma in neoadjuvant trials, for example. But we need to look for these other combinations, triplets, quadruplets, or whatever, of inhibitory antibodies, plus surgery, plus radiation, perhaps, and try to do this and cure as many patients as we can with all different kinds of cancer. There's no reason why we can't do that. The good news is we know we can do it. We've done it. We just need to get smarter and bring the benefit of these therapies to more patients, including India, actually, and that's what we're working on also. So with that, I'll stop. I hope I didn't go over too long. Thank you, and ready for questions. Thank you.
Yeah, thank you so much, Jim, for this talk, because you gave a history of T-cell co-stimulation and then went on to show its applications in anti-tumor therapy. And then you also discussed the future prospects. Thank you so much. I thought it would be better to involve others and they could ask the questions directly to you. So the first question is by Arushi Kaul. She is an undergraduate student in our institute. Arushi, please go ahead.
Thank you. It was great to hear your talk. So the question that I wanted to ask you is that in all the work carried out by you, right, from your work in understanding the structure of T-cell receptors, and then elucidating the activation mechanism of T cells, and then to the discovery of CTLA-4 and its potential as a target of cancer therapy, and later on, as you have described, the work on molecules like ICOS, which is one moment or one find or discovery which was personally the most exciting to you, and why?
I think there's two actually that are equal. One of them was the T cell receptor. When finally realized, what it looked like. We were the first to show that it was had an alpha and a beta chain that were disulfide linked, and that was biochemistry. I didn't know anything about immunology when I did that. I wanted to. It was a trade in biochemistry, and decided at the postdoc level I switched fields and said I want to learn immunology. It's too cool. Cells going through your body and doing all this stuff, and nobody understands how. So that's what I work on. So that was really a big thrill in the early 80s, 82, 81, 82. But I think just emotionally, the first set of mice, when we had all this foundation and understanding CTLA-4 in our lab, but in the field, the only other people that agreed with us that it was an inhibitor, the CTLA-4 was an inhibitor molecule, was Jeff Bluestone. Everybody else was saying it was a positive molecule. That was in the literature, that was in the textbooks. But when we showed that, and the argument was going on, but I said, I'm not, meetings, we would argue it. but we continued as if it was an inhibitory model. So we're right. We're just going to do our thing. But when I saw those mice, there's an interesting story there. The postdoc that set up the very first experiment was new in the lab. Everybody else was working on B7. So I asked Dana Leach, I said, do this experiment. And he came back to me and showed me these curves, and all the mice were cured and all the rest died. I mean, no statistics, high statistics, 100%, 0%. You don't need statistics there. I mean, you need to do it again, obviously. And so, but I was so excited. And then I said, you need to repeat this immediately. And he said, well, it's November and I'm going to go see my girlfriend in Denmark over Christmas. And I'm going to stay for New Year's and I'll come back in January. And I said, no. No, you set up the experiment. Let's do this properly. You set up the experiment. Go do whatever the hell you want to do. And I'll read the mics. I started measuring the tumors, and he only showed me, after they had big tumors and then the decline. He didn't show me anything before that. He just showed me halfway after they were declining if they were still alive. He didn't show me anything much in between. But I was measuring them every 2 days, and then my family came out from Texas, and so I missed a few. They were all the same. I thought, what's going wrong? They're all the same. And I skipped a couple of them. I'd been really went down on hemophilia, but I kind of skipped a few for over the Christmas holidays. And I went back, and there was a clear difference. I mean, I still didn't know what group was what group, but there was half the mice had really big tumors, and the other ones had smaller tumors. And then, a few days later, those smaller tumors turned necrotic and just went away. And I saw that with my own eyes. And I just went, oh my God, we've got something here, and that was it. So I spent the next 4 years trying to convince them up and help us move it into humans. But that was really it, seeing those mice.
Great. Thank you so much, Jim. In the interest of time, we'd like to move on. So the next question is by Anantika Sharma. She's a third-year graduate student in Dr. Keshav Sanola's lab in the Department of Biochemistry, IISc. Anantika, please go ahead.
Hello, sir. Wonderful talk. So I just have a couple of questions. I think Arushi might have asked like a part of it. But what really inspired you to pursue a career in research, in immunology and cancer research? And what advice do you have to give young scientists who are entering this field?
Yeah, I'll try to be brief. But when I was young, 13, 14, I got interested in science and chemistry, and played around and did a lot of reading. And my father was a doctor in a small town, and he really wanted me to go to medical school. And so I got into a special program one summer at the university while I was in junior in high school in the summer, and studied college level biology and worked in a lab. It was yeast, just doing some silly stuff on yeast, but I just loved it. And I just think the messing with just doing the experiments was just so much fun. And so I just started thinking about, medical school, you cram your head with facts so you can make an algorithm when a patient presents, and you better be right every time, because if you're wrong, the least you'll do is not help the patient. But if you're a scientist, you can be out there on the edge and you can be wrong a lot. That's what science is about. Making hypothesis, testing it, making hypothesis, testing it. You can never prove anything. You only fail to disprove it. I just like that. It's your mind against whatever, and you just know general principles, and you don't have to, you develop the details. You make the details for other people to memorize. So that was why, I guess. And so I just wedged after a while, and I just doing science. And the reason I got into, after a while, I got into immunology because I thought we'd do something about cancer, because I was influenced by the fact that my mother and 2 brothers and 2 uncles died of cancer when I was really young. And so I didn't purposely try to cure cancer, but everything I did in basic biology, I would try to think of how that could apply. Let me be clear about that. I still consider myself a basic science, fundamental scientist, fundamental principles. And then you figure out how you use it. You don't say, how do I cure cancer, and then dive at it, in my opinion. Do it the other way around. So that was it. I love science. I love doing the experiments. Got to have to train patients, students, a lot of repetition, a lot of experiments don't work. You got to do everything 3 times. No, a lot of this drudgery, but I find it all fun. So, but
Great. Thanks, Jim. We'll move to Dr. Aditya, who's a clinician in our institute's health center. So he will ask probably a clinician-based question. Dr. Aditya, please go ahead.
So, thank you, Professor Nandi, for this opportunity. It's a great honor to speak to such a great person. I've seen that you have explained that the response rates have improved so much, the clinical response and the survival rates have improved so much. I've seen in my experience that I think uncontrolled diabetes can also cause an immunosuppressive effect and cause this reduced responsive rates as well. So my question is it possible to produce immunoglobulins personalized to each patient? So right now we are, I think, in the immunotherapy, we are producing immunoglobulin, but is it possible to produce in a personalized fashion so that the cure rates increase?
I think it is, but I'm not sure that's the best way to go, because it takes a while, for one thing. And then if you just have an antibody, you have to have a tumor, something that's on tumors and not on normal cells. Leukemias, first the group at Stanford that did that with leukemias, finally, they were doing making them for every patient, and finally they found that a lot of them were the same. That's where CD20 came from, and CD19. Yes, they found a commonality there that ultimately works not because it's an individual, but because it's on those cells at that stage of development. If you could find something like that in solid tumors, I would say yes, you can. But then what you're limited to is putting a toxin on it or something, a chemotherapy agent or whatever, something that'll actually kill a cell, or delivering a cytokine to the area. All those things are possible. But so far the approach has been more like PSMA for bladder cancer and prostate cancer, so we have anchoring other things. And so that could be done, but it's to me that just becomes a drug that, is targeting the tumor and not really an immunotherapy from my description. You're just getting some way of delivering things. Because the tumor is not necessarily going to respond to it unless it's unless it's something it needs to evolve. And most of the unique things on the tumors are just random things which may not even be on the same. The whole molecule might not even be on the same surface. The peptides will be T-cell recognition, but the whole molecule probably, isn't there for most of them. So,
Okay, thank you so much, Jim. If I can ask the last question, I recall that once you had said that science selects you, and can you elaborate on that aspect for the young and budding scientists in the audience, and maybe for their benefit? Can you say a few words on that?
Yeah, I did tell you that, didn't I? And I still believe it, actually. I mean, I think that you really have to enjoy it. It has to be something that speaks to you, in the doing of it, not just the idea. I mean, I have so many, I've had a lot of students that come to my lab and say, I want to cure cancer. And get all excited, but then when they figure out it's going to take, 15 years, and maybe it'll work. They get frustrated and not do it. But if you enjoy the doing of it and intellectual aspect of asking a question and answering it. Maybe initially they're going to be small questions, you know, and you just think, and you got to work like hell to get it answered every one. So, but if you enjoy that, I think science is calling you, and you should go do it rather than something else, because it can be fun. I don't feel like I had to jump next. That's true. I've done what I love.
All right. Thank you so much, Jim. Thank you so much for your time. I just wish that you will come to Bangalore, visit our institute, interact with students. I think it'd be a different flavor. So please do try to come by sometime soon. And I'd like to thank you so much for your time, your generosity, and patience with all of us and the questions. Thank you so much. Thank you. Thank you. Bye-bye. Yeah.
Thank you. And
Please
good
do
luck,
come.
everybody.
Bye-bye.
Good luck
Thank
with
you.
study.
Welcome to IMMUNOCON 2020.
Switch. Thank you, Professor James P. Allison, for guiding us through such an engaging session. We'll now be taking a short break for high tea. Speakers and delegates are requested to proceed to the ground floor, while the students and other registrants are requested to proceed towards the tent in the back for high tea. The speakers who have not yet submitted their presentations are requested to hand over to our technical team. We kindly request everyone to reconvene by 11:40, as we have an exciting lineup of sessions still ahead. Enjoy your break. We'll see you shortly.
We request everyone to kindly come back to the hall and settle down as we will be starting soon. Welcome back, everyone. I hope you all enjoyed the break. As we now resume, we have more captivating sessions lined up. Now we move on to our plenary talk 2 by Professor Rudolf Valenta from Department of Pathophysiology and Allergy Research, University of Vienna. I request the chair of the session, Professor Narendra Mehra, who is well known for his contribution to the growth of immunology in India, to now take over and guide us through the session. You can introduce from here.
Thank you very much. I'm reminded of 1972 when we started the Indian Immunology Society. We used to be only 20 people in the hall. And today what I saw during the inauguration, the hall was all full. And I think this is the growing immunology power in the country. And you see that the average age of the scientists over here is below 30 years of age. Well, we start with Rudy. I call him Rudy, Professor Rudolf Valenta, who comes from the University of Vienna. I've known him for more than 25, 30 years, probably. We used to be partners in the IISc for many, many years. And we always called him the allergy guy. And he has remained true to allergy. I don't want to spend too much of time on your introduction, Rudy, because there's little time and we have to stick to time. But only a couple of things I want to mention about Rudy. Rudy, of course, is the chair for the next International Congress of Immunology, which is going to be held in Vienna in August next year. And we would expect many of our young scientists to actually take part in that Congress there. And Rudy's work has really brought us from what we used to call allergy molecule to now the process of even a vaccine. The vaccine can be made. We used to always talk about the role of IgE, but I think the vaccine has also given indication that you could also use the need for IgG as well. Rudy has, throughout his career, got a number of awards and others, some of them very prominent ones, and I must mention some of these. He received the International Pharmacia Allergy Research Foundation Award, the Paul Ehrlich Award of EACI, and the International SASDET Award. Furthermore, he is a member of the Academies of Sciences in several countries. And that's really a sort of hallmark of a scientist, that if you can become a member or a fellow of not only science academies in your own country, but also outside, that speaks volumes about your work. And his motto, Immunology Without Borders. Rudolf Valenta conducts immunology research with the aim of improving the lives of all people around the world. I don't want to stand between you and the speaker, and the floor is yours, Rudy. There you go.
Well, dear colleagues, I think I will need some help to set up the presentation. In the meantime, let me tell you that it's a privilege and honor to be with you in your beautiful country. It's my second time here and enjoy it always. I would like to thank Narendra for the kind words. We are friends in IISc for more than 20 years. I think that's correct. Let me give my thanks to Deepan Kanandi for making actually my visit possible here. And can you set up also the pointer on the computer, please? He has done a great lot of work here to bring us here, and I think he took care for each and everybody of us. No, the pointer on the screen. Thank you. Oh, fantastic. Now we're in a good shape to start talking about my theme, and this is molecular allergology, and it brings you through the journey of my life, what I'm doing here the last 35 years of my life, walking together with me from discovering the allergen molecules to set up diagnostic tests and molecular vaccines. And I also want to point out a little bit what will be the future, as I believe. On this slide, you find some details about me. You may take a snapshot if you want to dig into our homepages and allergy research programs in Vienna. I show my conflict of interests a little bit longer because I do not think it's a conflict of interest. I think it's very important to work with industry also to bring the vaccines and the diagnostics to the patients. So I think it's very important, and researchers always will have this. Now, when we come to the disease that I'm going to speak about, it's allergy. And I think allergy is very important. It affects 30% of the people. And what is even more important is about 60% of children that if we would test them for the presence of IgE antibodies would be positive. And we call this allergic sensitization. It's kind of a first stage before the disease develops. It's like a precancerosis. And allergy is actually put in your childhood shoes, so to say, after you are born. You can say allergy is a mild disease because it's only rhinitis, but it is not. Think about the many children that have problems to attend the school, who grow up, who cannot do sports, even when they suffer from a very mild form, and this is hay fever. Think about all the children that have atopic dermatitis, that scratch. The parents have terrible time. It's a burden to the families. Think about the people, the adult ones, the children that have severe forms of food allergy and that are afraid of dying from anaphylaxis. Think about the children and adults who have asthma and cannot do sports and have really a very poor life. And think about systemic anaphylaxis that can kill you, for example, by food allergy. So I think you will be with me to understand it's a very diverse disease, and it's in fact a perfect model of personalized medicine because each and every patient is different from the other, depending on the manifestation, the phenotype, and the allergic sensitization, which takes early place after you're born. And there's two elements in it, and they are very simple. There is the formation of IgE antibodies against allergens, and there's the allergens that come from outside into your body. And the allergic patient is characterized by the formation of a certain class of antibodies, IgE, against the allergens, and all the pathology comes from this. It is actually the triggering of the immediate type inflammation by mast cells. What is very important and interesting, there is IgE-facilitated antigen presentation by antigen presenting cells to T cells. So also the T cell activation is IgE dependent because you have IgE as a mediator molecule. And what is very important, the more often you have contact with allergens, the more severe your disease gets because you increase your IgE antibody production. It's like a kind of a unfavorable vaccination to make the wrong antibodies. Now my work started in 1988 when I was a young, unemployed medical doctor, because at that time all the medical doctors in Austria, they were unemployed because there were so many of them, and it was very difficult to get a job. So I came to my mentor, Professor Kraft, and he was working in the field of allergy, and he asked me, Valenta, what do you want to do? You can do immunology or you can do molecular cloning. And then I was reading and I realized that if you can actually understand the basis of the antigen, the gene, you have the tools to make all the antigens in a recombinant form. And I said, Kraft, this is what I want to do. And it was in the year 1988 when a technology came up, and this was expression cDNA cloning. And it's very simple if we speak about it now today, but it was the beginning when I started in the research, and this was making expression cDNA libraries by isolating mRNA from allergen sources, and then looking into these expression cDNA libraries with the IgE antibodies of the patients to isolate the DNA that codes for allergen and then to go to recombinant allergens. And at that time, believe me, there was only a handful of allergens known by the sequence and by the structure. And this technology enabled the breakthrough to knowing almost all of the allergens that are important. Where did it happen? It happened in a very poor building like this one. This is where we started in 1988. And this is one of the buildings that was very poor. And there was not much here, but we had enthusiasm as young people. And actually, this building is also the building where Karl Landsteiner was working when he discovered the blood group. So you can imagine how old it was. Now, what is the thing in the science? And this is, you work hard and you compete and you want to be the first to identify the allergen sequences. And we were basically racing with groups in the United States, cloning the venom allergen, antigen 5, the major house dust mite allergen in Australia, and the major allergen of birch. What you can already understand is that in different countries, different allergens are important. There was also another thing, and this is an allergen that is not specific for one allergen source, like the major allergen of birch. I also found in the expression cDNA library something very unusual. It was a cytoskeletal protein that is conserved in all the eukaryotic organisms. And I asked myself, how can it be that profilin, actin-binding protein, can be an allergen? And then I thought, what is the consequence of this? And the consequence is something that we call cross-reactivity. And this is something that is very interesting because a person that is sensitized to birch can be also allergic to, example, grass pollen that is taxonomically not related at all because of Ig recognition of a protein that is present in a very similar way in the birch, in the grass, and in the wheat. And this was kind of the discovery of cross-reactivity. Now I'm a simple-minded person because I'm a medical doctor, so I wanted to know what can we do after this. And I took 2 recombinant allergens from birch. And this was the paper, actually, that was the first paper describing in vitro allergy diagnosis with 2 recombinant proteins as opposed to a complete birch pollen extract. And the result was very simple. All the 100 patients that were diagnosed by the extract were also diagnosed by the 2 recombinant proteins. And I thought, wow, this is something that is important now because we can replace a whole birch pollen extract by 2 proteins. Now, what is it meaning? It means the transition of the allergen extracts where you skin a cat or you take the shit of the house dust mite and you make an extract where you have all kind of shit in it and also some allergens to the pure forms of the molecules. And I think the picture says it all. And actually, this is a decoding of Timothy grass pollen extract in the individual allergen molecules. And in 1999, a few years later, after the first description of the molecular allergy test system, that was basically brought and is available now to the whole world for molecular diagnosis in the form of molecules. What is the consequence of molecular testing? It allows you to identify precisely what is the sensitizing allergen source. Because I spoke to you before about cross-reactivity, it can mask the identification of it. What is very important is it helps you to identify the patient that should have the right therapeutic allergy vaccine. Because you can also give the wrong vaccine to a person because I will show you in a moment. You can monitor the effect of the treatment because people who are vaccinated, like for SARS-CoV-2, will make protective IgG antibodies, and you can measure this, and you understand cross-reactivity. A simple example. I like this picture because it says it all. There's people sensitized to cat and to dog, and there's some people who are cross-sensitized because they recognize a protein that is present in cat and in the dog. It's albumin. The name is FeLD2 and is CANF3. That's the allergen nomenclature. There's some that are only sensitized to the molecule that is only in the cat. They're only cat sensitized, and there is some that are sensitized only to the dog. So what we have is now the problem. Patient comes, what vaccine do you get? The cat, the dog vaccine, or both? And this can be simply answered by molecular diagnosis. Another example, food allergy. Let's take peanut or let's take milk. In the food allergen sources, you can have some, and I pointed it out in red, molecules that induce severe anaphylaxis and children will die when they eat it. However, there is also some molecules like cross-reactive carbodeterminants. They only bind Ig antibodies, but they do not cause any symptoms. And now with the molecular test, we can tell the parents and each child, are you on the risk of having a severe anaphylactic reaction or not? And then you save the complicated and also very dangerous food provocation testing. Imagine you take a child in the hospital and you provocate the child with milk or with peanut, and then you wait until the child gets anaphylactic, and here you have a simple in vitro test to answer the same question. However, when you have only single molecules, you have one test, one information. And clinicians were saying and complaining, now the costs of testing increases. And then we were going back in the laboratory and we were thinking about how can we cope with now 100, 150 allergen molecules. And at that time, I had a colleague in Vienna and he was printing oligonucleotides on slides. And that was the time when microarrays developed. And I went to my colleague, Manfred Müller, and I said, Manfred, can you also print proteins on the slides? And he said, let's try it. And we did it. And we printed all the allergen molecules by a microprinter on microscope slides. And that was the birth of the microarray. And what is it? It's a small little slide. It's called the array, where you have more than 100, 200 molecules printed. And you can analyze with a drop of blood the whole sensitization spectrum of a person in few hours. Why is this interesting? Because it takes only a drop of blood, 30 microliters, and this is very interesting for early diagnosis of allergy in childhood, in children. And this was actually then something that went into practice in a big European Union project, the MEDAL project, where clinicians had collected sera from patients or children when they grew up until they become adults. So we had the complete evolution of the development of allergies in children from the birth to becoming 20 years old. And we tested in this cohort more than 9,000 children. And it was one of the many results what we got. And we just published this recently, where you can have a molecular sensitization profile of whole Europe. And it's very interesting because you can see it that first of all, in different regions of Europe, you have different sensitizations, like in Sweden, the birch tree is dominating, the BATW1 molecule. When we go, for example, to the BIP cohort, it's from the UK, the house dust mite allergens are dominating. When we go to the south, it's again different allergen molecules. So the exposome is determining against what you are going to be sensitized. And believe me and trust me, if I would do such a survey in India, I can think about what I may find. I will find maybe house dust mite allergens and Plomia tropicalis, and maybe I find cat, but it has not yet been done, and it could be done with this tool in a scientific study. It's very easy. So why is it important to know this? Because you grow up and you do not change your profile over the life. Also this we found out. The adult is having the same profile as the child had, so it's staying stable. And based on this, you can design the immunotherapy vaccines that are important for a particular country. Now, immunotherapy vaccines is a vaccine, but it's not a prophylactic vaccine, not yet. I will bring you to that. It's a therapeutic vaccine. So you inject allergens. The injection of allergen can cause side effects, anaphylactic reactions that can even kill you. So we need vaccines that are safe. That's number one. They need to be easy to produce in a bioreactor, for example, not the allergen extracts that we had so far. And of course, they must have other features to stimulate protective antibodies and regulate events. The whole form that seems to be the one that will work for all the allergen sources. And this is done as. And what is very interesting, when you destroy the fold of a respiratory allergen, it loses its anaphylactic capacity. It's like you denature a toxin and make it a toxoid, but you can do it by genetic means. Here we take out peptides from the Ig binding sites of the allergen, and we graft it actually on a carrier protein to enhance the immunogenicity. The carrier protein is chosen in a very clever form. We use pre-S derived from hepatitis B, and I cannot show you this, but we get also that work, for example, in HPV non-responders, and Professor Wiedermann will show you an example of this. So I'm not dwelling on this. It's another story, but it's a very nice co-effect. The protein is made as a recombinant protein from a synthetic gene, and this is how we design for each of the important allergens, the antigens, and actually this design enables to completely abolish IgE activity, to reduce T-cell epitopes, because we do not want to have allergen-specific T-cell peptides that stimulate late-phase reactions. But what we are inducing is high levels of protective antibodies, and it's like in SARS-CoV-2. The only thing they have to do is they have to prevent the binding of IgE to the allergen afterwards, and that's bringing the relief to the patient, because I showed you before, the binding of IgE to the allergen is more or less the reason of all the symptoms that we can see. I can show you this also in a practical application. That's the design of a birch-borne vaccine, where we take the IgE binding sites as peptides from the major allergen of birch. They are surface exposed, and apple, because there we have cross-reactivity. We put it on the press protein. As you can see here, that's the EP press, and we produce it as a pure recombinant protein in kilogram amounts in Escherichia coli. If you want, it's a very easy process. Now, having said this, and I will tell you also a story about this protein or this vaccine, because I'm very excited to tell you, we have tested this concept for grass-borne allergy and went up to phase 2B. Technology has been licensed out to another company, tested in more than 300 patients, and the EP press actually just has been entering clinical trials, and the first 6 patients were vaccinated without any problems. So this is something that is ongoing, the therapeutic vaccination. Now, having shown you this, I have shown you that we have molecular tests in the microchip format to identify, for example, children when they are sensitized, so that we can think about what we call secondary preventive vaccination. And I have also something that is very exciting we learned in the birth cohort studies. And this is that we can do a molecular test in a child that is very young and does not yet have symptoms, but we can predict if the child becomes allergic later on. And this is what we did in a Swedish birth cohort, where we look in the development over the age of sensitization to PT1, a major allergen of birch. And there's something very interesting, because you can see here in 4 years, the pink bubble, these are all the children who have no symptoms, but they have already IgE antibodies. And you can easily see that these bubbles, that is the symptomatic children, this bubble becomes bigger and bigger because children are exposed more and more, and then they become symptomatic. So this is like finding the precancerosis and doing the early intervention. A very simple concept. And based on the IgE levels that we measure in children at 4 years, we can actually predict with a high accuracy if a child will develop symptoms with 8 and 16 years. And these will be the ones, of course, that will need intervention by allergen avoidance and early immunotherapy. This brings me to the vision, because we are now in the stage of the therapeutic vaccination, but we are thinking about also how we can make the allergen-specific vaccination as a preventive vaccine possible. And here we have, as a first step, that will be the one after the therapeutic, to identify the children that are sensitized at low levels. And then it's a very simple step. We actually perform the immunotherapy early in their life, and we induce a lot of IgG antibodies that will capture the allergen and prevent the IgE from the increase and keep the IgE levels down in the children. This is actually the intervention that you have to imagine. You go into elementary school, you do the testing, you identify the children, and then you vaccinate. This is the program we are following. We have done therapeutic vaccination. We have also done something very important that is a first step for actually showing that the vaccine is not inducing allergenic sensitization. We have vaccinated non-allergic adults to prove that our vaccine is not sensitizing, because that's also a risk. And our next step is now the secondary prevention. Therapeutic vaccination, we're in clinical phase 2B trials. The birch vaccine we expect, or we hope to be registered in the next year. So that's a big step for us. And of course, we will go in the prophylactic thing. So we have seen now a little bit the journey in my life, and you can see where I want to go. And I hope you can see that allergy is very important, and I hope we will also do some collaboration in India. This is also a purpose why I'm here, but there's a second purpose, and this is to express my most cordial invitation to all the delegates here. I see a vibrant Congress, and actually next year we will hold the International Union of Immunological Societies Congress in Austria. The theme is waltzing in the future of immunology for better health. So we think about how we can use immunology for improving the health of people. And we have here this person, and very elegant, it's Johann Strauss. He will celebrate his birthday if he could. He's very old, he's dead actually. But we choose him because everybody knows the Vienna waltz. And for those of you who want to inform yourself, we keep up the homepage of the Congress regularly. You can find the speakers, you can find the conditions of registration. And I can tell you one thing is, it's very easy to come to Vienna. I experienced to fly here, yeah, tried. There's very economic flights. And what is very important, it's easy to stay. We have a low budget accommodation with good quality and food. Use it. Congress prices are the same, but you can save on the other hand. And there's also a big cultural experience that you can make, and you will feel our history, our culture, and our hospitality. And we will bring this to you as we experience it from you now. Thank you very much.
Thank you very much for sticking to time. I think we will have time for at least 2 quick questions. I can see one hand there. Or one hand there. There are a few hands all over. Rudy, can I quickly ask you?
Yes.
It was very good for you to tell us about from allergen extracts to molecules to the vaccine now. I see that the vaccine acceptance is a problem. Is it due to the fact that due to some side effects?
This is no. I think the skepticism regarding vaccines come mainly from the COVID now. Because people have made some, I think, not so pleasant experiences, yeah. And I do not want to go into the detail of this, but what I want to say, this is why I call it also immunotherapy and not vaccination under many circumstances, yeah.
Okay.
Yeah, can I go?
Aruna? Oh, okay, you.
Yeah. So, usually food allergies go with age, but peanut allergy is pretty strong and stay the whole life. What is the reason and what's the current status of immunotherapy, because it's quite deadly?
Actually, you are right, there's different causes. It's also for milk allergy. We actually differentiate between what we call class 1 food allergens. They cause severe anaphylactic reactions. And we have, for example, the birch apple cross-reactivity that is giving mild reactions. For some people, it disappears. It disappears because patients do not continue to consume the food. And in fact, some of them consume the food and they develop IgG antibodies that protect them. But this goes on the side of the side effects and the problems that they have. And I have to say, unfortunately, immunotherapy of food allergy is underdeveloped. And the reason is simple, because there's not so many patients that are affected by it. Respiratory allergies affect 30% of the people, food allergies about 1 to 2%. And of course, we have in the pipeline for our vaccine platform also the food allergy. So if the spearhead hits the target, of course, the food allergy will also come. At the moment, there's oral immunotherapy. It's very bad because it causes very bad side effects. And of course, we want to improve this as well.
Taruna, you wanted to ask.
Thank you, Professor Mehra. Rudy, it was amazing, and to see that you have walked the whole path, starting from identifying first those allergens, making them recombinant, going to immunotherapy, and then now to the vaccines for preventive immunotherapy. I just wanted to bring your attention to the allergies that women face post their menopause. And that is another, group that requires the attention, because hormones do play a very important role in regulating your allergies. And once you are, going down the hormone lane, then it is very important. So I just wanted to bring your attention.
Yes, let me briefly comment, because what I want to say also is that we are ready in Vienna, in Austria, to produce all these microarrays for all kinds of studies here for ourselves. No, we will give it to you. It will be a donation to the people in this country. We can produce for you 5,000, 10,000 arrays to answer all your research questions. Because for me, it's a beautiful thing. We can do it. We have our own printer now. It's for the country, and then you can study all these questions you have here, because we will have all the allergens that you can dream about on it.
So one more, I think I saw a hand there. Yeah, please give the mic to this gentleman here.
Very nice talk. I'm wondering whether you could block IgE and expose to the allergens, and so that you could drive IgG or Tregs. What is your thoughts on that?
Yeah, it's very simple. The simple answer to the question is I didn't show you the historic slide. Immunotherapy was discovered because there was one guy before the pivotal paper by actually Leonard Noon, who applied an anti-serum to cure or to ameliorate the allergic reaction, and this was Dunbar in 1903. 1935, Cook showed that he could, with immunotherapy serum, neutralize the effects of the allergens, the inflammation. And very recently, Regeneron company has performed very successful trials by passively applied blocking antibodies. So if I break it down to a nutshell, if you would only apply the blocking antibodies, it's enough. The technical thing that Regeneron will not master is a very simple thing. You cannot make so many blocking antibodies to cover, for example, house dust mite, because there's 6 different allergens. And in their approach for a cat, there's one allergen, they had to have 2 blocking antibodies. For birch, they have 2 or 3. And if you come now to house dust mite, they will need 12 or 15, and that will not be economic anymore. But coming to your question, inducing blocking antibodies, in my opinion, is it.
Here is the last question, short answer.
Thank you for your very informative talk. So I have a very short question. So during the immunotherapy episodes, what you do it in the clinic, so the epitope of the food
The profile, we have measured this, the recognition profile in terms of the epitopes does not change. There is nothing like this in adults like epitope spreading. It does not exist. However, the magnitude or the levels of IgE antibodies increase in the patient, but they bind to the same epitope.
Okay.
So with this, we sort of close the lecture and thank the speaker for his wonderful talk, very educational.
Our thanks to the speaker and chair. We would like to invite the chair of this session and the speaker to kindly come up on the dais for a group picture.
Come on the IISc. Where is the memento? I don't know. Is there a memento to be given?
Sir, actually the mementos are back in the registration.
In the bag. Where is my book?
The elections for Indian Immunology Society were held, and we request Professor Pawan Sharma to kindly come up on the dais.
Good morning, namaskar to everyone. This was the election year of the society, and I'm here to present the results. First of all, I want to acknowledge the cooperation and very active help I received from my co-members in the committee, Professor Nibriti Das and Professor Satya and Das. I don't know if Satya is here or not. And of course, we also thank all those who came forward to shoulder the onerous responsibilities of running a society like IIS, prestigious society like IIS. And for conducting these elections, we used the list of members which were given to us by the Secretariat of Immunology Society. And this election was conducted using the election buddy online voting platform. And the details of these election is also available on the election buddy website. So I will announce the results of the election. First of all, I'm very happy to say that as many as 892 voters this time participated in casting their votes, which is a record number this time. And so thank you very much, all of you. Thank you, you participated in that. This is a record number. And I will announce the name of the officers which have been elected. And I request them if they can identify themselves or can come here. I don't know, whatever you can decide. So I will actually appreciate if they can stand up and identify themselves. For the post of president, it is Dr. Amit Awasthi, THSTI. Idhar aa jao. And we have 4 vice presidents, one each from each region. Vice President from North, Dr. Uma Kanga. I think she's not there. Vice President South, Dr. V. Aravindan. Both North and South are missing. Okay. Vice President East, Professor Santosh Kar. Okay. Vice President West, Dr. Sharad Dalai. Secretary, Dr. Kaustubh Nayak, ICGB. Joint Secretary, Dr. Gaurav Sharma, PGI, Chandigarh. Treasurer, Dr. Deepak Rathore, THSTI, Faridabad. Then we have the members of the executive committee. In this election, we introduced the concept of having members from each region, one each from each region, and then another 4 members less than age of 40, so that there is a representation of all regions and young and old mix up there. So, member of executive committee from West, Dr. Pramod Gupta. Member EC East, Dr. Dimpu Gogoi. Member EC North, Dr. Rajiv Tyagi. Okay. Member EC South, Dr. Amudhan Murugesan. He's there. And the next 4 members are from the category less than 40 years old. Dr. Shrikant Sadhu. Dr. Priyankar Day. Dr. Vignesh Sundararajan. Dr. Rajesh Meher. Dr. Rajesh Mehar. So, so this is the new executive committee. Some members are missing, but I think they are present in spirit here. So many congratulations to the, please join me in congratulating them. Thank you very much. You want to say something? You want to take picture? Picture ले लो. इसी की picture लो. Time नहीं है चलो.
Thank you, everyone. Thank you.
सुंदरराजन कौन है? युवा सुंदरराजन.
Thank you, Professor Sharma. Next lined up, we have a 15-minute technical presentation by 10x Genomics.
Is it on this one? What is this? Kind of clicker. Is this the clicker? Okay. Right. All right. Thanks. Thank you for the opportunity to speak today. My name is Benson. I'm the Science and Technology Advisor at 10x Genomics. And today, this is a presentation of my talk, Assess the Full Richness of Biological Complexity with Single-Cell and Spatial Multiomics. So I guess we can all agree that human biology is extremely complex. It's been reported there's roughly 40 trillion cells in the adult human body. Each cell has its own unique transcriptome profile, and these cells form the tissues, in turn form organs. And it's often this complexity that limits our understanding of human disease. And in order to resolve biological complexity, you actually need the best technology out there to give you the most insights. So at 10x Genomics, other than being a leader in single-cell technology, we also have taken a lead in the spatial field, introducing two different technology, the Visium Spatial and Xenium In Situ. So over here, just give you a very brief high-level overview of these three different technologies that you see here. So on your left, we have the Chromium single cell. It's a sequencing-based technology that allows you to look at the whole transcriptome down into a single cell scale. We have the Visium Spatial on the other hand, another sequencing-based technology that allows you to interrogate the whole transcriptome in the human and mouse samples. And we now have samples that could go down to the single cell scale. And Xenium In Situ is another spatial transcriptomic platform. It also combines the elements of single cell and Visium Spatial, giving you a very precise insight. So it's a targeted spatial technology that we actually have now assays to target up to 5,000 genes in the human and mouse. So it doesn't require any sequencing. It's an imaging-based technology, and the resolution is down to the subcellular resolution. So at 10x Genomics, one element that's unique to 10x is that all these three technologies, right, that we provided and supported, they are highly synergistic among one another. So meaning that they have shared sample compatibilities, the workflows are compatible, and more importantly, the data sets that generate from Chromium single cell, Visium Spatial, or Xenium In Situ can easily be integrated to give you a very rich biological insight into your tissue of question. So we had actually demonstrated these through a publication last year in November 2023. So in the high resolution mapping of a tumor microenvironment using the single cell, Visium Spatial, and Xenium In Situ analysis. So 10x is not just a solution provider. 10x is also a software provider. So softwares provided or supported from 10x are always free to download. So you're always free to go to our resource page to download these softwares. For our Chromium single cell and Visium Spatial, we have the Cell Ranger and Spatial Ranger for your analysis needs. We also have the Loop browser to visualize your data sets.
For Xenium In Situ, we have the onboard Xenium Analyzer tool. We also have the Xenium Explorer to allow you to visualize your Xenium In Situ data sets. And more importantly, for those who are not very familiar with script writing, bioinformatics, right, those who don't have any bioinformatics background, you can actually make use of our 10x Genomic Cloud. So we recently released the 10x Cloud to the APAC region, so you can actually analyze single-cell data sets without any of this bioinformatics background. So basically, you upload the raw sequencing file, which is a FASTQ file, to the cloud, and you automatically process and analyze your data set. So at the output, right, you have the file ready to be visualized using our Loop browser. So 10x is also compatible with long-read sequencing platforms like Oxford Nanopore and PacBio. So we partner with these long-read sequencing companies to allow users to look at more unique biological questions like structural variants, isoform detection, SNP detection. So these are all possible. So do check out these products with our partners over here. So I'm going to dive a little bit into Chromium single cell. So for those who are not familiar to single cell, I just want to give give a background why single cell is important. So in a very traditional way of doing RNA sequencing, so in a bulk RNA sequencing scenario is where you take a bunch of tissues or a group of cells, you munch them up, you get the RNA, perform RNA-seq. And this gives you some data sets. So if you compare control to treated, you do see some differential gene expression, but it also leads to a problem where the gene expression is actually coming from the average of all the cells that's combined in your tissue sample. So this leads to a problem because the cellular heterogeneity is masked. Whereas for single cell, we understand that every single cell has its own unique transcriptome profile that you see over here. And when you actually plot them in a 2D plot that you see here, so every single spot you see here is a single transcriptomic profile of one single cell. And you can see some cells tend to cluster together. So you see on the left of that graph, purple spots cluster together, suggesting they have similar transcriptome profile compared to a cluster on your right, the green clusters, suggesting they have different transcriptome profile. And with these, you can actually delineate what kind of cell types are present, and they have the ability to dissect that cellular heterogeneity. So through single cell analysis, since the introduction of single cell analysis into the research field, it has already been transforming our understanding of diseases. So a very strong implication in cancer, in autoimmune disease, in neuroscience, and also in other fields like retinal biology, cardiovascular research, and even in non-model organisms like fish, zebrafish, insects, and even plants. So at 10x, we actually invest heavily in our products. So when we first launched our commercial product in 2015, over the years, you can see that there's more and more capabilities, more multi-omic solutions being added to our portfolio. So at 10x, we have invested up to more than $1.5 billion to date. There's more than 10 different applications in our Chromium solution, more than 7 different product versions. We have 5 major breakthrough products, and you have the ability to analyze up to 8 different analytes from the same single cell. So every time we release a new single cell gene expression assay, we surpass ourselves in terms of data quality. So you can see here on the left is the sensitivity plot of the number of genes or transcript detected per cell, right? So you can see that since 2016 to 2018 to now, 2024, our sensitivity improved tremendously over time. So you can see that from a previous slide, there's a lot of different analytes or different assays available for 10x portfolio. So we kind of categorize our Chromium solution into 5 main categories you see here on the left. So we have the traditional 3' gene expression assay solutions. We have the immune profiling 5'. We also have an assay to look at fixed tissues, FFP tissue at single cell level. We have assays to look into epigenetics at a single cell, so through our ATAC single cell assay. We also have a multi-omic solution that combines ATAC and 3' from the same single nucleus. So a lot of people around the world has been using our products to do cell atlasing. So on the right, you see here is a cell atlas of a human brain, a third trimester fetal brain. Different cell types are in this particular experiment. So 10x has a regional office based in Singapore, and we also have a local team, the sales team and support team based in India as well. We're also supported by our distributor, our Genex. So we have a booth out there. So if you have any questions, feel free to check us out. So at 10x, we have a very comprehensive resource online. So in terms of user guides, protocols, technical guides, right? So all these are available to download from our website. So at 10x, we also have the broadest range of application and analytics. So I just taking this 5' immune profiling solution as an example. So over here, you can see that from one single cell, we have assays not only looking at the gene expression, we have assays to look at the immune profile solution, so immune sequence repertoire of the T cells and B cell receptors. We have assays to look at cell surface proteins. We also have assays to look at, functional genomics like CRISPR screening, right? So all these at least 5 or 6 different information coming from one single cell is actually possible from 10x Genomics. So using a multi-omics solution to understand disease is actually not uncommon. So during the COVID-19 pandemic, there's been multiple papers that published using a multi-omics solutions. So here on the left is a Cell paper published in 2021 where they profile up to around 1.5 million cells from roughly 200 donors from people suffering from different severity of COVID disease and also with healthy controls. They perform the 5' gene expression. On top of that, they also look into the VDJ region of T cells and B cells. And another paper published somewhere around the same period in 2021, Nature Medicine, they also did something very similar. And at this time, they also add in additional information, which is the cell surface protein. So 10x recently released the latest technology for our single cell solution, which is called a GEMX solution. So with GEMX, you are now going to profile more cells, so up to 20,000 cells per sample. So you can run up to 160,000 cells in a single chip run in less than 6 minutes, profiling up to 8 different samples in parallel. So we have a very high cell recovery efficiency, up to 80% of your cells can be recovered for single cell analysis. We also reduce the doublet rate to 0.4% per 1,000 cells. So one very interesting and important feature about GEMX is that you can actually now profile very fragile cells like granulocytes. So over here, between the left plot and the right plot is between the 2 different chemistry. The GEMX is latest on the right. If you look at the purple cluster, these are the neutrophil cells. So neutrophils are known to be very, very difficult to be captured because they are very fragile. They have low RNA content. They have very high RNAs in them as well. So with GEMX, because of the improving chemistry, we partition them better. We can capture them better. So we are going to see a significant improvement in capturing these granulocytes in your samples. So here is the 3 main solutions. So over here, the 3' traditional gene expression with the 5' immune profiling on your right. They are species agnostic, so if you work on other species like plants, zebrafish, or any other species from human mouse, you can actually make use of these solutions for your single cell needs. And the other solution is about gene expression flex, which I don't have time to talk about, but this flex assay is a different you to fix your cells, store your samples in your -80 freezer, and then batch them out and perform single cell analysis. So you do not need any viable cells to do your single cell analysis. So I want to also introduce the entry-level Chromium XO. So this is the platform that allows you to do single cell. It comes in at very affordable entry points, can be priced compatibly. It can be as cheap as a simple qPCR machine, or even cheaper than a qPCR machine. So if you're interested to know more about this, do speak to Alf at our booth. So I'm going to move on to the next part of my talk, which is the Visium Spatial. Right, so I've talked about traditional bulk RNA sequencing. You see on the left, Chromium single cell allow you to delineate cellular heterogeneity in samples. But a lot of times when people perform single cell, they actually want to know where these cells are located, especially in the tissue context. So this is where the Visium Spatial can come in to fill in this gap. At 10x, we are not new to spatial. We actually been in the spatial field since 2019. It's about 5 years from now. And since then, there's been an accumulation of more than 400 publications that using our Visium products, right? So a lot of them has very notable, impactful discovery you see on the left, and some customer even innovate and hack our system to answer unique questions in their in your experiment and research. So Vision HD is the latest from 10x Genomics. So we announced this a few months ago. So it's a whole transcriptome-based technology to look into your human or mouse genome. 6.5 by 6.5 capture area, 2 micron barcoded square. So we have actually released a new protocol that allows you to get a fresh frozen and fixed frozen on top of the FFP tissue that you have in the lab. So we also enable both both H&E image or immunofluorescence staining on the same tissue section on top of the transcriptome profile. And we also provide software for analysis and visualizations. So just to give you a perspective of how Vision HD is going to be like. So over here is to show you our previous version of spot-based transcriptome technology, 55 micron in diameters with gaps in between. With Vision HD, you can really appreciate that the higher resolution you can get with your spatial transcriptomic data sets. And if you actually zoom into the region on the bottom right, you can actually appreciate that the Vision HD can go down to the single cell scale. So 10x also encouraged people to integrate single cell into our Vision spatial data sets. So on your left is human FFP colorectal cancer tissues, unbiased clustering HD data sets. And on your right is where we actually project the single cell data sets onto the Vision HD data set to see unique biological features. For example, over here, we can look at the tumor score, look at how the tumor progress in this particular sample. So the next technology is Xenium in situ. So we kind of launched and shipped this in 2023 last year. And to date, I think we have more than 250 units installed globally. So over here are some of the very enthusiastic kind of chatters in the social media. And already we have actually more than 20 to 30 publications that uses the Xenium in situ for their spatial needs. So Xenium in situ is a little bit different from Vision Spatial that I talked about earlier. So it's an end-to-end platform, meaning that everything is provided from 10x, including the reagents, the slides, the instrument, and software. It doesn't require any NGS, no sequencing involved. So we have now access to dive into the 5,000 genes in the human and mouse transcriptome. Xenium has a very large analyzable area. So you can see that it's a 2 cm by 1 cm. So this is how big the tissue you can place. You can work on tissue microarrays, the mouse brain section, for example, here. So here it's the pre-designed panels that we have. So some of very popular design panels that we design over here. You can add on custom genes. So we also allow you to actually perform up to 480 custom genes for Xenium in situ. So we have done a lot of our tests. So here is just one single mouse pub that we perform on one single Xenium slide. You can see that it has a very broad tissue compatibility, allows you to resolve biological complexity at scale. So I think I'm at time. I will quickly just give me 1 moment. So we actually launched the 5K panels early, just a few months ago. So these are 5,000 genes covering all these different parameters over here. And I'll just go into this slide. You can actually appreciate Xenium in situ allows you to do automatic cell segmentation. So on the top right-hand corner, you can see that fibroblast cells identified by this marker interacting with 2 Treg cells. And the bottom here is a macrophages interacting with T cells over here. So this is my last slide. So I just want to end here that we are very encouraged and motivated to see that a lot of these products that we supported having very impactful in your research. So to date, we actually have more than 8,000 publications that uses 10x products. And we hope that people around here can continue to support us using our 10x products. So we are distributed by Genex in India. So if you have any questions, please feel free to reach out to our booth. I'll be happy to take on any question. Thank you.
Our thanks to the team of 10x Genomics. We now move to the next segment of our event, where we have 3 parallel sessions happening simultaneously in different halls. For those of you staying in the main hall, we have an exciting session lined up titled as Inflammatory Responses. For those attending the parallel sessions, you may now proceed to Hall A or Hall B. Please refer to the event program for specific details on topics in each hall. The main session will continue here in the main hall itself. We request all the speakers to kindly adhere to the allotted time for the presentations. I would like to invite our session chairs, Professor Nibriti Das and Dr. Annapurna Vyakarnam, to take over and lead the discussions. I request everyone to kindly settle down as we are going to start our next panel session. The chairs will introduce you. Okay.
I can start. She will. Okay, so we are having an interesting session with 3 speakers. Only thing I will request the speakers. So don't take the full 20 minutes, take a little less so that some discussion can happen. Okay, so Dr. Annapurna will further conduct.
So good morning. Our first speaker is Dr. Sandhya Ganeshan from IISc Thiruvananthapuram, and she's going to talk on innate and cell-autonomous immune response against vacuolar pathogens, lessons learned from Coxiella burnetii.
on October 18th. I encourage you all to visit her postal. We'll be thrilled to receive your feedback. I undertook a secondary screen where the idea was that in the presence of no interferon gamma signaling, this is the coxiella containing vacuole called CCV, where coxiella is growing to high numbers. If we add interferon gamma, as we saw earlier, the vacuole becomes very constricted with very few bacteria in it. If we supplement the cells with excess tryptophan, thereby negating IDO1 activity, we are able to partially rescue this replication. And what if we now negate IDO1 activity? In addition, we again knock down various interferon gamma induced genes to identify additional factors that mediate coxiella suppression. So through the secondary screen, we identify another gene called syntaxin 11, which is a snare protein, which also negatively regulates coxiella replication. If we were to knock down IDO1 and syntaxin 11 together, which is this checked bar over here, we see a much higher rescue in coxiella replication. In our lab, where we are looking as you can see in the nucleus on the side, to generate such a spacious compartment over several days of infection, it should be engaging in extensive vesicular fusion events. So this has now led us to understand more about snares, syntaxin 11, as well as other snares, and how they regulate the biogenesis of pathogen-containing vacuoles and how that affects bacterial replication inside those vacuoles. So this is syntaxin 11, the top band over here. This is also an interferon gamma-induced protein, and we generated syntaxin 11 deficient cells. We see that in the absence of syntaxin 11, which is the green bar over here, coxiella replicates to much higher levels in infected cells. And in addition to replication, in the absence of syntaxin 11, we in general see a much bigger sized vacuole with higher numbers of coxiella inside these vacuoles, as shown by here. This is the coxiella vacuolar area, and quantification of this area shows a significant increase in absence of syntaxin 11. We are now at the stage where we are piecing together different known and unknown functions of syntaxin 11. What we found was that syntaxin 11 is a very specific snare that is highly upregulated by interferon gamma. Not all, most vesicle traffic proteins involved in such vesicle fusion and vesicle traffic are not immune regulated. But syntaxin 11, as we see, is induced by interferon gamma signaling. And a very well-known function of syntaxin 11 in the case of NK cells and T cells, cytotoxic T cells, is that syntaxin 11 localizes onto the plasma membrane and it mediates the fusion of lysosome-derived vesicles, which release lytic granules into the immunological synapse.
You
so
need to wrap up.
Yeah.
Sorry.
Right. So I'll just finish up this slide. And what we have now been looking at is syntaxin 11 polymorphisms lead to an immune disorder called HLH and macrophage activation syndrome. And we have now collated all the polymorphisms that are available in literature. We have created these polymorphisms, reconstituted syntaxin 11 deficient cells with these variants. And we are now trying to look at both the role of syntaxin 11, how that is relevant in immune disorder, as well as why that affects bacterial replication in the case of Coxiella benetiae. I will skipped a couple of slides. I want to thank all the people who were involved in this work, all the lab members, some of whom a few more graduate students who were involved in this work, and finally all the lab members, the collaborators who work with us, Karthik from ICTVM, Karthik from RGCB, as well as Dr. Amituli and Dr. Lipitukral, my mentors, postdoctoral and graduate mentors, as well as our collaborators who we seek feedback and reagents from, our extensive funding support. Thank you again, IMMUNOCON, for providing this platform for us to share our work. Thank you very much.
Thank you very much. So we have time for a few questions. A couple there. A couple there.
Very good work. And it has a tremendous translational potential. Are you looking at the mechanism of survival or how to kill the bacteria in the organism? Because the more you look, I will repeat, extremely good work.
Thank you.
And I think you should be continuing this type of work more. Survival mechanisms we have been studying how many years? So your organism may not be as dangerous as Mycobacterium tuberculosis, but nevertheless it is important. Nature has produced a lot of molecules which can be antibacterial, and we have not even started looking at them very carefully. Are you planning to do some work where you are combining your mechanistic study for the survival of the bacteria and also use of some of these potentially, like berberine? I was reading, you see, I don't have a lab now anymore, I don't do research, but I advise. Berberine is a very important molecule with antibacterial activity, and this is worth testing in your system. Thank you.
Thank you, sir. Thank you very much. So this is also in our radar to test various antimicrobial compounds and how that can be effectively used to target intracellular bacteria, because bioavailability in these compartments can be very tricky. So I would love to discuss more with you and seek feedback. Sure.
And I will discuss.
Thank you, sir.
One more.
Do we have another question there? No.
Hi, Sandhya. Shashank from IISc. Very nice talk. So IDO1 is part of this kynurenine pathway, one of the many enzymes in that pathway. Have you looked at other enzymes? Are they activated by interferon gamma or other interferons?
So no, actually, we have not looked at immune inducibility of other enzymes in this pathway. But RNA-seq type of studies have shown what are all the immune inducible genes induced by interferon gamma, and they don't feature at least in the top few hits. IDO1 is there, and after which comes proteins like GBPs, CXCL10. So it didn't strike in our radar because we were looking at some of the top induced genes, but they may very well be, but that's an interesting thing.
So third question there.
So excellent stuff, Sandhya. The question is regarding, Coxiella is staying in the acidic lysosomal compartment. I wonder if you have looked at, or someone has looked at, whether it also tolerates redox in the lysosome, and whether your syntaxin 11 is actually involved in bringing redox mediators to the lysosome, and in the knockdown condition or depleted condition, you see better bacterial growth and survival.
Thank you. So for the first question, whether how Coxiella regulates this redox environment in the lysosome, there are enzymes known to be encoded by Coxiella, which are superoxide dismutases. There are enzymes known to be encoded by Coxiella, which are supposed to make this environment more favorable and tolerate the redox conditions. And in terms of finding how syntaxin 11 may affect this, we are looking into sensors that we can use to see whether in wild type or in knockout cells, whether it is different.
I think looking at mitochondria might help you.
Mitochondria?
Because it's possible that, certain mitochondrial vesicles with redox are being driven to lysosomes, and syntaxin 11 is involved in that.
That would be great. I will discuss with you further about this.
Sure.
Thank you. Thank you.
No, thank you very much, Dr. Ganeshan. Fantastic talk.
Thank you.
Thank you.
Thank you.
Next speaker is Dr. Taruna Madan. She is from Innate Immunity Group, ICMR, and she will speak about her work along with the title. Just to saving some time.
Okay. This is what there is. Okay.
So time starts now.
Thank you very much, ma'am, and it's an honor to be speaking in front of you in a session chaired by none other than Professor Nibriti Das and Dr. Annapurna Vyakarnam, whom I have started knowing recently after my move to ICMR headquarters. And I would be sharing our work on two recombinant molecules that we have evaluated for their translation potential in preeclampsia, which is a major cause of maternal mortality. And the work has been carried out at National Institute for Research in Reproductive and Child Health, Mumbai, where I was heading the laboratory of innate immunity. So I would like to thank Ms. Hajra Gupta, who is there in the audience today. And this is majority of this work is done by her towards her PhD program. And Dr. Ram Bahadur Subedi, who was an ICMR research associate in my lab in Mumbai, who is now a senior official at R&D division of High Media. Preeclampsia is a pregnancy-specific hypertensive disorder that affects 3 to 6% of pregnant women. It's a real, I'll say, fatal disorder, which results in both harming the mother as well as the fetus. The clinical features being the high blood pressure, you have proteinuria and high creatinine in the urine samples, you have puffing of feet, hands, and face. And we also see thrombocytopenia, impairment of liver functions, pulmonary edema, cerebral or visual disturbances. And in those cases which are very severe, the women can go into eclampsia, which is kind of an epilepsy attack. Long-term health risks are there to both PE mothers and babies, and you can see it here. Hypertension, renal dysfunction, venous thromboembolism, CVD, diabetes, dyslipidemia, depression, and preterm birth definitely is one of the outcome of preeclampsia. And you can see here another list of cerebral disorders, which are, and the ocular neuropathy, which are really affecting both the mother and the infant who have suffered through the preeclampsia. And these are the long-term health risks that they carry. And that's why it is very important to prevent preeclampsia. And for that, it is important to understand the implantation process. So we all know that the fetus is a semi-allogenic component, and the mother is trying to normalize her immune response or to regulate her immune response to enable this implantation happening of the embryo in the uterine wall, and which is followed by this spiral artery development, which is leading to a supply of increased blood supply to the increased blood supply with oxygen as well as nutrients to the fetus, and that's how the fetus gets all the necessary nutrients to grow. And here you are able to see that these are extravillar trophoblasts, EVTs, and this is the intravillous space, and this is the villi, and you have these outermost cells called syncytiotrophoblasts. So these are various kinds of cells that are present at the feto-maternal interface. So in the preeclampsia, there is failed spiral artery remodeling. As you can see here, the artery lumen for the spiral artery is huge here, and here you see a narrow lumen. And this narrow lumen is a characteristic hallmark of preeclampsia, especially the early onset preeclampsia, which is the more severe form of preeclampsia. To brief you about the immunology of preeclampsia, it is characterized by both reduced angiogenesis as well as immune tolerance, and you have decreased number of Treg cells and increased numbers of cytotoxic CD8 T cells in the preeclamptic pregnancy vis-a-vis the healthy pregnancy. So there is a systemic balance in preeclampsia. You can see the angiogenic imbalance. You have increased levels of SFLT1, which is the soluble marker of VEGF. You have soluble endoglin. You have TIMPs, which are tissue inhibitors of metallic proteinases. You have epithelial integrins, ICAMs and VCAMs, the adhesion molecules. And you have low levels of the growth factors, vascular endothelial growth factor, placental growth factor, TGF-beta, 1 Willebrand factor, MMP9, MMP2, and endothelial integrins. And on the other hand, there is immune imbalance, and you can see here pro-inflammatory cytokines, Th1 cells, M1 macrophages, TLRs, C1q, C3a, all going up, whereas the anti-inflammatory cytokines are going down, and Th2 and Tregs are going down. We have lower levels of M2 macrophages and decidualized NK cells at the feto-maternal interface. For management and treatment of PE, we have antihypertensives for management of blood pressure. We have corticosteroids that ameliorate the liver function issues and maturation and address maturation of fetal lungs. You have anti-inflammatory drugs used in the management of edema and inflammation. To tell you more importantly, that low-dose aspirin has been proven to be effective in preventing preeclampsia if started early on in these high-risk women from the week 12. And you have anticonvulsives for prevention of seizures. As I mentioned, when it is uncontrollable PE, it can become eclampsia. And you have antithrombotics also for elimination of clots. But these are all symptomatic management, none of them helping us to actually address the cause. The cause is addressed only post-delivery. When the fetus is delivered, the mother is able to regain her normal blood pressure, and that is the solution. But only thing is, when you have preterm babies, it is so difficult to manage these mothers and women from the preterm adverse effects. So here you have promising treatments for preeclampsia, the angiogenic therapies. So VEGF, PLGF, siRNA therapies for SFLT1, PPI, statins, they have all been tried in the rat models, different rat models, and as well as their levels have been evaluated in different trimesters. And we have seen that the outcome is that, yes, you do see downregulation of the angiogenic inhibitors, and the limitation is the toxicity owing to these, there are cardiovascular risks, there is a risk of increased permeability of the blood vessels when you give angiogenic promoters for therapy of preeclampsia. You do have immunomodulatory therapies, glibenclamide, polarized Treg cells, TNF-alpha inhibitor, metformin, Junbi inhibitor, ghrelin. They have all been tried. These are addressing various targets in the immune system, and in the animal models, they have been evaluated, and at different trimesters, they have been evaluated, and they have shown, some of them have shown promising impact. We do have antioxidant therapies working for preeclampsia. We have complement inhibitors working for preeclampsia, and there are certain others such as folic acid, carnitine, magnesium sulfate, and low-dose aspirin, as I mentioned earlier on. Low-dose aspirin is also acting as an anti-thrombolytic agent. So features desired in potential therapeutic agent for PE are a factor that can reinstate the angiogenic potential of trophoblast. It restores the balance of pro-angiogenic, anti-angiogenic factors and pro-inflammatory, anti-inflammatory cytokines, eliminates oxidative stress, improves kidney and liver function, contributes to fetal benefits, and alleviates the maternal syndrome. Basically, if we can give these therapies early on, we would like to see a more effective spiral artery remodeling in these high-risk women, and that is what low-dose aspirin is helping at. So home house is the best treasure. Start your hunt from within. So we were working on colectins, C-type, collagenous lectin, surfactant protein D, which is a power ranger of innate immune system. It's a calcium-dependent collagen-containing C-type lectin. It is secreted by type 2 alveolar cells in the lung. And then later on, it was discovered by our group as well as others that it is also present at the feto-maternal interface and is secreted by both syncytial trophoblast as well as trophoblasts. It is not only expressed by placenta and uterus, it is also expressed by mammary glands, prostate glands, and testis. It is the expression in the uterus is regulated by estradiol, and P4 was shown by our group, Dr. Sharon Kay, who is now a professor at one of the prime colleges in Mumbai. Our contribution in elucidating the role of SPD in PE, positive correlation, serum levels of SPD and P4E2 ratio, and I'll show you the graphs in my next slide. We had differential serum levels of SPD before and after onset of preeclampsia. We knew that RFH-SPD is modulating M1-M2 ratio. RFH-SPD decreased the TNF-alpha and IL-6. These were ex vivo cultures of trophoblasts derived from preeclamptic mothers treated with RFH-SPD, and that's how we saw these effects. And we are actually exploring and validating potential predictive RFH-SPD as a potential predictive biomarker of PE in one of the ICMR-supported grants. In the mouse model, we had access to SPD knockout mice thanks to Dr. Jefferson in Cincinnati Children's Hospital in US. He very kindly send us 2 pairs of these knockout mice, which we have bred in our animal house and are using for various studies. These SPD knockout mice, when they became pregnant, they showed low placental weight, they showed increased mean arterial pressure, high levels of TNF-alpha and low levels of IL-10, low levels of VEGF, placental growth factor, MMP2, high levels of TIMP2, which is the tissue inhibitor of metalloproteinase 2, but they showed normal kidney histology with no proteinuria. So this is a limited model of preeclampsia and symptoms that we see in the SPD knockout mice. Importantly, Sharon had also shown that these mice have problems associated with their implantation, and we had, if say normal mice gives rise to pup size of 9, we were having only 3 to 4 pups from these SPD knockout mice. So it was a difficult assignment to maintain and breed these mice. All these observations showed us that SPD is a potential immunomodulatory candidate in PE. It suppresses immune cell activation at maternal-fetal interface, decreases the production of anti-inflammatory cytokine, controls expression of angiogenic factors, increases the anti-inflammatory M2 macrophages, and hence is an ideal candidate to work upon. Another important factor that I would like to elucidate too is the presence of hypoxia in the placental milieu, which is required for the embryo development. And that is how our experiments were done in hypoxic conditions, as shown here. This is the in vitro experimental setup with which we are maintaining the hypoxic conditions during our experiments. So the first experiment was to see whether the cells are viable in presence of hypoxia using MTT assay, and these are HTR8 SV neo cells. We got them from Professor Satish Gupta at NII, who actually had gone them from the person who had created these cell lines. These are first trimester human trophoblast cell lines that are being propagated and in our lab. And we saw that when we are treating them with recombinant fragment of human SPD, we are seeing that there is a effect on the mortality. In 48 hours, at 24 hours, they were more or less stable at with 10 micrograms of concentration, 10 microgram per ml concentration. But 48 hours, we were seeing that these cells are dying. Yes.
Wind up first.
Sure, ma'am. Sure. So we saw that there is apoptosis happening in these cells, and we at the 48-hour time point, but it maintained cell viability at 10 microgram per ml for 48 hours in hypoxia. We saw that it induced a pro-angiogenic effect, as you see in the 2 bars at the end. VEGF, PLGF are going up, SFLT1 going down, TGF-beta going up, and endoglin going up, as well as MMP9. So these were all very heartening observations, and we saw that it is maintained, the pro-angiogenic effect is maintained even at the 48 hours. We saw it at the protein level as well. So these were, the previous results were from the mRNA transcription, and then we have now the protein level confirming the data that RFHSPD induces a pro-invasive EMT effect on HTR8 cells under hypoxic conditions. It induces an anti-inflammatory effect.
Time up, Taruna.
Sure, ma'am. Thank you.
It's open for discussion. Maybe I could just ask a brief question, Dr. Taruna.
Sure.
Is it understood? I mean, it's fantastic, elegant work. Is there any understanding as to how SPD is being shut off so early? What is inducing the shut off of SPD?
Sure, ma'am. As I mentioned on one of my slides, that there are hormones that are regulating. So this is a C-type lectin receptor, and it has steroid responsive elements in the promoter region, and that is the hormones are regulating the expression, and we know the hormonal changes that happen at the feto-maternal interface during pregnancy. Thank you.
Thank you. Are there any other questions? Yeah, please. Yeah.
Yeah. You know that my comment is, it is almost becoming impossible to understand the maternal fetal interaction in almost everything. You see, she's talking about a specific condition. We have been working together for quite some time now about pregnancy. Is the children who are born of a particular infected individual become tolerized to that parasite, as a result of which those children, when they grow up, they become non-resistant, they become tolerant to those organisms, like filariasis, on which I did some work. Tuberculosis now, malaria, mother-fetal interaction should be done for a translation potential. She's doing on a particular disease, but I don't want to mention about curcumin because that is where I work. And curcumin is doing wonderful things, she will herself tell you. So I would encourage more work in this area, because this is what is going to determine the health of our next generation.
Yeah, absolutely. I think more work in the mother-baby area. Absolutely.
Thank you so much.
Thank you very much.
Thank you. Thank you.
Hello.
Okay.
Hello. Ma'am,
Is there a...
I have a question.
Oh,
Yeah, this side.
sorry. Is there a...
Ma'am, this side, the middle rows.
Only the seniors talking.
Hello. Good afternoon, ma'am. This is Pratyusha. So I saw thrombocytopenia on the slide, and I just got curious to know the thrombocytopenia that occurs in PE, is it autoimmune or non-autoimmune? And in case if it's autoimmune, is it secondary or primary or secondary to PE? Because in thrombocytopenia and autoimmune diseases, it has been found that the menstrual cycle and the hormonal cycle affects the vulnerability to autoimmune conditions, and mostly puberty and menopause and pregnancy. These are the states where women are more, vulnerable to such conditions.
Thrombocytopenia, and I may not be able to address your query right now because we have just started working on these effects and also the effect of complement factors on the preeclampsia. So maybe in my next IMMUNOCON, we will definitely share these data.
Excellent work and excellent presentation. Taruna, there may be many more questions. So in the off time, lunch time, you can have enough time to interact. Okay, thank you so much.
And I would like to tell you that Hajra is presenting her poster, which I was showing on my last slide. She will be better explaining all the things because she's the one who has done this.
Thank you very much. Our next speaker is Dr. Shibnath Mazumdar from the University of Delhi, the Immunobiology Laboratory, Department of Zoology in the Faculty of Life Sciences and Biotechnology. And he's going to be talking on TLR22 signaling and mitochondrial dysfunction, implications in bacterial pathogenesis. Is he online? Is he online? Is he online? No.
Good afternoon, everybody, and thank you for being patiently waiting over here. And I'm really sorry because I'm standing between you and your lunch. So please be patient. And main thing is that I'm just not here to present my work, but also to sell fish, not as a food, but as an alternate model to study, to pursue immunological tests and questions. So the data that I'm going to present over here is basically, it is unpublished. So there are a number of dots, and with your suggestions, I think we will be able to fill up most of them. So briefly, we all know about PRRs and PAMs. The innate immune system recognizes the PAMs with the PRRs, and there are several groups of PRRs, which includes TLRs, CLRs, ALRs, and many more. And sensing of the PAMs by PRRs upregulate the transcription factors, which includes NF-kappa B, IRFs, and kinases. And this is a brief structure of the TLR, and we all know that these are evolutionary conserved, and they act as primary sensors for invading microbes. And I'm not going to go into this because this tells you about the TLR signaling. Now, I want to just seek your attention to this slide for a minute. You all know that humans have 10 TLRs and mouse has 13. But when you look into this slide, there is a whole lot of TLRs, especially those which I have marked over here with red. These are the TLRs that are not present in the mammalian system. These are present in the non-mammalian system, the non-mammalian vertebrates, which includes fish. And I will be talking primarily on TLR22. So TLR22 is a novel non-mammalian TLR. It has been reported in several fish species. We reported it in Clarias garipinus. Previously, it was thought that this is a functional substitute to TLR3, but later on, it has been recognized as a crucial sensor in innate immune signaling against both bacterial and parasitic infections. And interestingly, this TLR22 is present in immune and non-immune sites in various fish species, suggesting its functional plurality. So at present, we really do not have much idea about the location and the ligands that are recognized by TLR22. The other character that I'm going to introduce to you today of my story is Aeromonas hydrophila. Aeromonas hydrophila, as you see, is a gram-negative rod-shaped bacteria. It is one of the most ubiquitous bacteria. And interestingly and importantly, it infects a huge range of hosts, which includes both cold-blooded and warm-blooded vertebrates. It can also infect invertebrates too. What it causes is primarily septicemia and gastroenteritis. And in human, it is associated with septicemia, gastroenteritis. And previously, it was considered to be an innocuous pathogen, but of late, it has been included in the list of emergent pathogens of concern. And we really do not know much of the pathophysiology that is induced by this bacterium. So we have a bacteria which is infecting humans as well as fish, and we do not know much about the pathophysiology that is behind the pathogenesis of this bacteria. So the question is, should we take a fishy route to understand the pathophysiology of Aeromonas hydrophyllum? So if you look over here, especially for the students, if you look into this slide, you will see that all the hallmarks of immune system that you see in a mammalian system is present in fish. Particularly when you look at bony fish, it has thymus, it lacks bone marrow. Now this absence of bone marrow, whether this is a gastronomical problem for us Bengalis, or it is something related to immunology, that I can't say at present. But if you look and compare bony fish and mammals, you see everything that is there in mammals is present in fish. Just look into this slide and see all the things that you see and study in mammalian system is present in fish. The immunoglobulins are there, AID is there, class switch, affinity maturation, TCRs, MHC1, 2, except for germinal centers and lymph nodes. But of late, it has been suggested that germinal centers in some form or the other are present in fish. So this again tells you that fish could be a very interesting and alternate model to study immunology or to perform immunological tests. And this picture that I've kept over here is for the students to make out which one is a mammalian macrophage and which one is a fish macrophage. So see how similar they look like. And functionally, I've mentioned over here, they have much similarity in the way they function. So the first one is the mammalian macrophage and the last one is the fish macrophage. So what we can say is that fish appears to be a very good alternate model. Now, again, this is not the fish that we study, zebrafish. This is catfish, which we have tried to develop as an alternate model to look for immunological answers, questions to immunological questions. Now, you see over here, when we studied the expression of TLR22, nothing was known about TLR22. We saw that TLR22 is expressed in almost all the tissues. When I say all tissues means both immune and non-immune tissues, but it is maximally present in head kidney. What is head kidney? It is something like the spleen that we have in the mammalian system. So that is an immune organ, but there you see in muscle, it is there, but it is present in insignificant proportion, which suggests its functional plurality. It might be involved in immune as well as non-immune functions. Now, the next thing is that when we infected the fish, the first one is the in vivo data, and the second one is the in vitro data, we saw that TLR22 expression is significantly increased following Aeromonas hydrophylla infection. Now, when you look into the head kidney macrophages, when they were stimulated in vitro, you see a biphasic response. Though this is real-time data, qPCR-based data, but our Western blotting also suggests a similar biphasic response. Now, very interestingly, when we did this with another fish pathogen, which is Mycobacterium fortuitum, which is a non-tuberculous mycobacteria, there we don't see this biphasic response. So this again raises a question that does fish respond, or does fish TLRs respond in different ways to different pathogens? We don't know, but that we are really interested to study. Now, the question is that what role TLR22 is a TLR, it is a PRR. So what role it might be having in the pathophysiology that is induced by Aeromonas hydrophylla? So what we questioned was, does it play a role in phagocytosis? You can see over here, you can see over here that there is hardly any role of TLR22 in phagocytosis. But when we silence TLR22 in panel C, what you can see over here is that the intracellular growth of Aeromonas hydrophylla inside the fish macrophage is severely compromised. So this tells you that TLR22 is not involved in phagocytosis. It is involved in containing the bacterial growth. And then we asked that what happens to the infected cell? Does the infected cell gets affected by some TLR22 functioning? And we can see over here that when you silence TLR22, the head kidney macrophage death is compromised, which says that TLR22 is responsible for inducing the death of fish macrophages that are infected by Aeromonas hydrophila. And then we questioned that, well, the cells are dying, so what is the nature of cell death? And the whole panel tells you that TLR22 is primarily inducing the apoptosis of head kidney macrophages. So we did all the studies. We have the EM and other studies, but you can see over here that these studies conclusively suggest that TLR22 is involved in inducing apoptosis of A hydrophila infected head kidney macrophages. Now we started raising questions that how does TLR22 induce the apoptosis? And our previous studies suggested that calcium was playing a very important role in the induction of apoptosis. And we also observed that ER mitochondrial crosstalk, the imbalance in the intercellular cytoplasmic level, was in a big way influencing the apoptosis of head kidney macrophages. So what we did was we asked over here is that does the calcium that is coming out from the ER or from the cytoplasm move into the mitochondria, trigger a mitochondrial ROS, which in a way is responsible for inducing cell death. So as you can see over here that YCG is a mitochondrial ROS inhibitor. So what we see over here is that when you inhibit TLR22 by siRNA, you see that mitochondrial ROS level is decreased. And when you look over here, the calcium that is coming out from the endoplasmic reticulum goes to the mitochondria through the mitochondrial uniporters. So when we look over here, this is ruthenium red, which is a mitochondrial uniporter inhibitor, as well as we have the TLR2 siRNA. You can see that mitochondrial calcium level goes down. And at the same time, when you look into panel B, you can see that when you inhibit mitochondrial ROS production, or when you use TLR22, when you silence TLR22, the bacterial count goes down significantly. So in a way, you can tell from here that the mitochondrial ROS that is generated in response to Aeromonas hydrophila infection is playing a pro-host role. It is helping in containing the growth of intracellular Aeromonas hydrophila. Now, the next question we were asking is that how does TLR22 further, help in induction of apoptosis? And the first thing that came to mind was HIF, HIF1 alpha, because TLR is known to play a role in HIF1 alpha induction. So as you can see over here, that HIF1 alpha mRNA level goes high, and when you inhibit with the help of mitochondrial ROS, or when you use siRNA against TLR22, the levels of HIF1 alpha goes down, and that is reflected in that cell death that you see over here. That cell death is also significantly compromised in the absence of TLR2 signaling, as well as when you inhibit HIF1 production. Now, this is another extension of our work where we observed that ER stress is generated due to Aeromonas hydrophila infection. And what you can see over here is that HIF1 is in a way extending the expression of CHOP1. What is CHOP1? CHOP1 is a pro-apoptotic ER stress marker. So a story appears to us is that following infection, there is an intracellular calcium flux. The calcium is released from the ER. It goes to the mitochondria. It triggers mitochondrial ROS. Mitochondrial ROS triggers HIF1 alpha. HIF1 alpha triggers CHOP1 expression. And this leads to the triggering of mitochondrial UPR. We have used HSPD1, which is a protein marker for monitoring mitochondrial UPR. And you can see over here that mitochondrial UPR is taking place. When you block or when you inhibit the TLR2 mediated signaling, you can see that this is going down. And please focus over here that we have also used mitochondrial fission inhibitor MD1. And as a result of this, you can see that mitochondrial fragmentation is also compromised. So mitochondrial fragmentation, what does it lead to? It leads to the dissipation of the cytochrome C, and it alters the mitochondrial membrane potential, and all of them together. How much time do I have? 2 minutes. So I can prepare a Maggie for all of you know. I've got 2 minutes time with me. So just bear with me. And as you can see over here, that when you compromise or when you inhibit SIR, TLR2 mediated signaling, you can see that there is a severe change in the mitochondria and in the extent of cytochrome C that is present in the mitochondria and in the cytoplasm. And mitochondrial dysfunction activates caspase-9 and caspase-3 axis in aromatase hydrophylla. And this is the summary that we have. So now let us see what is happening. So when aromatase hydrophylla enters into the system and it is taken up, now TLR22 doesn't play any role in the phagocytosis. What it does is it triggers mitochondrial calcium influx. Now this mitochondrial calcium influx leads to the increased production of mitochondrial ROS. What does mitochondrial ROS do? It triggers the HIF1 alpha CHOP axis. Now HIF1 alpha CHOP axis, they converge towards they trigger the mitochondrial UPR, and which triggers mitochondrial fission. As a result of this, there is a loss in the mitochondrial membrane potential. Cytochrome C is released, and this triggers caspase-9 and caspase-3 cascade of the caspase-9 caspase-3 axis, and this in turn leads to apoptosis. Now the question that comes to our mind is that is this a pro-host or a pro-bacterial response? So as of now, we think that this apoptosis is helping in the reduction of the intracellular bacteria, as well as it is removing the infected cells. So we think that this entire cascade of events that is triggered in the fish macrophage helps in containing the important pathogen, Aromonas hydrophylla. So this is all that I have for you, and I hope I have finished on time.
Perfect. Absolutely. Thank you very much.
So, if you have any question, I'll be happy to
So
answer.
do we have questions?
Before you ask a question, I ask a question to all of you. Do you think fish is a good model? Do you think fish is a good model? Probably I'm not a good salesman.
I think it's an excellent model.
Yeah, please, if you have any question.
So whilst the audience is thinking, and there's one coming up, maybe I can ask you a question, which is, given that the pathogen is ubiquitous, but TLR2 is restricted, so is TLR22 being protective in some species, and what's the alternative to TLR22 where it is lacked?
We really don't know at this time point because this is not present in fish. But our other studies show that there are other TLRs like TLR2 and TLR4 and other TLRs, which could be playing I shouldn't say could be playing, it is playing a major role in containing the A. hydrophila infection.
Okay, so it's going through other TLRs as well. Yeah, no, interesting. Thank you. There's a question there, the couple. So the first at the back.
Very interesting talk. I just have one question since I also work on the intracellular calcium release. So have you checked the pathway through which after TLR this calcium is influx happens? Like what could be the upstream pathways like status of PIP2 or DAG? And
we have seen those. We have published those where all those pathways are involved. And we have also observed in the fish model that SOCE pathways are also playing a role. But if you ask me whether we could link up all of them, no, we couldn't. Because the difficulty in fish model is that you don't have all the antibodies. So that you can do a pull down assay or you can do anything. And even cloning and sequencing these available in the literature.
No, since this TLR is not present in mammalian system, so in fish, is it following the conventional pathway of calcium release?
Sort of, yes. Which again tells you that TLR signaling is quite conserved across evolution.
Okay, thank you.
Thank you. There's one more question here. Sorry, if we could just wait, there's a second question there and then followed by you.
Is it my turn?
Yeah.
Sorry for
please.
the late response to the question that you asked us. If fish can replace all the other animals, I think it's a great model.
No, it's
Particularly in view of the animals that are being used repeatedly in various research activities.
Yeah, actually,
so
I don't have much time over here,
that's okay.
but I can tell the students, particularly if you're interested, it is a very good model, I can tell you, especially if you want to do cellular immunology. Because with zebrafish, you can do any amount of molecular biology, but you cannot do much of a cellular immunology work. With the model that we work, you can do anything.
Okay.
If you want.
There's a last question over here, which is a brief one.
Yeah,
Thank
so it's just a follow-up to the same question, but I wanted to emphasize on the fact that although, I mean, I believe that since you mentioned fish is a very good model and you showed a lot of similarities between the mammalian and fish immune responses, but since the environment they grow in is completely different, like a lot of mammals also grow like on land and fish cannot survive on land, and the immune response that is initiated is based on the environment or allergens that are susceptible to that environment. So how can we how do you
See, the problem is that you should not get restricted. We are using fish for some of our questions. Like you see, even Drosophila, which is an invertebrate, is used for studying, neurological problems that are happening in human beings. So it is just, adding more models to look for different questions that are there in the field. So in some, fish would be a good model. Some it may not be, where you might have to look for other alternate models. So it is an alternate model, that is what I'm trying to say. It is not the only model.
Thank you very much.
Thank you so much.
Thank you. And I think we've got her.
Our sincere thanks to all the speakers and the chair of this session. We would like to invite all the speakers and the chair of this session to kindly come up on the dais for a group picture. We request everyone to please attend the posters and also visit the company stalls. Participants who have been assigned today's slot for poster presentation are requested to put up their posters according to the serial numbers which are provided. Speakers and delegates are requested to proceed to the ground floor, while the students and other registrants are requested to proceed towards the tent in the back.
Just a quick announcement for everybody. There's lunch right now outside. As mentioned, the ground floor is for the speakers and there's a tent area for others. It's just so that the crowd can be managed better. And 2:45, we will get the group photo session. There was a change in the schedule. 2:45, that's 45 minutes from now. At the front entrance of this auditorium, JN Tata, the whole seminar hall, the front entrance where you entered from for registration, that's where we'll have the photo session at 2:45. At 3 o'clock, we'll assemble again here for the next session. Thank you. Yes. Again, photo session at 2:45. Just a reminder, photo session at 2:45. In front of the auditorium. Please.
as Remembering Mohan Vani, one of our esteemed society members. I would like to invite our session chair, Dr. Arvind Shahu, a close colleague of Dr. Mohan Vani, to lead this session. We have three distinguished speakers lined up. First is Dr. Rupesh, who was Dr. Vani's student. Next is Dr. Girdhari Lal, a colleague of Dr. Vani. And finally, Dr. Jyoti Prasanna, who will offer an external perspective.
Should we go? Good afternoon, everyone. First of all, I want to thank the organizers for having a special session in the memory of my dear friend and colleague, Dr. Mohan Vani, a remarkable person. I can talk, at length about Mohan and not stop, but we have limited time, so I'll just brief you a little bit. Mohan, by training, was a veterinarian, and Mohan was so good in his studies that I don't think there was any exam where he stood second. He was throughout his career always the topper, always a gold medalist, right? And then he joined in a stud farm actually, and where his salary was very good. He had his own jeep and car and moving, but he was so keen in interest that he took 50% salary cut and joined the NCCS, and he was in NCCS since its inception when it was called NFATCC. It was a sort of tissue culture facility, and Mohan joined that. He was one of the first earlier ones where he did. So he was so keen in doing research that he joined. Then Mohan moved to UK and did his PhD, came back again. And that's when I also moved back from the US and joined as a faculty in NCCS. And both of us were very keen, interested in inflammation. And that's how, we became very close. Mohan was, a little shy person, was not, outward that he would talk to people very often. But since our interest was so common, we got along so well. We also collaborated, and Mohan was very keen in understanding, the bone biology and cartilage biology and pathophysiology, and how does it really work, particularly his work on, how the milieu, how the cytokines, how the other things really modulate this, and can we, change the environment, and can we really get the benefit, clinical benefit out of it. He would always think, and since he was veterinarian, he was also trying to use dogs and other animals as a model to really understand diseases like, osteoarthritis, in rheumatoid arthritis and other. He was very keen in understanding and doing work. Like I said, he was very keen, always outward going, talking to people, trying to understand what else can be involved. And that's how, we started doing a complement system, which had nothing to do at that time when we thought to something to do with the osteoclast or the osteoblast. And Mohan had a very clean model, which is without stromal cell. He would take the precursor cells, grow them, and then convert them with, MCSF and Rankel, and they would change it to osteoclast. And there, we could really demonstrate that this complement really plays a key role. If there's no complement, there's hardly osteoclast you really get. So he was pretty good in discussing things, explaining and almost pushing his ideas. That was Mohan's idea to get into that and try and do that. He, of course, went up the ladder and became director of NCCS, where he started helping everyone. But unfortunately, we lost him, last June. And suddenly it was, there was no precondition which we were aware of. And suddenly one afternoon he collapsed. He had a massive heart attack and we lost him. So I'm really thankful that, today we are honoring him, his research, his memories, and we have people, and Rupesh is here. He was wonderful mentor to all his students, and Rupesh continued what he learned and what Mohan mentored him for, and still doing the same. So I think let's get to the session. We have 3 talks in this. The first one is by Rupesh, who is also a student. So we'll start with his talk. I invite Rupesh. And the title of his talk is The Landscape of Immunoporosis and Bone Health in Post-COVID-19 Era. Rupesh, it's all yours.
Yeah. It's fine. Yeah. So I thank you, all of you. Thank you, sir, for the brief introduction. And I firstly thank the Indian Immunology Society, Dr. Taruna Madan and Dr. Deepankar Nandi, because when I approached both of them, they were very, in the first instance only they agreed that we should have a session on this. So without wasting much time, I would now want to come to the, Dr. Vani was my mentor. And to be very frank, I have never seen such a humble person who used to call his students also with the prefix of aap. He never called me tum, ki tum karo. He always called me aap karo, aap aise karo. To all of his students, he was also very humble. Very humble, never had he shouted on me for a single instance which I can recall. And I joined NCCS in 2006. So, but I went not to join a bone biology lab, very frankly. I went there to do immunology. So when I was there, the projects which were there, one project was there on rheumatoid arthritis, studying the immunobiology of rheumatoid arthritis. So that was a combined project between Dr. G.C. Mishra, the then director, and Dr. Vani. So I was keen in doing immunology, so I went there. Dr. Mishra told me that he has a project because he was about to retire that time, only 2 years was there, and he asked me to meet Dr. Vani because he was having a project. So with a half-hearted approach, I actually went to Dr. Vani because the project was a bone biology. And I have never thought that I will be doing a bone biology. I was always interested in doing immunology. So, but I went because I wanted to complete the process. When I went there, when I entered his cabin, he was so humble, he was so calm that he interacted with me at length. And then he said, okay, you can now go and we will declare the results. So I was selected and I didn't have any other positions, so I had to join. So I started slowly. So I thought, okay, I will be only doing immunology in this lab, not bone biology at all. Slowly and steadily, he instilled in me the fascination for bone biology. And that is what I'm still continuing. And now I'm using bone biology as my primary objective to study immunology. So that is how things have changed a lot. And he has been a pivotal point in my career, how he changed that vision of mine. Because whatever ideas I used to have, I used to always go to him, sir, this is there, this is there. He never said no. He said, okay, you can order the reagent, discuss it with me, and we can work on that. So this is how it started working. And I didn't know either immunology or either bone biology. And Dr. Sahu is there. He knows all the thing. So I started learning from the seniors. So they were not much seniors also, because just 6 months only seniors were leaving the lab. So we didn't have that much of bridging also. So we learned mostly with interaction directly with Dr. Vani and Dr. Mishra. So he was teaching me pneumology and Dr. Vani was teaching me bone biology. And this is how I learned the field of osteoimmunology. And that fascinated me so much that today it's my bread and butter. And I have developed a new field that is called as immunoprocess. So talking about, sir, being a mentalist today is really something which is not acceptable at this very moment, because I've always interacted with sir a number of times, and he was so happy with, he never told me on my face that, okay, he's appreciating his students. But yes, at instances, we came to know that, yeah, he's so happy, because I remember still the day when I called him up, that, sir, my call letter has come for a assistant professor job at AIMS Delhi. And he was so excited that day. And he was in a market, busy market. He was not able to hear my voice clearly. And he said that, let me call you back. And within 5 to 10 minutes, he called me back. And the fascination, the excitement was even more than me in his voice. And he was so happy that day. I really felt, yeah, being how your students, if they perform well, that is the happiness which the mentor gets. So this is, I actually shared a very special bond with him. We didn't meet very much often, but yes, we were in touch. And I will always remember him as a person who has shaped my scientific career and toward the fascination towards bone biology. So now I think I will start how I developed this new field of immunoprocess. And this is just a hybrid, because when I came back in India, I was searching to do something which was slightly different and unique also at the same time. So I thought, okay, osteoporosis is something which has never been taken up, because I wanted something which should be on the interface between bone and immune system. So I searched the literature very hard, because I was initially at Central University in Sagar, MP. So due to the limited facilities there, I had to start something that was feasible there. I cannot do any infectious biology because we do not have a animal house there. So I started that. This is the start. This model of developing in postmenopausal osteoporotic mice will be fine enough, because here we are not dealing with any infections. So that will help me to do my immunology. So this is how I started, how immune system is actually governing your osteoporosis. And this is called as immunoprocess, which I later coined the term as immunoprocess. So this is 2018, I coined this term, and this is how we are working in this field. And this is what different things are there here. A lot of things are there. So if you see here, this present what I'm doing, a lot of angles to the bone field with respect to different. And this is the future we are looking into. Many more things, developing diagnostic markers, using artificial intelligence and machine learning, and how we can use immunotherapy for developing therapies for immunoprocess. So now coming directly to osteoporosis, we all know, because this is very important field, and this is how I was fascinated that osteoporosis is a silent killer, because you will know only when it is you have broken bone. And once you have broken a bone, your morbidity and mortality risk are very high for the second more fractures. And it is actually affecting 1 in 3 females and 1 in 2 females and 1 in 3 males. It was earlier 1-4 and 1-3 till last year. So these are the people at high risk. That doesn't mean that all the younger people sitting on the behind desk are much safer on the side, because this is the time when you are actually developing your bone. And this is the time, if you're developing your bone to the full potential with proper diet and nutrition, that it will help you to prolong your whole life. So the increase, there is a lot of going to be increased risk of fractures also associated. And these are some of the important factors which I have been working on: immune system, gut health, and maybe your viral infections. So infections and bone, because I will be diverting myself to a different perspective, because after COVID, the immune system has played a very key role in all the COVID-19 pathophysiology. And also I have shown and approved that immune system is also responsible for your bone loss and osteoporosis. And this is what led me to start this research, what is happening in post-COVID era or during the COVID time about the bone health, because nobody has thought about that. So we do have viral, bacterial, and fungal infections, and they all are actually leading to a lot of bone loss, which we have never paid attention to, and we are still not paying attention to that. And this is how we started. If I was looking at what are the long-term effects of COVID-19, you can see in this figure, every organ was there except bone. This I have added here through by animation. So this I have added. So if you see, there was no mention of bone. But so this is how I searched that. We reviewed that extensively. And now it is known that almost 20% of the patients, almost 50% of the patients who have been gone through coronavirus infections will be having an osteoporosis or bone loss, significant. And these were some of the mechanisms which are already known for your COVID-19 immune dysregulation, microbial dysbiosis, and your immune priming. This is how there's a common link between both of them. There is an hyperactivation of immune system for both the bone loss and COVID. There is your gut microbiome dysbiosis, and also the therapies which have been used for for the COVID-19 can also lead to your osteoporosis. So this is the fact that till 2 days back, there had been 77.6 crores infections and 70 lakhs death. And if you see, there have been more than 50% of people are going to have osteoporosis-related bone loss. That equivalent to 40 crores. Right now, 400 million people will be solely affected with osteoporosis in the near future just due to coronavirus infections and not anything else. And the Indian scenario is 30 million. Because in India, still this is not a health care priority, and we are not documenting the cases for osteoporosis. So this is how it leads. A lot of cytokine storm leading to osteoclastogenesis. To study this, what we were done is that we developed an ALI and ARDS model in mice. And we, apart from looking at the BALF and the lung, we also looked at the bone tissues also, the bone marrow also. And what we did find was that, if you see here, the data is very clear. After developing the mice model, you can see the ARDS mice model, a lot of cytokine storm was there. And this was actually when we cultured that bone marrow, even after short-term exposure, that is only LPS I have given for 24 hours, and that was actually enhancing both the osteoclastogenesis in the ARDS mice, even after 24 hours exposure, and also that was significantly inhibiting the osteoblastogenesis. The completely bone remodeling was taken off the roads. So then I was interested in what is happening in the long run. So what is happening in the long run? So we did this. So we developed, we took the 3 control mice and your low dose mice and the high dose, which is equivalent to your COVID-19 pathophysiology. And after 45 days, we sacrificed this. And you can see that. So there were a lot of mortality was also there. So we increased the number accordingly because 50% mortality we have taken care of. And you can see the low dose and high dose both are actually having a significant bone loss if you see this micro-CT data. And all the parameters for your bone loss enhancing, for example, Rankel trap was significantly high in case of both low and high doses of this LPS. So ARDS was actually dysregulating the bone remodeling, of course, in the long run, because it was actually sufficient even for 24 hours exposure is also sufficient. So there comes a study when I was searching literature in Lancet itself, 2022, that osteoporosis is actually a risk factor for your infections in your future and sepsis also. So that's a meta-analysis what they have done. If you have a lower BMD means that you could be having a high susceptibility rate for future infections and leading to your sepsis also. And this elevated risk is actually more complicated if you're having a COVID-19 infection. So that was actually very interesting, these 2 papers. So people have used a lot of corticosteroids also, and corticosteroids is already linked with osteoporosis. That is a glucocorticoid-induced osteoporosis. So that is the second phase of that key. Once you're infected, you're already having a bone loss, and you're treating with corticosteroids, rampant use, and that is again leading to your bone loss. So we did that also to prove what is happening, and we used the dexamethasone from high to low concentration, and we did find that, okay, your osteoblastogenesis is taken off. It is completely inhibited at high doses of your corticosteroids. And we did also the in vivo model also for the GIOP for both the low and high doses, and you can see here that it is actually leading to a bone loss. And this has been done by my 3 PhD students, Megha, Anurag, and Simran. So they also do, apart from immune system and the cytokine storm, there had been a role of gut dysbiosis also, gut health also in osteoporosis. And if you see this is sham in ovariectomy mice, you can see the colon length, gut integrity, tight junction proteins, and the dysbiosis and the alpha diversity is all dysregulated completely, significantly. And this is the case how it is composed and how the different bacterial composition is going up and down. So complete dysbiosis is there. And COVID-19 has also been reported with dysbiosis, and dysbiosis is already enhancing. So what I was thinking was that maybe from if you are having an infections initial and is going toward the COVID-19 pathophysiology, that could also be linked with your dysbiotic microbiota. And when we looked into deeper into that, we compared both dysbiosis in osteoporosis and dysbiosis in ARDS in both the mice models. And you can see here that in ovariectomy, there is a lot of increase in the pathogenic bacteria, especially this E. coli and sulfoviber piger. Apart from that, the healthy bacteria or the probiotic bacteria, which have a small, so they are also significantly inhibited. And among them, lactobacillus species was completely going off. So this led to, okay, if you can we, and this is also happening in the ARDS also, you can see ARDS also having lactobacillus, all the lactobacillus order, family, genus, and species level also, it was completely, the diversity was lost. So this let me hypothesize, so can we use probiotics? So it could be a game changer in your COVID-19 induced bone loss. So we took up the LR strain and we gave that to the ARDS mice model. And you can see here that it is even better than a control mice, the pathophysiology. When we gave it for the blindly to the sections were given to a histopathologist in AIIMS itself, they were completely amazed. What is this third compound we have given me? Is actually better than the control also. So this you can see all the parameters histopathologically, So what we did was that we again looked into what is happening. So we took both extrapulmonary ARDS, that is your, we firstly looked into this. This was the pulmonary ARDS, and after that we also looked into the extrapulmonary ARDS, that is a sepsis, because sepsis also leads to ARDS. And this is CLP mice model we developed, and actually LR was able to, at both 7 days prophylactic treatment and 14 days, it was actually inhibiting the mortality by more than 4 times and 2.5 times at 7 days and 14 days. So this actually led us to believe that actually LR is reducing the mortality burden has been reduced, and this was basically also leading to reduced level of osteoclastogenic cytokines both at the serum and the bone marrows. So bone marrow is the site where actually the bone remodeling, osteoclastogenesis and blastogenesis is happening.
Rupesh, you'll have to wrap up.
Yes, sir.
Quickly.
So this is how it is happening. The cytokine storm is leading to your dysregulation, and this was actually we also related with both in pulmonary and extrapulmonary with neutrophils, and we did find the neutrophils were high, and not only in the lungs, they were also high in the bone marrow. So this led us to, okay, what is the role of neutrophils in osteoclastogenesis? Then we looked into that. When we cultured neutrophils activated with osteoclast precursors, it was actually enhancing osteoclastogenesis. And it was happening with the short chain fatty acids because we did the mass spec analysis, and the butyrate was found to be most effective in inhibiting the phagocytic activity and the NETs quantification from these neutrophils. And also it was inhibiting the phagocytic potential of the neutrophils, these LR, via the butyrate, because we only gave the butyrate in some of the mice. So this is how LR was enhancing bone health by regulating your cytokine storm and your osteoclastogenesis and osteoblastogenesis. So this is the whole scenario, how it is happening. And this work was done by my PhD Lina Sapra and a postdoc, Chaman Sani. And this is the take-home message, that both under COVID-19 conditions, they have a similar overlapping nexus, COVID-19 and bone loss. And this is mainly why maybe it could be neutrophils, because this is the first time we have reported that neutrophils could also have an osteoclastogenic activity, and that could be related with the phagocytic and the NET drilling potential, apart from their inflammatory cytokine production, and that is actually dysregulating this bone remodeling, leading to your bone loss. So this is just with the case of a certain viral infection. It could also happen with bacterial and fungal infection also. So I thank all of my group there and all of my collaborators, and thank you for listening me up.
Thank you, Rupesh. Maybe we can have one question. It's pressing.
So all I have understood well.
Maybe then we can go at Gidari, we can discuss it outside. Okay, there is one.
I have one quick question. Here. Hello, sir. So, sir, when you showed, like in the case of acute lung injury model or the ARDS model, it's generally the alveolar capillary barrier that is disintegrated. So what do you think? Is it because of the inflammatory response that the junctions are going off, or is it because of the junctions going off the inflammation is going high. So
So-
which one is causing what?
So we have shown that data also. We have done that extravasation method for Evans blue also, where we have seen into the vascular leakage in the lungs itself. That data was there on the slides. So it can actually lead both way. Your leakage can also, firstly, all of your occludins and zona pellucidin or zona, these genes could be disintegrated, and this could lead to vascular leakage. And also there are other mechanisms also in which this pathophysiology, which is actually unknown, because that's why we are not able to control this ARDS rated. That's why patients are going on ventilator. So this is something which needs to be worked on, whether this is happening first or the other way which you are telling is happening first. Okay?
So we can't figure out as in
Actually
using
not
this-
right now, because we are still dealing with that. And people across the world, it's not in my focus of research because we are dealing with a different angle. But yes, people are working on that.
Thank you.
Thank you. She's my IM Biotech student also.
Okay, the next one is Girdhari Lal's talk. The title is Neuroimmune Communications of Tachykinin Receptor 1 Signaling in Gut Inflammation and Tolerance. So Girdhari, please go ahead.
All right. Yeah, it's like a little nostalgic moment for me from 2000 to now 2024. So this place where I learned immunology, Indian Institute of Science, microbiology, and some of my teachers are here. Dr. Nandi and Dr. Anjali Karande, who taught us immunology in IISc. So that's wonderful, actually. And also, I thank Dr. Nandi for putting Dr. Vani's memorable here event that we should actually honor him. He's a wonderful colleague at NCCS. In fact, when I joined, I have to go, if I need anything, I have to just walk through his place and then say, how do I do this? And partially, the Rupesh was also there at that time as a student. So Dr. Sahu and Rupesh already told that what his things, academic activities. And also, he is a scientist and also member of several academies in the country. And several awards he got for his credit. And like Rupesh mentioned, bone biology and regenerative medicine is the key area where he was working on. So I thought I'll just few slides I'll put for the students' sakes where exactly he worked on. So I just took this review article just to put that if you look at synovial or in the synovium of rheumatoid arthritis, there are a lot of cellular interaction happen. You have T cells, you have TS17 cells, and then there is osteoblast cells, and then macrophages. All these cells interact each other through different receptor. Now, I'm not going to go in detail, but what I wanted to put perspective that there are a lot of cellular and molecular event happen in the synovium. And where Dr. Mani has contributed, some of the paper which I have listed here, his work done in the NCCS. These are published in Journal of Immunology and several others. So you look at these dots where these things are connected now. He provide evidence that these cells interact either through the molecule which is mentioned here, the interleukin 3, or the cell type, how they talk to each other to either promote or control. So IL-3 mostly regulatory component. So regulate these events at different level. And if you look at, like Rupesh mentioned, the osteoclast and osteoblast, the cells which is actually degrade the bone, and also there are osteoclast which actually forms. Now, whenever you have disbalance in these two type of cells, you have degradation or loss of the bone. Now, you see some of these, like I mentioned, regenerative medicine. So he published several papers and wonderful research that how mesenchymal stem cells actually help in inducing tolerance in the local microenvironment and how they promote it. And obviously in the community, in the immunology community, he definitely was loved one. And then you can see in several meetings, a lot of people actually, those who knows probably knows that. And this is the picture from this recent last year in IISc itself. And in fact, Dr. Nandi has organized this CIRCON meeting, and we were part of it in the IISc itself. So this is some pictures where you see that pictures from NCCS, and also we have interesting get together all the time. So it's wonderful. Actually, he was a wonderful colleague, a wonderful mentor, and also first thing is very good heart, ready to help anytime. So with these things, I think we miss him. We miss him here and NCCS. I will move on to the topic what we assigned to discuss here. So my lab work on neuroimmunology or try to understand how the immune system and neuronal system talk to each other. So what I'm going to discuss to you is that how tachykinin receptor, that is the nociceptor, that means sensing receptor, a sense for the pain, how this receptor sense actually the pain in the gut, and how does it contribute to inflammation and tolerance in the gut. Because the fine balance between inflammation and the tolerance is the mix that your gut is going to be inflamed, and it has to be inflamed all the time because we are always exposed to different microenvironment, but how to keep this balance. So little bit about this tachykinin receptor, the ligand which is actually binds to it, substance P. The P name comes because originally when they were actually fractionating different molecules, the P fraction is the one which shows activity, neurological activity. That's how the name got as substance P. And it binds to the receptor called tachykinin receptor. This is also called as neurokinin receptor. And it controls a lot of biological process. In fact, it actually helps in the pain, like I mentioned to you, immune cell proliferation, inflammation, and vasodilation. And if you look into the disease perspective, lot of disease conditions, substance P goes up or dysregulated. And now question, and the way substance is produced, it produces as inactive molecule. There's an enzyme which cleaves this to make it 11 amino acid peptide. So it's a neuropeptide which binds to this receptor and does this job. And since it is a neuropeptide, it cannot survive for long. So there's an enzyme which degrades this also. So that's ACE enzyme which degrades this. Now the question what you see that if you go to clinic and the clinic you have inflammatory bowel disease or irritable bowel syndrome where you don't know the cause, but if you go to colonoscopy you see everything is reddish and inflamed. Now question is what is there? What is causing this inflammation? So we were interested in that. We went to clinic and then asked the clinician, okay, tell us what you see there and then can we have some sample from there to see that what the cell and biology tells us from there. And when we did actually looking at different receptors, what I'm showing you here is just receptor because the talk is focused on that. If you look at IBS patient, the IBS patient has more of this receptor TACR1 and TACR2. TACR3 we did not find expression, especially in the gut biology, in the colon biopsy. Then we thought, does it has any role in terms of inflammation, gut inflammation? And we know that CD4 T cells or the T cells play a very important role in the gut inflammation. So then we ask question, does T cell express this receptor? Because this is a neurotransmitter and the receptor has to be mostly on nerve cells, central nervous system, sensory neurons. And then we went back and we went to human and mouse, the data what you saw that, so the human data I'm not just showing, so now I'm going to talk about mouse, so I'll just focus on only mouse. So what you see here that if you look at different organs, the receptor which is three forms, TACR1, TACR2 or TACR3, they express in different tissue type. And if you talk about T cells comes in different flavor. You can call it regulatory T cells, TS17 cells, or naive cell or memory cells. So we make use of the transgenic mice, let's say FOXP3 GFP or ROR gamma GFP, and then you can directly sort these cells either TS17 or Treg cell without disturbing them in a homeostatic condition. What you find that these receptors are mostly upregulated on TS17 or Treg cells. You can also generate these cells in vitro. You take naive T cells, in vitro give right cytokine and the growth factor or the activation. Then these cells can differentiate into Th1, Th2, Th17 or Treg cells. And then ask question, if they are in vitro differentiated, do they express? So the answer is yes, there is Th2 cells which predominantly express in vitro. And also there is some fair amount of Th1. So this is RNA data, and then you can go back and do flow cytometry, and you find that these cells do express this receptor. So what? What is importance of this receptor in terms of the pathophysiology? Then we use a mouse model for the gut inflammation. So the regular C57 mice, if you give dextran sodium sulfate, which is a mild detergent, and it induces gut inflammation. And then ask question, what happens if you actually promote or activate, agonize this receptor or antagonize this receptor? So if you actually give DSS, what you see here, in this one, the red line, you will see that the mice actually develop diarrhea. And then because of diarrhea, you have weight loss. and also the change in the colon length. Now, if you actually give antagonist, which is here CP, is a chemical antagonist, which binds specifically with the TACR1 receptor, now you can actually control the disease. So as disease activity scores, which also account for different other parameter of the disease. And not only that, if you agonize this receptor by giving external agent agonist, which is substance P itself, you see it has much more mortality. So that means if you have more substance P, it drives much more inflammation, and that's why probably the disease score is much more high. And so as you can see, the pathophysiological of disease. Now, since we are interested in this in terms of immune parameter, we went back and see, okay, where is the receptor expression in the tissue, especially in the spleen, colon, or the colon, especially receptor expression of this TACR1. What we find is that whenever we have antagonized, the receptor expression goes down, so as production of substance P also has gone down in the colon and as well as in the spleen. Like I mentioned, T cell express, you can clearly visualize whenever you stain with the marker specific for CD4, you will start finding them, they are expressing in the tissue. And especially in disease condition, they are much more, and when you actually give the antagonist, they are down regulated. Now, the question is, in the gut physiology, you will have that something called regulatory T cells, which are marked by FOXP3, and also pathogenic cells, which is RO or gamma, or expressing cells. So then we ask question, which among these CD4 T cells, because imagine that this receptor is not just on T cell, there are several cell types which express. Now, question is, when I'm giving antagonist, are it going to work on T cell? It can be working on other cells, so fine. So in that, to answer that question, what we use almost like 25, 26 different antibodies, and you did the spectral flow cytometry to find each one of them, how they respond to it. What I'm showing the data only gated on CD4 T cell. So what do you see that moment you have this, you see that the CD4 T cells start clustering in a different group. And there are some clusters which become biased towards one specific condition when you give agonism. And these clusters, if you start looking in terms of molecular level, molecular expression of the pattern, you find that these cells actually start expressing a different cluster having different phenotype. Now, what I'm showing to you that here, there's a more altered, the disbalance between inflammatory and regulatory T cell mark. Here is just to show the marker. I don't want you to read it, but what I wanted to point out here that there's when you agonize the receptor, you have increased Treg response and decreased TS17 cells. Now, question is that if it is Treg cells, how does it work? Does it bias toward specific subtypes of regulatory T cells, or it is what we know as FOXP3 positive cells that way? So now, what I'm showing to you here is the FOXP3 T cells, and these cells actually having different markers, so that you can actually now just what I showed you, the cluster is just representation that you have TH1, TH2, and TS17 at Treg cells. You have more Treg cells here, and then down regulation of the inflammatory cell like TH1 cell. Now, since we focus on now regulatory T cells, so I'm going to take digress from the other cell type and then ask question, what is that FOXP3, or what is among them is causing more or less, or what is the phenotype this particular receptor is modulating? So now these cells, what you're seeing, these cluster is focused only on FOXP3 positive cell. And now when you see that the FOXP3 positive cell is not just FOXP3 positive cells. You look at these different subset, it becomes forms. So now you don't have just one FOXP3, you have a number of FOXP3 positive cells, probably, and these markers which is used, they are actually going to target either using chemokine receptor, so that where they localize, or the cell adhesion molecule, or the type of receptor it used to inhibit the immune response. So now if you start clustering them, you will find there are a lot of clusterings of the various clusters are formed. Now among them, how they work, where they work, now that's another area to look for how they will working. Now that's part of this kind of what I'm showing you in the secondary lymphoid or the gut associated lymphoid tissue. Now if you look at the secondary lymphoid structure, there are cell types like follicular regulatory T cells, which actually goes to the follicle and they control the B cell and other cells also not to go haywire, right? So then we ask question, what happened to those cells which goes beyond the T cell zone and inhibit them? Now what you see that those cells also upregulate, not only upregulate, obviously the marker for them to tag them using the chemokine receptor, and what you'll find that they are also upregulated. All right, so far so good. Now question is, All this what we finding is just because everything else is there, these cells are also there. And it so happened that we capture these cells just because we are getting and analyzing them. Now, to understand the biology, that if this is the case, can I take out the naive cells and then differentiate in vitro where I know what I have taken and how they are going to differentiate it? And if I agonize or antagonize, what happened to them? So if you antagonize them, then you see that in vitro also these cells try to expand much more rapidly. And not only that, they have much more suppressive capability in vitro. Now, if that is the case, which is doing in vitro, can I take these cells, since they have got more suppressive function, and transfer into another animal which has got the inflammation? Can they suppress that inflammation? So for that, what we did actually, we use a model, a mouse model again. The mice, what we purify the naive T cells was from FOXP3 GFP mice. So now, naive cells which we purified, they don't express GFP, but they convert into Treg cells, they will start expressing GFP. And the recipient mice, the Tregs actually which is transferred is actually RFP mice. So now the Treg which we differentiated is expressing RFP, and the naive cells which we purified is not expressing GFP, but they can express GFP if they convert into Treg, so that we can monitor in vivo what's happening to them. And now, once we have these two types of naive cells and the Treg cells, we use a NRG mice. NRG mice do not have IL-2 receptors, so it does not have CD4 T cells, CD8 T cells, or even NK cells. Yeah, I'm just last slide actually. Now, if you give these CP-treated Tregs, they actually protect the mice from the disease. Then you go back because you have tagged these molecules, these two T types of Treg, and they start looking at what happened to them. Does these Tregs which were exposed, they themselves suppress, or they also take along, convert the naive cells also more efficiently, the Treg cells? And then what you see that the naive cells which were injected, along with them, they also start converting into Treg. Now, so here I will conclude. What I showed to you is a neurokinin receptor or TACR1, tachykinin receptor. When you have homeostasis, you have these cells in a control mechanism, control way, or the homeostatic manner. But whenever you have the disease, there is increased inflammatory cell and decreased regulatory T cells. And when this happens, if you want to control therapeutic control, you drive this to TACR1 antagonism, and then you take over the Treg over the inflammatory cells, and you control the disease. And that's where I closed the talk here. I acknowledge all the people who did. Mostly this work was done by Amrita. She has finished her PhD. And thank you very much for patient listening.
Thank you, Giridhari. We can have one or two questions. I'll start. Let me ask and then Showman. Giridhari, in your model, you're using mouse, right? But the TACR1 receptor is not just present on T cells, it's also present on many other immune cells. Right. So the effect may not be particularly with respect to direct action of these on the T cell. It could be some total of everything which you see, right?
Right.
But the model you made probably because you do that in vitro, and that's why. But what do you think is that, in vivo, what is happening?
Right. So to test that it works on T cell, the data I showed you, you purify only T cell and differentiate into Treg and incorporate CP. Antagonize this receptor and you have more. Now you don't inject actually antagonism in the another disease model. Rather you inject the cells which are actually pre-exposed to antagonist. So other cells in the body is not going to see the antagonist, and then you see that these cells actually which has pre-exposed, they are much more stable and much more differentiated and probably protecting it. So what you said is right, that there will be a number of cells, but the question what we ask, does CD4 T cell do with this receptor?
Showman.
Hey, Giridhari, wonderful talk. Quick two questions. Substance P, is it also changing in abundance during the course of colitis?
Yes,
in your animal
it
model?
does. So that's what I did in the, probably I don't know, it was very clear. When you have inflammation, substance P expression goes up, as well as TACR1 expression goes up.
Thank you.
Both in mouse and human.
Second one is this, what is going on downstream of TACR?
That's what we are doing right now in the lab. So we have some clue, but yeah. So, yeah, we can discuss that part. Yeah, right. So we know there are some signalings with the TACR1 which actually drives this. And then that's how the change in the Treg phenotype or inhibiting the TS17 or TH1 cell is happening.
Go ahead.
So Giridhari, So nice talk. So I just wanted to know, so the expression of this receptor is predominant of the Th17 cells, right?
Th17 and Treg cells.
But when you showed the data, the expression is highest on the Th17, followed by Treg cells.
Right.
So my question is that when you are adding the antagonists in the Th17 differentiated condition, so have you checked that those are this blocking the differentiation and promoting the Treg, because there was a paper that because you need a similar cytokine cocktail, like TGF-beta, IL-6 for Th17 and only TGF-beta
Yeah,
for the...
good question. So I'll tell, that's what I showed you the data. Even if you have Th17 differentiation program, and when you block this receptor or antagonize this receptor, the pathogenicity of the Th17. So you have Th17, but it's not necessarily they're pathogenic. So the pathogenicity of the Th17s get altered. And then if you look at the Treg cells, their suppressive activity goes up. Right? So the way it modulates that, it shuts down program 1 in one cell type and upregulates the program 2 in the other cell type. And that's how it overall effect, what you see is the protective in nature. Yes, so like I showed few data, one data actually does it exist, and the human story is wonderful, and we are now looking into it. The TAC-R1 receptor, if you look at, it has got 2 receptors, short form and long form. And if you start looking the different cell types in human, immune cell types, the expression pattern is really wonderful. Now we're trying to understand who, where, and how it works in human. So that part we are looking into it.
Thank you, Girdhari. I think we'll move on. Maybe others can discuss outside. So we can go to the next one. The next talk is the last one, is by Dr. Jyoti Prasanna. And the title is Ectonucleotidase, Key Damage Sensors at Crossroads of Tissue Regeneration, Repair, and Immune Modulation.
First and foremost, I'd like to thank Professor Nandi for inviting me for this conference. And it is really surreal for me to talk before my mentors. I have Dr. Santosh Kar, who was my direct mentor at JNU during MSc. And because of him and because of his interesting lectures, I chose immunology as a field. And then, of course, Professor Nandi, because of whom I did my PhD in IISc, and despite all bottlenecks and all problems, I'm still sustaining in this field. And the third is definitely Professor Vani, because I was an oddball out in a regenerative stem cell medicine institute, and being an immunologist, I didn't know what I was doing in a stem cell and regenerative medicine institute. And he was the one, and his work was very inspirational because he had managed to bridge the gap between developmental biology, stem cell biology, and immunology. And he had been always inspirational. In fact, if I could, life has its own surprises. He was supposed to come as an examiner for my PhD student, and he apologized over WhatsApp that I have not been able to do the thesis on time, but I'll be there, I'll give a talk. And we were on the verge of booking tickets for him. And I called him at 10 in the morning, and at 12, Dr. Bonde told me that he's no more. Of course, that memory would always be there in my mind. But of course, his work would be inspirational for youngsters in the days to come. So coming back to the coming back to my talk, sensing damage and responding to damage is very, very key for organismal existence. And so is the case with tissues. So tissues are endowed with lot of cells, boundary cells such as mesenchymal stem cells, macrophages, and endothelial cells, which have molecules to sense this damage and annihilate it. And in case we are not able to annihilate it, it leads to pathology. So why are we talking about ectonucleotidases? Ectonucleotidases are basically the phosphohydrolases which converts nucleotides to nucleosides. Although there is a lot of redundancy with respect to NTDPases which convert ATP to AMP, but there is only one unique enzyme called as NT5E or CD73 which converts AMP to adenosine. ATP is immune activating, whereas adenosine is shown to have immune suppressive node. So in this context, CD73 serves as a rate limiting step in deciding between immune activation versus immune suppression. So I got into the ectonucleotidase field by serendipity rather than by choice, because we were working on mesenchymal stem cells. Mesenchymal stem cells are known for their reparative functions. They differentiate into connective tissue lineages such as the adipocytes, osteocytes. They immune modulate. What you see here are T cell blasts, and when you co-culture them with mesenchymal stem cells, we see blast suppression. But interestingly, the immune modulatory properties of mesenchymal stem cells are not constitutive. They need immune priming, as you can see here. In a case of allogenic bone marrow transplantation, when you give mesenchymal stem cells, they result in graft acceptance and reduced GVHT. But IFN gamma receptor knockout, IFN gamma receptor knockout MSCs fail to do so. So immune sensing is a key parameter to mediate the reparative effects of MSCs. And mesenchymal stem cells have abundance of CD73 on their cell surface, and in fact, it is used as a characterizing marker for mesenchymal stem cells. So we wanted to ask whether is it just a marker present or does it have any functional significance? So we generated shRNA knockdown lines of mesenchymal, stable lines of mesenchymal stem cells, and looked at the osteoadipopotential because they are stem cells. And we do see that there is a decrease in the adipogenic potential, but there is an increase in the osteogenic potential. So when you don't have CD73 on their cell surface, mesenchymal stem cells are primed to the osteogenic lineage, and we showed that by transplanting them, by using TCPHA scaffolds, putting them in mice, and we did in fact see that the bone volume has increased. So what happens to the other part of the mesenchymal stem cells? Mesenchymal stem cells, which do not have CD73, they fail to attenuate T cell blast responses. And this we have attributed to the reduction in the immune modulatory secretome of mesenchymal stem cells. So now the second question was, is it that CD73 knockout MSCs are not able to immune modulate because they can't sense inflammation? As I told you before, the immunomodulatory properties is not constitutive, but it is licensed by inflammation. So we looked at the responses of mesenchymal stem cells with IFN gamma, and we looked at the prototypic enzyme IDO, which is a prototypic enzyme for IFN gamma signaling, and we see that in CD73 knockout, we have reduction in IDO. Apart from that, the entire IFN gamma signaling machinery is downregulated in the CD73 knockdown cells. So a couple of years back, we had shown that mesenchymal stem cells regulate macrophage activation and polarization, and this is dependent upon the activation threshold of the macrophages. For example, when mesenchymal stem cells are co-cultured with naive macrophages, they cause immune activation, whereas when they're co-cultured with inflammatory macrophages, they suppress. So definitely inflammation plays a role in endowing mesenchymal stem cells with their immune modulatory abilities. So now we wanted to check whether the CD73 knockdown MSCs would be able to suppress M1 macrophages, and we do see that they do not. And we attribute this to the reduction in IFN gamma, because IFN gamma is secreted and is also there in the cocktail of the co-culture of M1 macrophages and MSCs. And when we substitute CD73 or IDO, which is one of the downstream target of the IFN gamma signaling, we do see that M1 suppression is re-established. So the story this far is that CD73 is not just a marker present on mesenchymal stem cells, but it also helps in sensing inflammation and endowing mesenchymal stem cells with immune modulatory properties, and also influences its fate choice. So we propose that CD73, since mesenchymal stem cells have been used therapeutically, clinically to cause regeneration, we think that CD73 or its activity could be a good marker to earmark therapeutically relevant mesenchymal stem cells. Now, coming back to, is there an associated disease related to these molecules? And we looked up at diseases, and we came across a rare disease called as ACDC, which is arterial calcification due to deficiency of CD73. As you can see here, there is joint calcification. Apart from that, the arteries are getting stiffened. So we thought, but interestingly, the mouse model of CD73 knockout mice do not phenocopy the effect which we see in the ACDC human model. So we thought we need to have a humanized model to study vascular defects, because in the mice model is mainly the defects are related to ectopic calcification in the bone, whereas in the humans, people have noticed vascular defects. So in order to understand the vascular roles of CD73, we created a CRISPR-Cas9 based knockout in human aortic endothelial stem cells, and we started looking at the endothelial properties. What we saw in the CD73 knockout lines was there was no obvious change in the cell surface characteristics like CD31 and VEGFR2, but there was a breach in the barrier function. As you can see, there is disorganization of the CAD and ZO1 junction proteins. Apart from that, when we try to put these cells to an angiogenic capillary formation assay, we saw a reduction in capillary formation. So in order to find what is there, why is it that CD73 is causing reduction in CD73 in angiogenesis, we did a single cell RNA-seq analysis, and we found that a lot of molecules which are involved in ECM remodulation and ECM generation are down modulated. One of it is BWF, the other is endoglin and amylin. So why one molecule which we picked up from the lot was CD105. CD105 or endoglin is particularly interesting when we talk about angiogenesis, because endoglin regulates the level of VEGF reception on the cell surface. In fact, VEGF receptor is stabilized by endoglin interaction. And in our CD73 knockout cells, we do not have endoglin. So this is very evident when we did a sprouting angiogenic assay. We see that the control cells form long sprouts, whereas the mutant fails to do so. Now we wanted to check whether these effects are because of the activity of CD73 or is it because of the protein CD73. It is very interesting because CD73 is associated in membrane rafts, and it has been shown to be co-associated with caveolin. Interestingly, when we tried to inhibit the CD73 activity, we didn't see a change in the angiogenic potential. So at that time, people did not know what are the residues which are involved in CD73 activity. So we did an MD simulation to basically look at the activity site. And as you can see here, there is a pi stack, and in between is your AMP molecule. We tried to, the pi stack constitutes of the AMP molecule and two ring residues, which are tryptophan. We converted them to valine. And this was the first time people were trying to look at these residues. So we put it into HEK system and saw whether, in fact, is it surface stable? Changing these residues or mutating these two residues did not hamper its cell surface expression, but only activity was modulated. So we are planning to use this to put back and check like what are the activity dependent roles versus the activity independent roles of CD73 in modulating inflammation as well as angiogenesis. The other interesting facet which we noticed was the responses of CD73 knockout HAECs to inflammation. In fact, it was very surprising for us to know that mineralization in mesenchymal cells or mesoderm-derived cells is a default route. And there are several mineralization breaks which have to be put to prevent these mineralization. So we thought like CD73 knockout cells lost their epithelial morphology. So is it that they ossify? Because we wanted to look at it because the ACDC disease people have ectopic ossification. So what we see here is that CD73 knockout cells, they have drifted from their state. They have started expressing RUNX2, and under inflammation, this is further exaggerated, and there is expression of the osteogenic commitment factor ostrix. So clearly, not having CD73 or the signals downstream of it has resulted in a fade drift of an endothelial cell to a mesenchymal cell and probably osteo. And as you can see here, this is just n is equal to 2. I have showed it with 2 independent clones of CD73 using 2 sgRNA construct. We see that in the presence of inflammation, endothelial cells are able to buffer by overexpressing mineralization break proteins such as matrix glycoprotein, whereas in your inhibitors, when you inhibit CD73, these mineralization breaks are not being upregulated. Now, what is the significance of all this in the context of pathology? At that time, an MD PhD, she's an MD in histopathology, joined my lab as a PhD student, and she wanted to look at CD73 in oral submucosal fibrosis. And what we see here is that even in the submucosal fibrosis, where you have epithelial dystrophy, and most of it is stroma, we see a complete reduction in CD73. And there is a concomitant reduction in 105, which is downstream, which we saw downregulated in our CD73 knockdown cells. We also saw an upregulation of TGF-beta signaling pathway. So we feel that CD73 plays a very, very important role in buffering inflammation and buffering fibrotic responses. This is something which is an ongoing work on the lab. This is summarize what I have talked till now. You have the CD73 pathway in endothelial cells, which prevents it from end-met. It is involved in normal angiogenesis. It is involved in maintenance of endothelial integrity. It inhibits default mineralization and prevents promiscuous transdifferentiation or fade drift from a endothelial lineage to a mesenchymal lineage, whereas in the mutant, you have the end-met progresses, you have impaired angiogenesis, there is loss of endothelial integrity, there is increased mineralization, which is particularly prominent when you have inflammation. So to summarize, under inflammatory conditions, probably CD73 or NT5E is acting as a sink or buffer to control fade drifts and also to manage inflammation. So we are planning to extrapolate these observations in context of diabetes and fibrosis, because diabetic patients generally have low levels of CD73. A parallel study in my group is working at looking at CD73 expression on osteoblasts and osteoclasts isolated from DBDB mice, and we are studying how it affects mineralization and osteoclastogenesis. And to our surprise, even CD73 knockout cells, pre-osteoblasts, have higher mineralization capacity. Although we started with something called as a rare disease like ACDC, it might probably, the relevance of CD73 might not be as rare. We would try to extrapolate these studies in checking whether CD73 is also secreted. So we would want to extrapolate or check if it can be used as a serological marker for fibrosis and also predict the propensity for arterial calcification. I think I am on time. So my acknowledgements. This is our beautiful campus. These are my students. We call ourselves a DAMP lab because we look at damage responses in stem cells. This is a picture which is taken from IISc Open Day. We had all come as a group. And this is a lab trip. Of course, Professor Nandi thanking him for inviting me and introducing me to the field. Professor Colin Jamura, who helped us with the transplantation of CD73 knockout cells in NRG mice and studying ossification. Anujit, who has been my comrade since my PhD, and a lab where we can steal reagents, a lab without doors. And of course, Manipal Hospital, which, and my initial work at Manipal Hospital, which made me think beyond basic immunology. And of course, this is our beautiful Mahe Bangalore campus. And as the Dean of MIRM, I am looking for, we have started a course on translational immunology and MSc by research in translational immunology, which is one of its kind in India. And I'm looking for faculties who have experience in translational immunology. Thank you. I hope I kept to time.
Yeah, thank you, Jyoti, for finishing in time and a wonderful talk. We can have questions. Yeah, please go ahead. Can you please hand over the mic?
Some of the diabetic patients, they show calcification at the aorta level. Aorta. What is your opinion on that?
In macrophages isolated from diabetic wounds, there is a reduction in CD73 as well as CD73 activity. So we don't really know whether macrophages which are there at the plaque interface also have low levels of CD73. Yeah.
But they have a high mineralization at the aorta.
Yes. So we really don't know, is it because of the fact that some of the endothelial cells have fade drifted, or is it because of something else? Yeah.
The sprouting of the blood vessel that you have mentioned,
Yeah.
have you checked the CD31? Because some of the hemangiomas, the vascular tumor, can have those sprouting of the blood vessels.
Surprisingly, there is no reduction in CD31.
Okay.
There is reduction in sensing of VEGF reception. As I told you, in CD73 knockout cells, we had a reduction of endoglyne. And endoglyne is involved in stabilizing VEGF receptor at the surface of endothelial cells. So we think that the VEGF reception is sort of changed in the knockout cells.
Thank you.
And that could be one of the reasons why you have all these diabetic complications like diabetic vasculopathy and reduction in angiogenesis. And in fact, I would like to point out that CD73 is being used, inhibitors to CD73 is being used as a third level of immunotherapy, apart from the checkpoint blockade therapy. And most of the papers are from this field. Yeah.
Thank you.
Okay, we can have one, but very short question and short answer.
Yeah.
Excellent presentation. I just have one query, that whether CD39 knockout will have the similar effect as that of CD73?
CD39 is not so abundant in our system, at least the mesenchymal stem cell system. But in context of lung injury, people
But in absence of CD39, then how this AMP will be produced from ATP?
CD73 is only one of the enzymes which produces AMP. There is a lot of redundancy with respect to production of AMP. But there is only one unique enzyme which converts AMP to adenosine. So even if CD39 is not there, you would have other anti-TPases which could salvage the situation.
And what could be the effect if those are knocked down, like the similar kind of effect will be observed?
We have not done it. We have not done it, but in context of lung injury, people have shown that CD73 knockout has a more prominent effect than 39. So I won't be able to comment with respect to endothelial cells.
Okay.
Yeah.
Thank you.
Okay, so we'll stop here. So I thank all the speakers for wonderful talks. And I again sincerely thank the organizers for having this special session in honoring Dr. Mohan Wani. And I thank all of you to join this session to honor Dr. Wani and his research. Thank you so much.
Thank you, everyone. I would request the speakers and chair of this session to please come up on the dais for a picture. Now, we move on to our next session, which is a plenary talk by Professor Carla Rothlin, Professor of Immunobiology and Pharmacology, Yale School of Medicine. I request Chair Professor Sandhya Vishweshwariya to now take over and guide us through the session.
Thank you very much, Deepanka, for this kind invitation to chair this session. As mentioned, Carla is at the Yale University School of Medicine, Department of Pharmacology, or is in the Department of Pharmacology and Immunology. And she's going to talk to us today about some really exciting work on decoding cell death. Carla, please.
Thank you so much. Thank you so much for having me. This is my first time at the Immunology Conference, but my 11th time to India. And I can share with you that every time I go back home, I really miss India and I look forward to being back. So thank you, Deepankar, and all the other organizers for having me here today. Now, as you can tell from my title, I changed a little bit the topic. I'll share with you something that we're very excited about in the lab and that I think might be of broader interest to this audience. So what I would like to discuss with you is about a molecular mechanism that we think underlies immune diversity. Now, you would wonder, why would this be important? I think this kind of needs no introduction. A few years ago, we all experienced that if we get vaccinated, we all have a little bit of different responses. It's the same if we get infected. So it's evident that each of us respond a little bit different to an immune stimuli. So that is going to be the focus of this talk. Can we actually understand what could be the molecular and cellular mechanisms that might underlie this, broad immune diversity? Now, when you think about the immune system, you can think about it as, turning on a car. We're exposed to this stimuli and we turn on this immune response. In the lab, we have been very much thinking about this, and we realized that, of course, a key feature of the immune response is how much you will respond. What defines the magnitude of the immune response? I'm going to give you an example of that, but with this in mind, we started to think that maybe differences in those built-in mechanisms that can regulate the magnitude of the immune response could maybe underlie this immune diversity. Just as an example, in the lab, for instance, we have been studying a lot a family of receptor tyrosine kinases that are known as TAM receptors. And over many years, we identified that they act as brakes of the immune response. These particularly act on cells of the innate immune system. If you imagine that you have, for instance, an infection, an injury, and you have cells of the innate immune system, such as dendritic cells that get activated, they can migrate to the lymph node to do some of their fundamental work, which is to present antigens to T cells. What we discover over years is that the magnitude of this innate immune response, the magnitude of dendritic cell activation, is under very tight control by these TAM receptors that can get activated by their TAM ligand, which is induced when T cells get activated. So we reason that probably the immune diversity could emerge from differences on how we engage different regulatory pathways. So I would like to introduce different stages, for instance, in the life of key immune cells. The focus of this talk are going to be T cells, and think about how at different states on the life of a T cell, there could be regulation of their activity. Something that we were very much introduced today in Jim Allison's talk. We know that once T cells are activated, they're also tightly regulated. They can become exhausted. And this, of course, as you know, led to the recognition of this fundamental work of Jim Allison and Honjo by the Nobel Prize. But this is not the only time in the life of a T cell when T cells are regulated. As I've just mentioned, even at the very beginning, when T cells are activated by dendritic cells, we have mechanisms that can regulate the magnitude of the T cell activation through these negative regulatory pathways that activate to control the innate immune arm. We can go even earlier at the life of a T cell before it experiences any T cell receptor activation by their antigen, and this can actually lead to anergy, which can also be a mode of regulation. But a step in the life of a T cell that has not been very much a focus in terms of understanding whether it could underlie this immune diversity is at the very beginning, is when they are naive and they have not yet been able to experience what would stimulate them, what would be their antigen. So this is going to be the focus of the talk, and we're going to ask whether there could be modes of regulating the immune diversity that we have, the ability to respond by regulating this very early stage in the life of the T cells. Now, if you think about naive T cells and the magnitude of the immune response that they can lead to, you can think that there could be features that could define that magnitude. For instance, we know that the frequency of the clones of the different naive T cells can be different, and that can underlie how much you will respond. We also know that the affinity of the T cell receptor for its antigen can be different, and every naive T cell expresses a different T cell receptor, so that can eventually lead to a different magnitude of the immune response. Now, this has been known for quite some time, and therefore it has been more like the diversity of T cells in terms of their T cell receptor that has been the primary focus of our understanding on how naive T cells respond. But we decided to take a step back and ask a broader question, and that was whether all naive T cells, irrespective of their T cell receptor, irrespective of the antigen that they will recognize, whether all naive T cells are equally poised to respond to their cognate antigen, or whether there could be differences on their basal state that will make some naive T cells more ready to respond to their cognate antigens than others. And if that would be true, that could underlie this immune diversity. So when we started to ask this question, there was a very beautiful study that was published that started to reveal that this could indeed be the case. So let me share with you some of the highlights of these studies and then go into our findings. So this study from Randy Noel's lab identified that indeed the expression of a checkpoint known as VISTA could regulate the ability of naive T cells to respond to their cognate antigen. And it did it in a very interesting, peculiar way. So if you take naive T cells and you do single cell analysis, you can identify that first, they are in very different transcriptional states. So you can identify different clusters. I'm going to tell you more about these clusters. Now, what was interesting in the study is that if they now took naive T cells, but from VISTA knockout mice, the state of the naive T cells was quite different. I think you can appreciate that naive T cells that were, for instance, in this cluster 1 in a wild-type setting, now contracted, and then there was a new cluster that expanded, that it was this cluster 2. When you look into what is the gene expression of the naive T cells in these different clusters, while all of them are quiescent, this cluster 1 or blue expresses much higher level of a quiescent module, but this cluster 2 that expanded in the VISTA knockout expresses markers of more memory, although these are naive T cells. They have never seen their cognate antigen. And importantly, this change in this naive T cell heterogeneity really translates in changes on the ability of these cells to respond to self-antigen and to lead to increased autoimmunity. So this was the first evidence that first, naive T cells exist in heterogeneous states, and that maybe that heterogeneity can be different across individuals and could potentially account to difference in their response once the naive T cells get activated by their cognate antigen. And so this was the first genetic evidence that genetics can contribute to the diversity of the naive T cell heterogeneity. Now, we decided to ask the question on whether this naive T cell heterogeneity that can impact how you will respond to a cognate antigen would be only controlled by genetics, or importantly, could be controlled by pathogenic experience. In fact, we know that variation in the human immune response is largely driven by non-heritable influences. It's largely driven by what we are exposed in our daily life, by infection, maybe by commensals. This opens all these questions, right? So could this naive T-cell transcriptional heterogeneity be affected not just by genetics, but by microbial exposure? So we decided to ask very simple but fundamental questions with regards to this biology. We first ask whether this basal naive T-cell heterogeneity was probably dictated by the fact that all of us, including the mice that were studied, the wild-type mice, are exposed to commensal microbiota. Could it be that that is dictating those different clusters that I've showed you at the beginning? So what we first did, of course, very important, we ran these findings. We took naive T cells. What I'm showing you here is in the spleen, but it's the same if we take it from peripheral lymphoid organs, and did single cell RNA-seq. And again, we're able to identify that there are different clusters. Importantly, if we analyze the congruency between the clusters in our studies and those reported by Randy Noel, there's a very significant correlation. So we can identify the same type of clusters. And just to give you a flavor of what does this mean in terms of the genes that these naive T cells are expressing when they are in the different clusters, we can see that, for instance, naive T cells can be in this more memory-like cluster, this cluster 2, expressing high levels of KLF3. There could be naive T cells that are responding to type 1 interferons, like the ones in this cluster 3, expressing high levels of type 1 interferon responsive genes. There are even naive T cells that express high levels of NER77, which usually is induced upon T cell receptor activation. But as I'll show you, these are truly, truly naive. So we could see that we have the same basal transcriptional heterogeneity, and we thought most likely this must be defined by commensal microbiota. And so we developed this following settings where we could ask that question. We could take naive T cells from our conventional specific pathogen-free mice and do the single cell that I just shown you. We could take from germ-free mice that we indeed could corroborate that had no microbiome in their guts, or we could take germ-free mice and reconventionalize them, re-expose them to bacteria. We know we can rescue the appearance of commensals in the gut. And in these three settings, analyze the naive T-cell transcriptional heterogeneity. And while this was a very obvious question maybe to ask, the results were quite surprising. I guess we had hypothesized that this basal transcriptional heterogeneity, these T cells in a type 1 interferon responsive cluster and a TCR cluster, were going to emerge because of the commensal bacteria. However, when we took naive T cells from germ-free mice, they were in an exactly same state of heterogeneity as the SPF mice, and the same from our germ-free mice that were reconventionalized with the microbiome, indicating that this basal state of naive T cells is not dictated by the microbial exposure, something that really surprises us, but especially for the young people in the audience, I want to stress how science always opens new questions, right? And so now we're wondering, where is this naive T-cell transcriptional heterogeneity imparted? And you can start thinking about the life of a T-cell going even early on, maybe in the thymus, maybe in another stage is when these new clusters emerge. And this is something that we're very interested in addressing. So indeed, this was agnostic from the commensal microbiome. But of course, this is not the only experience. In fact, this is kind of a boring experience, if you think about it, to which we expose our mice under normal circumstances. So we decided to ask whether maybe this naive T-cell transcriptional heterogeneity could change with actual real life exposures in the context maybe of an infection. And we reasoned that because we have co-evolved with macrobiota in this case for 1 million years, maybe we should get started with a model of infection with a helminth and ask whether it is in this situation whether changes in naive T-cell heterogeneity could emerge. And this became really exciting. So we took again naive T-cells from the spleen or also from the mesenteric lymph nodes. This helminth goes through the gut and the data is exactly the same. If we take them from an uninfected mouse, I show you again the different clusters. Interestingly, when we took these naive T-cells, but from mice that were infected with Nipostrongylus brasiliensis for 1 week, we could detect a very significant emergence of an entirely new state in this naive T-cell transcriptional state. This infection in mice is acute. The mice can resolve the infection by 3 weeks, and you can detect that this state is starting to regress as the mice eliminate the helminth. So here we were indeed in the context of a real life type of exposure where the naive T-cell state change, this transcriptional change, state change with the emergence of a new cluster only in the context of the helminth infection that had high expression of very interesting genes. Genes such as, for instance, the IL-4 receptor. Of course, those of you that probably study helminths, as you know, this is a very type 2 type of inducer. And so it made sense that maybe there was an emergence of an IL-4 type of response. And indeed, if we look at the top 20 genes that mark this new cluster, these are entirely enriched in genes associated with an IL-4 response. And I'll tell you a little bit more about that as I progress on the talk. Now, I definitely want to convince you before going into what does it matter to have this new cluster, I want to convince you that indeed these are naive T cells that are changing. To do that, we took all the T cells, all the CD4 T cells from the spleen of mice that are uninfected or mice after 1 week of infection with Nipostrongylus brasiliensis. And we did again single-cell RNA-seq, and we coupled this with TCR-seq to identify whether there's clonal expansion or not. This is a very busy slide, so I'll walk you slowly through it. These are all the clusters that we can identify in the CD4 T cells from the spleen in non-infected and infected mice. I'll show them with 2 different colors, which I think will really show the remarkable change that happens in the CD4 T cell population when we have this infection. In blue, you have the SPF condition. In red, you have the the CD4 T cells in the spleen when you have an infection. In this area is the IL-4 responsive naive CD4 T cell. I'll convince you about that in the next slides. In this area, because the mice are responding to a helminth infection, you have your proliferating Th2 and TFH cells. So really a very remarkable, distinct state of T cells in the context of an infection, with T cells responding to the helminth, proliferating and becoming Th2 cells, but also with this bystander naive T cells being very much acquiring an IL-4 type of response. Let me convince you that indeed those are naive T cells. If we look into markers such as the gene cell that encodes for the cell addition molecule CD62L that keeps the naive T cells in these organs, or we look for markers for proliferation or IL-4, you can see that if you are exploring a non-infected condition, most of your cells are naive. If you now take CD4 T cells from the spleen of mice that have been infected with Nipostrongylus brasiliensis, you can see that in this area that I've shown you before, that is this IL-4 responsive, they indeed express this marker CD62L. They are not proliferating, they are not expressing IL-4. And if it didn't convince you, let me show you the TCR sequencing data. What I'm showing you here are all the cells in the different clusters, color-coded from 3 independent, non-infected mice. These lines indicate cells that share their TCR. In a non-infected condition, most of the shared TCRs are in the T follicular helper cells that have clonally expanded normally in those mice. If you now look into Nepo-infected mice, I'm showing you 3 different Nepo-infected mice. Again, all these clusters on the T cells. You can see that the vast majority of shared clones are in the TFH, but also in red are going to be your Th2 cells. And importantly, in this entirely new cluster that emerges in the context of an infection, which is this IL-4 responsive cluster, there's hardly any shared TCR, indicating that indeed this increased transcriptional heterogeneity happens without clonal expansion. Now, okay, this is all transcription. Does this matter? What does it matter for our naive T cells to acquire this new state when we have a helminth infection? Could this underlie changes in the magnitude of the response of those T cells when they encounter their cognate antigen? And so now let's go to this functional evidence that this can indeed impact the ability of the naive T cells to respond when we activate them. So let me introduce you to the paradigm. What we did is we took naive T cells from non-infected mice or naive T cells from mice that have been infected with Nepo. We did this in OT2 mice. So now we can take naive T cells from a normal mouse or a mouse that has experienced the helminth infection, and therefore a mouse where there is this new IL-4 responsive state. We can transfer these T cells to non-infected mice. So now we forget about the infection and we inject these mice with the cognate antigen for these T cells, with ovalbumin, in this case with this adjuvant alum, and ask whether these T cells that have experienced a helminth infection environment can respond equally or can respond differently from the one from a non-infected mouse. And so what I'm showing you here is the ability of these T cells to proliferate in black, and in each of these points is a different mouse. This is the early points of proliferation. This is the peak 8 of proliferation. You can see that in black, the naive T cells transfer into a mouse that receives alum, can proliferate. But if we now evaluate the ability of the naive T cells that experience a Nepo infected environment, you can appreciate that there's much less naive T cells that proliferated at the peak 8, and they accumulate at early stages of proliferation, indicating that their ability to respond to the cognate antigen is impaired. And these T cells not only proliferate less, but also acquire less expression of key markers of their activation. If we take these activated T cells that have been transferred into a mouse from a non-infected condition versus those that came from a Nepo-infected condition, and we perform a nanostring analysis and explore the genes that are expressed in these activated cells, we can see that many genes are induced if the naive T cells come from a non-infected mouse, but these are significantly suppressed if they come from a Nepo experience. And you can appreciate that the modules that are suppressed are those related to T cell function, to expression of chemokines, cytokines, and to cell cycle. Indeed, supporting more the conclusion that naive T cells that experience a Nepo-infected environment are less able to respond to their cognate antigen. We then wanted to understand what is the mechanism driving these changes in heterogeneity, and importantly, changes in the function of these naive T cells in their ability to respond. We noticed initially that the genes expressed in this new cluster appear to be very much related to a response to IL-4. So what we did is we took naive T cells from a Nepo-infected condition versus a non-infected condition, perform RNA-seq, and of course obtain many genes that are induced in this setting, and compare them to the genes that are induced in naive T cells if we do a much more simple perturbation, which is that instead of injecting Nepostrongylus brasiliensis, we solely injected the cytokine IL-4. And we observe that there's a very high degree of overlap in the gene expression of the naive T cells in the context of an EPO infection versus just providing IL-4, suggesting but not really proving that IL-4 is the driver of these changes. We could then do the reverse experiment and really have better understanding that indeed IL-4 is driving this change, because we could take naive T cells from a non-infected mouse, naive T cells from a NIPO-infected mouse, or naive T cells from a NIPO-infected mouse that has also received an anti-IL-4 treatment. So the cytokine IL-4 has been neutralized. And if we do RNA-seq, you can see that many of these genes, I have expanded this list for you here, many of these genes that are induced in the naive T cells upon NIPO infection are neutralized if we add the anti-IL-4 treatment, really clearly indicating that IL-4 drives these changes in the transcriptional heterogeneity.
Carla,
now the last experiment.
A minute or two.
Thank you. The last experiment to really draw the point that it is IL-4 that changes the transcriptional heterogeneity and that accounts for this, we can test the functional impact. And so we did the same experiments that I've shown you before. We took naive T cells from a non-infected mouse, from a NIPO-infected mouse, or from a NIPO-infected mouse that also received the anti-IL-4, where those changes cannot occur, transfer them into non-infected mice, inject them with ova alum that will activate now these naive T cells. I've shown you before, we can look into the proliferation. In black is the control. In red is what I've shown you before, in significant suppression in the ability of the T cells to proliferate if they come from a NIPO-infected condition. But importantly, and really closing the loop, a restoration in the ability of naive T cells to respond when they come from a mouse that also receive NIPO plus anti-IL-4. Just the last comment is, how long is this effect? I think this is important. If we test this at 3 weeks, which is when the mice can recover from the NIPO infection, you can see that all the genes that are induced in these naive T cells are no longer induced, so we go back to normal. And importantly, if we do the function, I've shown you before that by 1 week, we have a significant change in transcriptional heterogeneity of naive T cells, an impact on their ability to respond. But if we now take naive T cells after 3 weeks of NIPO infection, they can equally respond to their cognate antigen. So this is good news. So for helminths, we can restore the naive T cell transcriptional heterogeneity with a treatment, and we can now hope for a normal magnitude of the immune response. These are the last slides, so I'll be finishing just with a concept. You can think about this, that we can all be in a particular state. Sometimes we get infections like a helminth infection that can dramatically change our naive T cell state. And if we now need those T cells to respond to their cognate antigen, at least while we have a helminth infection, or at least in these mouse models, it remains to be tested in humans, this will significantly impact those naive T cells to respond. But we can clear it and get it to baseline. I think this has implications, and primarily it's important for us to think in our context, in our daily life. We co-evolve with helminths for millions of years. Probably the setting, the threshold of our immune response has been set up in the context of existence of helminth infections, where we have a particular magnitude of the immune response that we can achieve if we have helminth infections. I'd like to propose that, of course, in a more modern world where now is more sanitized and we have less exposure to these helminths, this can change this transcriptional heterogeneity and affect the magnitude of the immune response. So I leave you with the last thought. It's not just genetics, but it's also our experience in the context of infections that changes this transcriptional heterogeneity that can have tremendous impacts in dictating this immune diversity. I'd like to thank the people that did the work. The ones that were involved in this particular study that has been very recently published are the ones indicated with the asterisks. We had wonderful colleagues at Yale and outside of Yale. I'm sure many of you recognize some of them, like Molon. And I would like to very much thank Saurabh Ghosh, with whom I co-direct the lab and I co-directed this project. He's given a talk tomorrow, so stay tuned for other projects in the lab. And the last thing I want to mention is last year we were incredibly fortunate to launch the first global immunocourse right here at IISc. It was one of the most wonderful things I did in my life. You can see here a picture. Here we are. I think Dipankar is somewhere there. Siddharth, I want to really thank. He was my co-organizer. And I want to announce that we'll be having the second global immunocourse in December, now hosted by Ashoka University. If you're a student, a master's student, please take a picture. Here you have a link for the application. Everything is covered. We do fundraising so that you don't have to spend any money to come to our introductory course. So also if you're a sponsor, we look forward to your support. Thank you, and sorry for going a little bit over time.
Thank you very much, Carla, for a really wonderful talk. I think we can have maybe one or two burning questions, which...
Yeah, I promise to be fast in my
yes.
answers. Let's see, I'll try to focus.
So what is the reason that the naive T cell heterogeneity was driven by helminth antigen like NIPO and not by the gut microbes? It seems to be quite surprising.
Yeah, so as I showed, this is IL-4 dependent, and in the context of the helminth infection, you'll have a very robust IL-4 response. You can even measure IL-4 in circulation.
But that is true with gut microbes as well.
Well, you don't have not in the context of the SPF conditions that we have in the lab. We do not have helminths in our facility. So it's possible that in other settings, but in our specific pathogen-free condition, we do not have helminths. Maybe I'll go to the next one, yes.
Yeah, very nice talk. So I just wanted to know that in your infected mice versus that infected mice with anti-IL-4, is there any differential ability to form the memory T cells?
Great question. We were asked about that, and I owe you the answer for that. We would think that because after 3 weeks, this goes back to normal. I think that if your stimuli is beyond that, I think it's going to be okay. But it's a very good question. I think we need to, explore that more in depth.
Yeah, because the recently Rudensky's paper, they have shown that this type 1 interferon is actually in the naive T cells, the memory for
yes, so Sasha and Chrissy has recently shown that there is this type 1 interferon cluster. Yeah. So this is really very timely because many of us have been seeing this. And so they have described that type 1 interferon, which is not the IL-4 responsive, but is the other, has a role in the context of memory formation. But
so in that context, so do you think that helminth versus
could we
this...
continue the discussion
okay,
one-on-one
okay,
later?
sure, I'll catch you later.
Yeah? Sure.
We should, yeah.
Sure. Thank you very much, Carla,
Thank
for
you
a
so
really
much.
wonderful talk. A lot more questions will come.
Yeah, happy to address questions. Oh, okay. I go this way.
You can just keep. I would request the speaker and chair for a group picture. Please come on the dais. Sandhya ma'am. इसी से कनेक्ट करना है क्या?
Stay so small.
Moving on to our next session, which is yet another plenary talk by Professor Ursula Wiedermann Schmidt, Institute of Specific Prophylaxis and Tropical Medicine, Center of Pathophysiology, Infectiology and Immunology, University of Vienna. I would request Chair Professor Hema Balram to take over and guide us through the session.
Thank you, Dipankar, for inviting me to chair the session. The last few talks that I attended have been wonderful. Our next speaker is Professor Ursula Wiedermann from University of Vienna. She has contributed extensively to the field of allergy and cancer immunotherapy. And she's going to talk to us today about the vaccination failures in aging population and immune-compromised individuals. Dr. Ursula.
Thank you very much. Good afternoon. It's a great pleasure to be here in India. It's my second visit, but the first time here in the South. And I want to thank very much Professor Nandi for inviting me to come. Yeah, what I would like to focus today is on vaccine failures, in particular aging and immunocompromised. It fits to the topic somehow we heard before. There is a big heterogeneity in vaccine responsiveness. And there are three topics or questions I would like to address. And the first is, what is vaccine failure? Who is affected and why? And what to do against this. Now, starting with the first question, what is vaccine failures? When it comes to vaccination challenges, we have to see that there are two important aspects actually influencing this. And this is, on the one hand, we have big changes in demographics. As you can see here all over the world, we have an aging society all over the world. It's expected in 2050 that the majority of aged people are older than 80 years. And along with this comes an increase in non-communicable diseases like cardiovascular diseases, metabolic diseases, and cancer. And on the other hand, of course, we have a change in emerging infectious diseases. You all know the major killer that still are really a threat to many people are diarrheal diseases, respiratory diseases, HIV, tuberculosis, or malaria. But we learned the latest now with the pandemic that there is also an increase in emerging viral infections leading to devastating respiratory infection, as we have seen with COVID now. So the problem actually is due to these changes in the demographics that the question really is, do the vaccines we have today that what the majority of people should respond to, will these be effective also in these populations that previously were the minority in our population, namely the elderly or immunocompromised people? So what are vaccine failures and how can you define them? Well, you can define between so-called vaccine-related failures, that is mainly due to problems in manufacturing, that there can be antigenic interference with other vaccines, co-administration, or there can be simply incorrect application, like the wrong schedule or the wrong immunization route. But more importantly, actually, are the host-related failures. They are determined by age, by host genetics, by nutritional state, and immune status. And these host-related failures, you can again divide into two aspects, namely in primary failures, meaning that a person that gets vaccinated doesn't respond to this vaccine from the very beginning. That is mainly due to genetic factors, age, and immunodeficiency. And the other one is secondary failures, meaning that one responds in the beginning, but the response is very weak, and there is an early waning, and there can be also different reasons for this, and both aspects I would like to address now. And that's the question, who is affected and why? We have since 10 years we run an outpatient clinic for vaccination in risk, frail, and immunocompromised people. And this gave us the possibility to produce cohort studies. And this is what we did actually within the last years. We looked at a specific risk population, namely elderly, obese, and immunocompromised. And our question was, how is the level of non-responsiveness in these people? And is there a common pattern, a common denominator of non-responsiveness in these three different populations? Now, coming first to age, you all know that the hallmarks of immunosenescence are mainly due to two. On the one hand, we are facing an impaired immunity that is due to changes in the bone marrow with lower B cell numbers. This is changes in T cell area with due to the thymic involation. This is due to T cell exhaustion, but we also have the problem of so-called chronic infections, like for instance, cytomegalovirus infections that are permanent in our body and that also have an extreme impact on the vaccine responses, and in particular declined vaccine responses. So actually, we were interested to look at these primary vaccinations in elderly people. And before the pandemic, this was not so easy to do. So we had to choose a vaccine that is not very common in Austria. And we have chosen a vaccine against Japanese encephalitis. You know this very well because this is a disease endemic in Asia. In Austria, it is not. And so we had the possibility to more or less vaccinate young and older people on a naive state regarding this antigen and compare the immune responses. And what we clearly could see was that the elderly people, which in the average were about 68 years and the young ones were about 25 years, that the elderly really had significantly lower antibody levels, as you can see here. And if we look at the percentage of those who were really non-responding, this was enormous. 46% did not respond to this vaccine, while the majority of the young ones did. So the question was what was influencing this result, and we were looking at the T cells primarily at that time, or were interested mostly, and we saw there is a shift in the immune responses, and we can see primarily that in elderly there are Th1, Th2 responses, particularly IL-10 more prominent. This is associated with increased levels of T regulatory cells. But importantly, we also saw that the elderly had a high number, 70% had cytomegalovirus infections. And these infectious stage correlated very well with the low antibodies and the low cellular responses. Now, when after some years we boosted this cohort, and what we saw was actually that the elderly could respond to booster vaccination, but in a little bit different way than the younger ones. Firstly, the immune response was lower or slower coming up. And then when the antibodies were found, they were at a lower stage. And importantly is also one aspect which we see not only in this vaccine, but with very many other vaccines, that once antibodies are built in elderly, there is a faster decline. So the elderly people lose their protection much earlier than the younger people did. So the question was, what is the reason for this? What is the how can this be explained? And there was a very nice publication by Lee and Linterman putting up this hypothesis of non-responsiveness in elderly. And what they have shown, and this fits very well to our data, is that on the one hand, you don't have a problem in the so-called extrafollicular responses in the lymph nodes. There is no intrinsic defect in B cells. There is no intrinsic defect in differentiation of B cells into plasma cells. But there is clearly fewer somatic mutations post-vaccination and also a problem in class switching. So what works here is more or less that you have an extra follicular response with a lower number of short-lived plasma cells and a lower amount of antibodies. But what does not work is really building up terminal centers. And this is due to several factors. On the one hand, the dendritic cells don't work very well because they have a defective activation, mainly of interferon gamma signaling. Then there is an accumulation of pre-follicular helper T cells, and there is a large number of regulatory follicular cells. And additional, the follicular dendritic cells are not well in function, meaning at the end, you shortly have here the formation of terminal centers, and there is more or less a very low number of memory cells being developed, and also a low number of long-lasting plasma cells, which explains why these antibody responses wane more quickly in elderly. The second group I want to discuss is people with obesity, and that is really a problem, you know, well, all over the world. More than nearly 40% of the population is obese all over, so it is really a problem. And the problem here is again due to some sort of low-grade inflammation. Other than in elderly, the reasons are different. The reasons for this low-grade inflammation obesity is mainly due to this obese cells, and is like an organ of adipocytes that is built, and these produce a lot of leptins, and leptin is not only having leptin receptors within the brain, where there is some sort of interaction that you don't feel hungry anymore, but the leptin is also leptin receptor is also expressed on immune cells, meaning that leptin can influence the activity of immune cells dramatically. And additionally, this adipose organ is also releasing pro-inflammatory cytokines like IL-6, which will then in the liver foster the production of C-reactive protein, a pro-inflammatory protein. Now, we did a vaccination study in obese individuals. They were really obese. They were had a body mass index of more than 30 to 32, which is really a lot. And what we could see here, clear markers were that we found high leptin levels, high insulin levels, and high CRP levels. And when looking here at the antibody responses, in this case, this was a booster vaccination with a vaccine, tick-borne encephalitis, very related to Japanese encephalitis. It's also a flavivirus, but this is endemic in Austria. So what we saw was that the obese people, they in the beginning responded well, but there was again a very fast decline of antibodies, and this decline correlated or inversely correlated with the metabolic parameters of CRP, leptin, and insulin. When looking at the B cells here in these obese individuals, we saw initially that the CD19 B cells were enhanced in obese compared to non-obese individuals. But when we looked at their memory state, unswitched and switched memory B cells, we could see that they were significantly lower compared to the normal weight individuals. And the question here was, again, is this systemic low-grade inflammation, does this also lead to impaired terminal center function, similar as we have seen in the aging population? And is there reduced B memory cell development? To answer this, actually, we went also in a mouse model of obesity, and what we could do there, we were performing vaccination with the SARS-CoV-2, with one of the SARS-CoV-2 vaccines, mRNA vaccines, and what we could here clearly see, that was also seen in humans, that the obese mice had really significantly lower antibody levels. When looking at their specific, namely SARS-CoV-2 as specific memory B cells, they were significantly lower compared to lean mice. And again, we were seeing an increase in T regulatory cells. So we were looking also in the spleens of these cells, and here only in this very simple histology, you can maybe appreciate that the terminal centers in lean mice are really bigger than they are in obese mice, but really to have this more concretely shown in the future, we are now performing single cell RNA sequencing and using Xenium Prime tissue and pathway panels. Now, the last that I would like to address are the immunocompromised people. And the SARS-CoV-2 vaccination has clearly shown us these were the ones that were of need to get vaccinated, but responded the worst of all individuals. And so again, we wanted to use our cohort from our vaccine clinic to show at this primary vaccination in immunocompromised individuals using an mRNA vaccine against COVID. And here, the example I want to show you is from intestinal bowel disease patients. And when you look at the first responses after 2 vaccines, actually it's not a big difference seen to healthy controls. But what is seen is after 6 months, there is a much steeper decline of the antibodies in the IBD patients. But there is a big variety actually in responses. And we looked who they are and what treatment these patients got. And importantly, it was mainly those patients that got anti-TNF treatment that showed this lower antibody response and a faster decline. You can see this here more clear that people under anti-TNF treatment, they had lower responsiveness, faster decline. They could be boosted, but the responses were lower compared to healthy controls. And if we also looked at neutralizing antibodies against variants of concern, these individuals could not produce any or did not have any cross-reactive antibodies. When looking more closer also in the direction, what has this to do with the B memory cells? When looking at the follicular T helper cells in healthy controls, we know that there is a clear correlation between building memory B cells and an activation of T follicular helper 1 cells. But in people with anti-TNF treatment, we saw that these T follicular helper cells were hardly produced, and you can see also here the activated ones were not present. That was showing also a clear picture, a very interesting picture regarding the production of memory B cells. If you look here on the right side, we have seen that actually these people with under anti-TNF treatment, they didn't hardly build the non-responders antibodies. They also didn't build memory cells, but what they really showed, they showed high levels of plasmablast. And this is really the contrary to well responders, where you see that antibody responses go along with the production of memory B cells. So here again, the question was, is this response we are seeing rather due to an extra follicular immune response? And this idea was really triggered by important data that was generated during the pandemic when infected people that died, where lymph nodes were taken, and it was shown that people who had been severely sick and had a high inflammation during the COVID infection, that these people could not develop germinal center because the production or the development of follicular helper cells were firstly blocked, and also the migration into germinal center was not the case. So that means this was this inflammation that was going on was an important factor of preventing germinal center formation. And we do believe that this is very similar in anti-TNF treated people, that actually we also see here, because there is quite a baseline inflammation ongoing in this anti-TNF treated people, that we do have extrafollicular responses with antibodies, but they are only of low affinity and short-lived plasma cells, while these germinal center reactions are not present or activated in anti-TNF treated patients. Now, last question, what can we do against this? And there are, we have to learn, of course, constantly and try to put these aspects we have seen into account. Well, one thing is that we always do is if somebody is not responding, we try to boost them frequently and also take the shorter intervals. And here is an example of SARS-CoV-2 vaccination in another group of immunocompromised, namely multiple myeloma patients, where initially they also had delay in antibody responses. And what you can see if they get 3, 4, 5 and more vaccinations, finally they really go up with the antibodies, and they can also produce memory B cells. And importantly, when they even have more contact with the antigen, namely also an infection, then you can see that it is really a possibility to get antibody responses, and in particular the generation of memory cells much more on a higher level. The other possibility is to use different adjuvants you would normally use. You know, in most cases, mRNA vaccines are, of course, a new has been a new generation, but in other vaccines, actually, alum is mostly used. But we know that alum is not really affecting too much on the innate system. And as we have learned, the innate immune system, and in particular, the activation of dendritic cells and their action have to be propagated. And that is possible by using adjuvants that are working via toll-like receptors, like here, for instance, toll-like receptor 4. And indeed, if you compare different adjuvants, you can see that it is possible to enhance immune responses, vaccine responses, when you just change or take the right adjuvant. Another possibility is more still in an not commonly used, but it is from that what we have learned from this low-grade inflammation, that it makes sense maybe to block or try to block on a limited level this inflammation by using rapamycin and doing mTOR inhibition. And this has been done actually here in patients prior to vaccination against influenza. And indeed, you can see that the immune responses to all types of the influenza virus can be enhanced if you manage to block this activation or block this or inhibit this non-functioning of the T cells and activate them more. The other possibility is, and we have seen this in hepatitis B non-responder, that actually those who have a genetic background have initially already very high IL-10 levels. So there's a possibility really of using the blockade of IL-10 together, probably with PD-1, and at least in mice, this is shown that activation of T cells can be upregulated by such a blockade, but this is not done in humans yet. This is a very new possibility. Last one is, of course, to change the platform. I mean, the mRNA vaccines were one possibility really to have a better response in non-responder, in low responder, and at least we have seen better effects than, for instance, with the whole cell vaccine. So we could compare this also with SARS-CoV-2. But I also want to mention, because Professor Valenta has talked today, and he has really performed a new vaccine type against hepatitis B non-responders, where he's using not only the HBs antigen, but really expanding the antigen and thereby giving it a better chance for the immune system using pre-S-based recombinant vaccines. And here you can see really that this change in the antigen gives a chance also to the immune system with better neutralization, and not only on the antibody level, but also on the T cell responses, which can be a chance for population of about 10% of hepatitis B non-responders that would be happy to have such a vaccine. Last thing, and I think this is very important for the clinic also when you deal with people who don't respond, is to look for biomarkers and what are indicators that would show you that there is a not a good response, so that you from the very beginning can change your vaccination schedule or look for another vaccine. There are biomarkers studied in different publications. For the innate immune system, it's very often IL-6, CRP, or TNF. For the adaptive immune system, it is also cytokines, but in particular the number of B cells and the ratios of CD4 and CD8 cells. And that is actually what we do in our clinic, that we do in our problematic patients, immunoprofiling, that we really look how is the immune system fit or unfit by looking at the percentages of T cells, B cells in particular on the B cell level, to simply know, does it make sense to make a normal vaccination schedule? So do we have to start from the very beginning in a different way? So I hope I could show you with these different examples that there is actually a common denominator, and that goes via inflammation, which really can have a big impact, for instance, on the production of B cells. That might not be for all patients, but it is just worth really to look in detail in your cohort so that you know what is actually the next step to change the vaccination schedules. Because one is clear, one shot doesn't fit all. So this is something we have realized very clearly meanwhile. And with this, I would like to thank you for attention. This is my fantastic group in Vienna. Here, Erika Garner-Spitzer and Angelika Wagner were the ones working with obesity and age people, Nora Geisler with the obese mouse model. And this is our team for the clinical studies, and under the guidance of Ines Zwarzel, who is really doing a perfect job for all our clinical trials. And of course, I don't end before also inviting you to come to Vienna. We really hope that a lot of you will have the possibilities to join us in Vienna and continue these discussions. Thank you very much for your attention.
Thank you, Ursula, for the wonderful talk and also for sticking to the time. I think we have time for a couple of questions. Yeah.
No questions.
So adipose tissues tends to secrete a lot of IL-4 and IL-13.
Excuse me?
Adipose tissues, they secrete a lot of IL-4 and IL-13. And IL-4 is a differentiating factor for B cells to secrete IgG1, and whereas IL-13, it induces secretion of IgG4. But in your case, why obese people, they were not producing antibodies against the vaccine?
Why they don't produce more of these? I did not understand your question, excuse me.
The question is, adipose tissues, they produce IL-4 and IL-13, which can induce secretion of IgG1 and IgG4 respectively. But in your case, why obese individuals, they fail to respond to secrete antibodies to vaccines?
Well, they did respond actually in the very beginning. They had even higher levels in the very beginning, but they faint, or the waning of the antibody response is much faster. So that is the major thing, is that they are not unable to produce antibodies. I think this is not the major point. There might be examples where also the levels of antibodies are lower, but the main thing is that they cannot keep the antibody levels, and the protection doesn't last very much. And that goes most probably also together with the fact that they probably have more extrafollicular responses, where they can produce some antibodies, but they cannot produce long-lived plasma cells, and they don't produce a lot of memory cells. And that is what we have seen in the patients as well as in the mouse model. So that is more the problem, meaning that they have to be either vaccinated more often, or also that possibly other particular adjuvants have to be taken for them to have a better response of these short-lived responses we get.
That was very nice. The IL-10 you see elevated in some of your groups, the elderly. Do you know whether that's coming from conventional T cells? We often see that in infectious diseases where when strong pro-inflammatory responses are turned on, the cells develop an autologous IL-10 response to dampen things down. Do you think that might be occurring in some of these elderly patients, and that might at least partly explain why their vaccine responses, when they see the antigen again, their main responding cell populations are these IL-10 producing CD4 T cells, be it TFH cells or Th1 cells.
Do you mean in the hepatitis B non-responders, as I said? Yeah, in the beginning. Well, actually, these people is it's very interesting. At least we have seen this there are diverse picture when it comes to antibody and cellular responses. On the cellular response, due to these high IL-10 levels, they are not only non-responders to hepatitis B, but also to other antigens, unrelated antigens. So they don't respond very well to TBE or to tetanus or whatever, even though they might show antibodies. As I said, again, probably over this short extrafollicular loop, but there is really an incomplete immune response going on, which in these cases is more on the T cell level.
Here. So this might be a naive question. So in the hello. Yeah.
Yes.
So in the immunocompromised population, why is infection infection causing more antibody production as compared to vaccination?
Well, we think that it has to do that the repertoire of antigens that are presented there is much higher, you know, because if you have an infection, you have thousands of different antigens being presented, while if you have only a vaccine, in this case, an mRNA vaccine that has one limited antigen, and there we have seen that due to the lack of germinal center formation, it is not possible that there is a lot of diversification, obviously in the B cell repertoire with this one antigen. But if you come with a whole virus, and there are different aspects being presented here, then this might be a factor how you overcome this initial limited diversification of the B cell receptor you find.
That's the last question. We're running out of time. Thank you, Ursula. Thank you very much for the
Thank you.
I request the professor and the chair to assemble for a photograph. Yeah, thank you, ma'am. Next lined up, we have a 10-minute talk, 10-minute technical presentation by UR Advanced Therapeutics. Should I take this one?
Hello. Hello, everybody. Thank you for staying.
just before the tea time. I would not take much time. We are your advanced therapeutic. I'm a startup company actually, and thank you, Professor Nandi, for giving us this great opportunity to present before everybody. So actually, we have won quite a lot of competitions in India and abroad. What we do is we work on designing the peptides, artificial peptide-based biomaterials for tissue engineering and various applications, how we can go for gene delivery, and especially in therapy and drug screening and diagnostics. One of our successful story, I was an academician before. I left my academic career and then started this company after 2020 COVID. So actually one of my success stories is that we developed a peptide-based biomaterial for corneal regeneration. So this is for cornea has 3 layers, epithelium, stroma, and endothelium. We are able to regenerate endogenously both epithelium and stroma. Now we are working on the endothelium as well. So this is a corneal implant. As you can see, this is successfully tested in the human trial, at least among 40 patients. This is now in phase 2, phase 3 clinical trial in India, Sweden, and Ukraine. So this is one patient sample that I'm showing here. So before the treatment or before the implant, they had absolute blindness, 20 by 800. And then within 5 weeks, it's almost like a 20 by moderate vision is restored. And within 6 months, they have a complete vision restoration, almost better than my eyesight actually. So yeah, so that's one of our claim to fame. This was done from 2011 to 2016, and the patient trials were 2016 and carrying forward. And after this, because there is a lack of endothelial layer, which almost 40% corneal diseases are endothelial diseases. So we try to now focus on the complete regeneration of the endothelium. So this is the work that we are doing right now, both using 3D printing and as well as the normal implant-based material that we are preparing and testing it on various animal studies right now. So this is my scientific talk. Now let me go for the sales talk. So because I'm a startup company with low funds, I have to sustain the company, and I'm not deep-pocketed. As you can see, my name is Jagan Mohan Reddy, and I'm not the same person. So basically, because we are designing the entire matrix, we are coming up with the peptides that can mimic the extracellular matrix. So we thought we prepared the some kind of biomaterials that can be supplied as bioinks or for tissue engineering for those who are working in 3D cell cultures. So actually we made different materials. We optimized for mainly this is for retinoblastoma that we have optimized these different peptides. Generally people use matrigel, and we are releasing these ones alternative to matrigels. We can customize this peptide formulation depending upon the tissue and specific type. And also we can tune the mechanical properties and functional properties as well. And actually, because these peptides are non-immunogenic, if you are working on the extracellular matrix component analysis, you can go for absolute estimation of the extracellular matrix that is derived from the cells that you are working on. And this is another peptide that we have developed. Most of you would have been using apoptosis kits, mainly annexin 5, which is a calcium-dependent assay, at least multi-step process. So here what we have developed is a peptide that binds to phosphatidylserine. It's a single step. Just add it to your cells in the media, in your native conditions. You can restore you can study the apoptosis, or you can identify the apoptotic cells. This is our data. As you can see, in the presence of calcium, annexin 5 binds very well. This is the one. Annexin binds very well, and without the calcium, there is no detection of the cell death. But our molecule can detect with or without the presence of calcium. And people have been using, they have been publishing it, so it's in the commercial state. And the extension of that, most EVs people are working on, actually EVs also has phosphatidylserine on the membrane. So we are extending that application, and we are trying to sort the EVs not just based upon their size, but also based upon their lipid molecular content on the above. So that way you don't have to use the antibodies, which are quite expensive kits. So, and actually we are working with a organization in US. Actually, we are trying to sign the MOU in the last stages. I will reveal after that. So once that is made, we are actually developing a contact lens that can detect the EVs through the TS samples. So that's the next code, so that it will be used for diagnostics. And since we work on designing the proteins and peptides, we have the AML facility, everything. So we developed this luciferase. Nanolic is by Promega, which is about 19 kilodaltons. General luciferase is 61 kilodaltons. We have developed a ATP independent secretory luciferase that is stable for more than 45 to 60 minutes in the conditions. So you don't have to kill your cells for the luciferase assay. You can just take the media and you can go and assess your. So we are developing the cell lines, TLR3 and all the TLRs, GPCRs. We can develop water. So it will go on slowly. This is the work we are doing. So we have the capabilities. We got some funding, decent funding recently. So we build our infrastructure. We can do the peptide synthesis to purification analysis. We can do the 3D cell cultures. Actually, I was the first one in India in 2017 to establish retinoblastoma cancer biobanking. So we did that one. And we have the internal entire GPUs and storage system and computing power. And this is our story, startup by an academician in 2021. And we are a group of biologists, chemists, and informaticians as of now. And we are looking big, actually. We are collaborating with a research park, life science research park, which is developing about 230 acres of research environment. And we will be the center of a research organization for that. So we are looking for the support from IISc and all the advisors so that we can develop into a proper research organization focused on innovation and services. So this is us. We are looking for bioinformaticians and programmers. Slowly we will advertise for other fields as well. But, we are looking for, thank you very much for your time. Pardon me? Radiation? Treatment.
If post-radiation, enucleation eye, you are putting a hydrogel implant, and if there is a histopathology showing a high risk factor, you require to give a radiation. In hydrogel, radiation can be given?
Yes, it's a quite transparent hydrogel. That is the truth actually. But the thing is that we are not putting the hydrogel for the retinoblastoma treatment. The hydrogels, you can culture the retinoblastoma cells outside the hydrogels so that they can prepare the tumor models.
Okay.
We are not giving it as of now as a therapy. So that's the future. Actually, definitely we would like to look into the retinoblastoma treatment as well, mainly because the RPE cells, retinal pigment epithelial cells, these are post-mitotic cells like corneal endothelial cells. We are trying to make the hydrogels that can regenerate the retinal pigment epithelial cells more better. Yeah.
Hi. So congratulations on the startup.
Thank you very much.
Huge achievement. Actually, I was interested in the your peptide that can detect dead cells.
Yeah.
So I'm guessing it can't be used as a substitute for the live dead amine reactive dyes that are used in flow cytometry on fixable?
Yeah, actually, you see, live dead assay is very different. This can detect apoptotic cells very specifically.
And
Live
on
dead is
live, not, I mean, unfixed cells, right?
Pardon me?
Unfixed.
No. You can use it for live cells. Yes.
No. I mean, can you use it on fixed cells?
Yeah.
The advantage of live dead is that of the live dead amine reactive dyes is that you can use it on fixed cells.
No, actually, even live cells, you can use it because it just binds to the phosphatidylserine on the membrane of the cell that is about to die.
Okay, I think I'm not getting my question across correctly. See, the thing is, amine reactive dyes, the advantage is that you don't have to use live cells. Because of flow cytometry, often you fix your cells. And even after you fix your cells, they can, they can distinguish between not apoptotic, but live and dead. I guess that was my thing. Okay,
So
and anyway, we can talk about that
absolutely.
later. My other question is, has it been tried on human cells, human PBMCs?
Yeah.
And if it's a
So
peptide,
T cells, we have tested on T cells.
okay, so if it's a peptide, isn't it taken up at the peptide you don't get some sort of a like response or something?
No, actually it's a very small peptide. It's about less than 12 amino acids actually.
But
And it's a cyclic peptide, so it does not create any immune response. Yeah.
Okay. It's a cyclic peptide.
Yeah.
Okay, it's not that it's small because
It's also
CD8 T
cyclic.
cells. Okay,
Yeah.
epitopes are small. All
Yeah.
right. Thank you. Nice
Thank
talk.
you. Yeah, pleasure. Thank you very much. Thank you.
Yeah, we'll now be taking a short break for tea. We kindly request everyone to reassemble by 5:50.
As we now resume, we have more captivating sessions lined up. Next lined up, we have a 10-minute technical presentation by Thermo Fisher.
Thank you. Thank you, everyone. And thank you to the organizers and Professor Nandi for giving us this opportunity to talk about our work here. We have a antibodies-based R&D unit here in Bangalore. And today I'm going to be talking about our group's work on highly validated antibodies for research use in immunology. So I'm going to start off with talking about the antibody reproducibility crisis, what this is, and what we are doing to address this. And I'm going to spend most of my short talk talking about high-quality recombinant primary and secondary antibodies, and also talk about why recombinant antibodies are important, how we are converting our hybridoma, very valued clones of hybridoma into recombinants for the researchers' benefits, and also talk about certain specific case studies, both from primary and secondary antibodies, where we are engineering our antibodies for research use. So the antibody reproducibility crisis, and as we all know, antibodies are critical reagents in research. And as researchers, I'm sure all of us must have had kind of a varying experience when we shopped around for antibodies and when we use them in our assays. Usually not very often we find that antibodies, even when we are shopping for them, have little to no data or no relevant data. When we do purchase them, they do not work in the applications that we would like them to. They have non-specific reactivity, and all of these actually causes a lot of wastage in time, money, and effort. And so there is really a critical need for enhanced antibody validation. So when we talk about recombinant antibodies, I think when we talk about antibodies for research use, there are two main criteria that are very important. One is the specificity, which talks about how specific the antibody is to its target antigen, and whether it only recognizes its target antigen or does it also pick up other related proteins. And the second one is sensitivity. So first, going into specificity, we employ a very vigorous validation and testing strategy for our research use antibodies. We test them in a variety of assays and employ multiple tests that kind of ensure specificity, like employing genetic knockdowns or knockouts, relative expression, or cell treatment assays. We also test these antibodies in very relevant applications for that particular target, so that when the customer is looking for that antibody, they are already very convinced that it would work for their experiment. Moving on to recombinant antibodies, we have recently been seeing that there is an increased customer need and ask for recombinant antibodies. And if we were to compare the three main kinds of antibodies that we typically use, those are the traditional monoclonal antibodies, which are the hybridomas, polyclonal antibodies, and recombinant antibodies. We can see that the recombinant antibodies are projected to grow at a growth rate of nearly twice that of the traditional monoclonal or the polyclonal antibodies. And so why is this? So recombinant antibodies provide a variety of benefits over traditional hybridomas. For example, they ensure lot-to-lot consistency, and they eliminate the risk of hybridoma rifts or shifts that can happen when we maintain hybridoma lines over a long period of time, and sometimes hybridomas can get contaminated. So all of these risks are eliminated when we convert antibodies into recombinants, and we are able to adhere to timelines and processes for production, because these are very controlled processes processes now that we can adhere to. We are dramatically minimizing animal usage in the production of when we switch from traditional antibodies to recombinant antibodies. And because they are recombinant, they can now be subjected to a wide variety of things that we would not have been able to do with traditional antibodies, such as customizing to recombinant formats, such as scFvs or Fabs or chimeric proteins, as well as antibody engineering to confer very specific benefits, especially for research use. So how are we converting our hybridomas into recombinants? So we start with our hybridoma antibodies, and we have developed a proprietary hybridoma sequencing pipeline using our ION platform, where we can very accurately and with high confidence identify the variable regions of the hybridoma cell lines, which can then be cloned into expression vectors and then produced recombinantly. And so again, just to reiterate, I think these kind of this process of converting well-loved and well-known hybridoma clones of many targets into recombinant antibodies maintain all the exact antigen-antibody interactions and the same epitope recognitions, but with the added benefit that now they are available in a recombinant format, and so we can ensure better performance for the researcher. So this is an example of a SOX2 hybridoma. The clone is BTJCE, which has been converted into recombinant format. And as we can see here, the recombinant antibody is very comparable to the parent hybridoma in multiple assays that we have tested in Western blotting, flow cytometry, as well as immunohistochemistry. In addition, we established that these recombinant antibodies are highly specific. In this experiment, the cells have been treated with siRNA targeting SOX2, and the specific nuclear signal that's being picked up by the recombinant antibody has been dramatically reduced, thus confirming that the antibody is highly specific to its target antigen. These are other examples, and we are adding to this every day. We have a program where we are converting very well-used and historically well-cited hybridoma clones into recombinant formats so that they are more beneficial for the research community. And I've just listed some of the antibodies to targets that are relevant to this audience here. And so I hope you all had a chance to stop by poster 113, in which Nazia had talked about a lot more of hybridoma and recombinant comparisons and some of these activities that we are doing here. Moving on to antibody engineering. So when we are mainly undertaking antibody engineering to enhance antibody sensitivity, and because we have a wide portfolio of research antibodies, any strategy that we take, we want to be able to scale it to many different kinds of antibodies to different targets. And to do this, so our strategy revolves around engineering the Fc portion of the antibody, and we are engineering both the recombinant primary as well as the secondary antibodies. And so one of the main focus areas that we are focusing on for engineering is to enhance sensitivity. And why do we need to enhance sensitivity? It comes with several benefits. It results in a stronger signal in immunoassays. Increasing the antibody sensitivity also lowers the limit of detection, thus expanding application coverage in certain cases for the particular antibody. We are also able to detect low abundant species or isoforms, and we are also engineering to increase signal to noise. So this is an example of an engineered Parkin recombinant rabbit antibody, in which we have engineered the Fc portion of the Parkin antibody. And in this slide, I'm showing you a lot of data that compares the enhanced sensitivity of the engineered antibody to the parent So in Western blotting, we can see that the engineered antibody produces a much brighter signal compared to the wild type antibody, and the enhancement is anywhere between 2 to 8-fold increase in sensitivity. We are also able to we are able to significantly increase it in other applications such as flow cytometry and immunohistochemistry. Moving on, I'm going to also talk about chimeric formats of antibodies, where we offer antibodies with the same clone, so the same antigen binding site, but with different species backbones. This really enhances the flexibility for the researcher in their experimental design. And we're also, because now these antibodies are recombinant, we are also offering humanized versions of very important targets that are very useful as controls in preclinical studies. Yeah, I'll very quickly go on to secondary antibody engineering, and we offer secondary antibody pools of recombinants, which maintain the sensitivity of polyclonals, but the specificity of monoclonals. You can see here that our GOAT anti-human IgG light chain superclonal antibody is very specific to the kappa and the lambda light chains of human IgG, with little to no reactivity for the other species. And we engineer mutations into the Fc portions that reduce the or eliminate the binding, non-specific binding of the antibody to Fc gamma receptors. This usually increases a lot of background staining in assays such as flow cytometry or immunocytochemistry. And by introducing these mutations, we can actually do away with the Fc block step that typically is used in flow cytometry. Sure. Yeah, and so just to summarize, I think we have been able to demonstrate the benefits of antibody engineering for both primary and secondary antibodies for research use, and want to just call out all the contributors from the team who have been primarily contributing to this work, as well as the larger teams in Vitrogen. Thank you all for your time. Yeah.
Thank you, Thermo Fisher. Now we move on to our next session, which is a special lecture by Professor G.P. Talwar, the Talwar Research Foundation, New Delhi, India. I would request the chair, Professor G. Padmanabhan, to now take over and guide us through the session. This is view full screen.
That's full screen, full screen.
No, this is view for him on the
Chairman,
laptop.
chairman को तो वक़्त दो.
I will go there.
Well, of course, he is my old. No, we were of the same age, more or less, and or maybe she's younger than me.
I am younger than you.
But it's a pleasure to have him chair this session, which I've been asked to do.
Just yeah.
Please do.
What is it? You are presenting him here?
Yeah. You can pass the speech.
Friends, I have a write-up which does not do real justice to the personality of Professor Talwar. We consider him as a living legend, without exaggeration. Of course, this write-up is there, but so I will just quickly go through. He was Professor Head Biochemistry, All India Institute of Medical Sciences, Jawaharlal Nehru Fellow, Scholar in Residence, National Institutes of Health, Bethesda, Founder Director, National Institute of Immunology, Welcome Professor Johns Hopkins, Baltimore, Professor of Eminence, International Center of Genetic Engineering and Biotechnology, Director Research, Talwar Foundation from 2000 onwards. Received many awards, National Award in Biomedicine, Basan Devi Amirchand Prize of ICMR, National Lecturer UGC, J.C. Bose Award of Life Sciences, FICCI Award, Sundarlal Hora Golden Jubilee Commemoration Medals of INSA, Jalma Trust Prize in Leprosy, Ramesh Bhatt's Birla National Award, Glaxo Oration, NAMS, Ranbaxy Research Award, Mestrioni Siegel Award, Friedrich von Siemens Lecture. He has been the president of Indian Immunological Society, Endocrine Society of India, Indian College of Allergy and Applied Immunology, Association of Clinical Biochemists of India, and Indian Academy of Vaccinology. Elected as the president of International Society of Immunology of Reproduction and first president of the Federation of Immunological Societies of Asia, Oceania. Professor Talwar was conferred Padma Bhushan by President of India, Officier de la Légion d'Honneur. I hope I'm pronouncing it right.
Légion d'Honneur.
Okay, you know French, by President of France. Well, just add a minute or two. I said, he has had a not an easy career, I should say. He always says both are GP. I am called GP, he is also GP. But he says, I am followed Talwar, I carry a sword. You are Padma, you carry a lotus. And it is very typical of him. He's a fighter all his life. Yes, he has to, if you look at his career, he lost his mother 8 days when he was a child. From that time onwards, I read his history. Youngsters should read Journal of Bioscience 2005, which gives the biodata or, it's written his biography, short biography. It's worth reading it, it's... And, he was invited to France. He went to France, got his DSc from Pasteur Institute, or equivalent to from Pasteur Institute. And when he came back to India, he went as a von Humboldt fellow to Germany, and he came back to India, and professor of biochemistry at All India Institute of Medical Sciences. And it was a nondescript building. It's not a grand building which you see today when he went there. And he not only built the biochemistry department, when I go through the history, I think he built the All India Institute of Medical Science itself. And that is Talwar for you. This is what he has come through. And he's a fighter all the way. He's not been. And he clearly says, for example, he was the first one to discover the fertility control vaccine in India, which could not make it because what he calls as crab syndrome. It doesn't allow people to climb up. That's the other one he has made is the Lepre, which probably he will be talking today for other applications, which I don't want to take the limelight, especially leprosy. To my mind, if leprosy has been eliminated in this country, Talwar's vaccine is largely responsible. I do feel like that. So it's indeed a great pleasure for me to formally introduce. He doesn't need introduction, but the author should know, youngsters should know. You're going to listen to a living legend. He has made enormous contributions to immunology in India, vaccinology in India. And he's going to talk to us on Mycobacterium indicus prani. Prani stands for Pran and NII. Pran is his family name. NII is National Institute of Immunology. A unique immunoprophylactic immunotherapeutic vaccine for two major infections, leprosy and tuberculosis. Professor Talwar.
I want to also thank Dipankar Nandi. You see, he's put IMMUNOCON on a different level. I know from the beginning IMMUNOCON, never did it have as much audience as it is, never so many speakers, and a very nice program of them. It's not just chosen elsewhere. So I think it deserves credit. Okay, now let me go to this subject of today. In 1970, Dr. Howard Goodman, who was the chief of immunology at that time in WHO, he came with Elvin Cabot and Barry Bloom and maybe one other immunologist of repute, and asked me to take up the headship of WHO Research and Training Center in Immunology for India and Southeast Asia region. And I was reluctant. Then he told me that, look, India has the world's largest number of lepers. Do I expect Americans to come and solve our problems? This really ashamed me. I knew nothing of leprosy, by the way, at that time, but it is a very ugly disease. And India had the largest number of lepers in the world. Obviously, I said I will take up whatever he's offering and try to do whatever I can on that. भाई ये आगे करने के लिए क्या है? This is for? Okay. So, the very first thing was to know why people get leprosy. So we did a basic studies to understand what is wrong in those who become leprosy patients. All of us don't. So what is wrong with them? And it came out that the people who become lepers, they have an inability to mount cell-mediated immune response to M. leprae. So having learned that, The next very question was, can we do something to upgrade their lack of immunity? And they become immune to, so to say, not becoming patients. So for that, we eventually developed an immunotherapeutic vaccine based on heterologous approach. You see, heterologous because it had to be, used. In any case, M. leprae is not cultivable easily, and maybe not cultivable. And also, so anyway, we thought so we learned first what is wrong in the immune responses of these people who become leprosy patients. And then, the obvious next question was, can we do something to enable that those who lack immunity against leprosy or M. leprae can have that immunity to resist becoming patients? Ultimately, we've developed a non-pathogenic, you see, after doing all the study in order to boost the immunity, we worked on cultivable mycobacteria we obtained from the international storage facilities, plus also our own. And eventually, by these studies that I will probably describe briefly, because I was told I should restrict myself to 12 minutes, which I don't know how I can do. But anyway, but the whole approach was to take a cultivable, because ultimately if we have to make a vaccine and it has to be used by a large number of people, it must be cultivable. And it has to be a sort of such bacillus which can boost the lagging immune responses of the individual against M. leprae. So in the case of MW, which was the code used for that, eventually this bacillus has been, it is cultivable, but it has also been, its gene sequence has been determined by the laboratories of Hassan and Anil and Akhil Tiwaris. And it looks to be a vaccine, a sort of a bacillus which is not yet been brought into usage. So it is, let's say, new for human usage. What was interesting is that this bacillus shared antigens not only with M. leprae, but also with M. tuberculosis. So therefore, there was a unique opportunity that two of the major diseases, and I think tuberculosis is a major disease of the country, over and above the fact that we may have the largest number of lepers. So you see, therefore, it's a very interesting finding to discover this mycobacteria. Its key antigens are heat stable. Therefore, it could be used as an autoclaved vaccine, and therefore there will be less problems of toxicology if the bacillus is already killed. This is what the bacillus is, and let me tell you that this picture of the bacillus was taken right in this institute through our organizer, Dipankar Nandi, he made this, I asked him to give and this is what it looks like. It's rather an interesting bacillus, quite solid in its action also. And this is what it is like. If it is used in combination, you see, nobody can tell to people who are dealing with leprosy to give up the drugs that they use. So, but the combined immunotherapy with chemotherapy, let them give the drugs, but let them also use MIP as an additional element in it. Then interestingly, the inclusion of MIP with other drugs improves the cures. There are no granulomas formed. The reactions number and severity are reduced, and leproamine conversion, which is a ability in future to not to catch any new infection, that is increased, and it is effective in slow responders. This is how, this is totally unexpected. See the leprosy patients, they are now treated with the drugs plus MIP. And look, this patient at the top is when she was come into the fold of treatment after 4 doses of the drugs plus MIP. This is what she looks like. And after 1 year, she becomes a normal person. You know, you never expect that this woman has ever been a victim of leprosy with all those ugly marks. Same here, another patient I show you. After 5 doses, he's clear. You see, normally these leprosy patients have in their back these blemishes. And look how clear his back becomes if you have used the MIP along with the drugs. Another thing, another patient. Normally, drugs do not remove these lesions. After, even if they are cured by drugs, the lesions persist. But using MIP along, you have this miraculous sort of effect. This fellow was from Nepal, and he came and touched my feet and said, "Kaise aapko shukriya ada karo?" So I was touched. Another one. Another one. The MIP is approved by not only the Drug Controller of India, but also by the US FDA. It's approved in America also. And it is licensed to Cadila. Cadila, you know, their owners, they approached me. And I said, well, if they can make it, why not? It should be easy to distribute to others. I never knew that they were use it for many other purposes, psoriasis and cancer at much higher rate, and give low supply for leprosy. You know, many people who are leprologists blame me that Cadila does not supply the MIP. But what to do, this is the companies. Now let me tell you about tuberculosis. There is a category of tuberculosis which is quite difficult to treat. And there is also research carried on such patients with the multi-drug use for tuberculosis. But in one group, they were also given MW, which was the previous name before its name changed to MIP. And you see 48 out of 49 patients in that group were completely cured. And the one who wasn't was for some reasons that he didn't take all the treatment properly. And with MDT alone, without MW or MIP, there was not all of the 27 patients in this group were cured, but only 21. So the cure rate is much lower if with drugs alone. But if you include MIP, it rises to nearly 100%. And another property of those who had received MIP is that there are no relapses. You see, the light blue is the one who received the MW or MIP in addition to drugs, and the dark blue are the bars of those who were given only drugs. And you see in two years, there is a relapse in a very large number of former leprosy patients, whereas in those who received also MIP or MW, the relapse rate is very low, hardly any. Now you see this I never knew, but our professor in my institute, All India Institute of Medical Sciences, he told me that he has patients with very ugly anogenital warts. And he has been giving all types of drugs and nothing is working. So he said, "Can I use your birth control vaccine?" And no. It's not a birth control vaccine, but he asked me at that time, ultimately it was the MIP that he used. MW, MIP, what I'm talking about. And this is what happens. See on the top, the gentle wart, and see how clear that whole thing becomes. It absolutely clears up, which according to him, he didn't find any drug doing. See, on the feet, absolutely clear feet after using MIP. This is a slide which is in fact the work of Dipankar Nandi. And Nandi, would you please comment on it? It was carried out by him. No, you say something. This is what you did.
So one of the ideas was to look at the effect of MIP on whether it affects tumor progression or not. So that is what was done. So we used a myeloma, SP2O myeloma, and you can see in the control or PBS treated, there is growth of the myeloma. And then with MIP, there is reduction. So that was the idea. So that's shown the MIP given pre-treatment, which is -1, Then post, once you give the myeloma and then wait for 3 days, because mostly, you're going to see it after tumor develops. And then if you give it at a later point, you don't see the effect much. So you have to, there is a window, either -1 or plus after the tumor is given, and to try and see the effects of MIP. Yeah.
So you see those tumors in the third row almost becoming small. Hence, this to make it a summary of what MIP is, it's a potent invigorator of immune responses. And it is applicable to a number of conditions where many times there are no drugs, but they are cured by MIP. And as somebody has already, as my former colleague and continuing friend, the chairman, Professor Ganguly, has already told you that it's a marvelous vaccine. Its gene sequence is determined, and it expedites bacterial clearances of granulomas, upgrades CMI responses of BLL patients. So, it has a number of very useful properties, which in situations where they are difficult to treat has provided a way to get rid of the problem. And these are the credits which must go. And I'm very proud that it is named after me. The people who, did the gene sequence, they said that your name must be there. Then I added, Pran is my family name, and NII is the National Institute of Immunology, which was in fact founded by me. I was the first founder director of that institute. So I'm very thankful to you for asking me to speak here. And if there are any questions, please don't hesitate, load them on. I'm there to answer any questions if anybody has. If not, then I will take leave.
So hello, Professor GP Talwar. Here.
Okay.
So my question is that you saw data about it also working as a for treatment of drug-resistant TB, where only drug-resistant tuberculosis when was treated with drugs.
सुनाई नहीं दे रहा, आप क्या साफ तरीके से पूछिए क्या
Okay.
यानी समझ नहीं आ रही जो आप क्या पूछ रहे हैं. अब किसी अपने किसी colleague को कह दो कि सवाल डाल दे.
So when you saw data about it treating M tuberculosis, the drug-resistant tuberculosis, was it tested without the drugs, only the MW? How much effective was it?
No, you see, you cannot conduct. The DCGI will not give you permission to conduct such trials. You have to give the drugs. But you see, in one case, you add your so-called new candidate. So you have the control is with drugs alone, the normal drugs which are used, and Then the experimental is the drugs plus MIP.
Okay, thank you.
Anybody else would like to ask anything? You are welcome.
Very short. Sir, we can contact some other company. We can leave the Cadila and then the vaccine should be developed and things should go on.
Yes, I think if you can do that, you will be serving a cause, because you see it is 20 years since Cadila was licensed. And in the agreement, only 5 years was the exclusive license to them. Now we are free to give it also to any other company who would like to make it. Make it and not become like Cadila, serving it for sclerosis and for some types of cancers. So you see, as this is a potent invigorator for many other purposes. So therefore, the companies sell more where they make more profit. But in your licensing, you would lay down that it should be not a high price, so that it's affordable for leprosy and tuberculosis patients who are obviously not very rich. A photo? Yes, with great pleasure. With great pleasure. ये तो मेरे हमजुल्फ है.
Our heartfelt thanks to Professor G. Padmanabhan and Professor G.P. Talwar. Thank you so much for the group lecture, sir.
Thank you.
Thank you, sir. So you need help? We will be moving to our next plenary talk by Benedict Seddon from Institute of Immunity and Transplantation, University College London, Royal Free Hospital. I would request the chair of this session, Professor Archana Singh and Professor Ravi Gutti, to kindly take over and guide us through this session.
Good evening, everyone. And after this spectacular motivational talk by Professor G.P. Talwar, we move on to the next session, the plenary lecture 5, which is given by Professor Benedict Seddon. And to give a brief introduction about Professor Seddon, he has done his bachelor's degree in biochemistry from University of Oxford, followed by PhD in immunology from University of London. And he did his postdoctoral research at National Institute of Medical Research. And currently he is serving as Professor of Immunology at University College London. His research focus is on T-cell biology and immune regulation, autoimmunity and immune tolerance, and cancer immunology. He received several awards, just to name few. He's the British Society of Immunology President's Medal holder, and he's a fellow of Academy of Medical Sciences. And with this, I invite Professor Seddon to give his talk on the ontogeny of regulatory T cells in health and malignant disease. Professor Seddon.
Thank you very much. Thank you for the kind introduction, and I have to also thank Dipankar and the organisers for the magnificent opportunity to come and present our data. I came here expecting to learn some immunology, but I wasn't expecting to learn new English as well. So I'm very grateful for that. Thank you. So the work I'm going to present today is as I said, is about, I'm going to tell you some work we've been doing about understanding the ontogeny of regulatory T cells, which is a sort of topic that's been very close to my heart for many years, actually. And it's work done in collaboration with the lab of Andrew Yates, who's a computational biologist in New York, and my lab at UCL. So historically, my lab has been very interested in the mechanisms which regulate the development and maintenance of the T cell compartment throughout the lifespan, and how a healthy balance of different T cell populations is maintained throughout life, because this is obviously very important for normal, healthy immunity. And in particular, that balance is especially important for regulatory T cells. And so obviously if you have an imbalance of regulatory T cells, then this can result in different sort of pathologies. So if you have insufficient regulation, then this can open the door to autoimmunity. But conversely, if you have over-regulation of the immune system, then this can prevent important immune surveillance against sort of malignancy and transformation. So understanding the dynamics and mechanisms regulating the regulatory T cell compartment is clearly a very important question. So we've sort of done a lot of work using mouse genetic models and fate mapping systems to understand the biology of conventional T cells. And so we thought it'd be interesting to use those same systems and models that we'd use for the conventional T cells to try and better understand the maintenance of the regulatory T cell pool. And this is quite a challenge, because for the regulatory T cell pool, it's a little bit more complex than conventional T cells, where you have this quite sort of simple linear progression of development in the thymus and then activation and differentiation in the periphery. And one of the complexities of the Treg pool is that there's actually sort of multiple sources of by which Tregs can be generated. So we know that they're generated intrathymically during selection events, but studies of looking at sort of microbial responses in the gut clearly reveal that there's alternative sources of Tregs whereby you can get conventional T cells, given the right cues, can differentiate into FOXP3 expressing Treg, and the gut has a sort of mixture of sort of thymic and inducible Tregs. So the question for us was really trying to understand how this peripheral recirculating pool is maintained, where actually it's far less clear what the relative contribution of thymic and inducible Tregs is to the maintenance of that pool. And in particular, we're interested to try and understand, if we could gain understanding about the maintenance of the compartment, what implications this might have for a tumor scenario, where of course you get a lot of infiltrating T cells, both of conventional and regulatory type, and of course interfering with that sort of the regulation has been a very important sort of target for immunotherapy. So the systems that we use very successfully to try and better understand the dynamics of conventional T cells, systems I've shown here is a system we term temporal fate mapping, which is a chimeric mouse model whereby we can specifically ablate hematopoietic stem cells in mice by treating them with this conditioning drug busulfan, which has this very useful property that it targets stem cells but not mature peripheral lymphocytes. So the existing mature cells are maintained in an intact state, and this contrasts with sort of conventional conditioning regimes such as radiation, which really wipes out both HSCs, but also all mature cells. So with this busulfan drug chimeric system, we can ablate the stem cell compartment and we can replace it with a congenitally labeled stem cell. And then with intact peripheral compartments, we can observe how the progeny of the new stem cells, which have a genetic label, and how they infuse all the mature compartments. And the dynamics of this infusion and the extent gives us actually a lot of information about the cellular mechanisms which are responsible for maintaining those mature compartments. So just showing you some sort of illustrative data from the conventional T cell setting. So first of all, this is just looking at absolute numbers of naive and memory T cells within these treated mice. And you can see that they match very closely to what you see in a normal wild type mouse. But following transplantation with the stem cells, you can see very nicely that you get a very rapid donor reconstitution in the thymus where the cells are being made. And then in different compartments in the periphery, you see different varying levels of reconstitution. And as I say, the dynamics and extent of this reconstitution varies between the different T cell subsets and gives us insights into how these different compartments are being maintained. So we use this very successfully in the conventional T cell setting to get us insights into regulation of naive and memory T cells, but we wanted to really turn these models to look at regulatory T cell compartments and try and understand what was going on there. So we know that the Treg compartments, peripheral compartment is very heterogeneous, but at just a very simple sort of high level, it's very simple, easy to see that actually there's heterogeneity within the Treg pool in a very similar way to what you observe with conventional T cells, where you have sort of naive-like cells and effector memory type cells. So we are interested to try and understand the dynamics of those sort of very crude populations within the Treg pool in the first instance with these systems. So first of all, I mean, just to sort of highlight that these are two quite different pools of regulatory T cells. This is just looking at the numbers of cells that you find in experimental mice over the life course. And what you can see is that the naive Treg pool sort of expands very rapidly early on in life and then reaches a plateau. And the memory Treg pool establishes much slower, but gradually increases over the lifespan and over mice. And this is reflected very nicely just by looking at the sort of naive memory ratio that you observe in mice over time, which starts off as a very high ratio, but as the memory compartment, Treg compartment expands over time, then you can see actually there's that memory pool overtakes the naive compartment in size. So then when we generated our chimeras and we observed then asking what does the infusion of the donor cells look like in these two different compartments. So this is what's shown on this slide here. So if we focus firstly on the naive phenotype regulatory T cells, as expected, because they're being generated by the thymus constitutively throughout life, you get a very nice, clear dynamic of donor reconstitution within this naive compartment, which is what we'd anticipate. And it reaches about sort of 70 to 80% replacement of that compartment over the space of about a year. But what was very striking is that when we look at the experience, the effector memory phenotype Treg, that actually there as well, we can see that it's a distinct dynamic, but there's very clearly constitutive generation of these effector memory Treg over the life course of a mouse. So the question for us then is, where are these cells actually emerging from? So there's a number of different scenarios that you could envisage. So of course, one potential precursor for these effector memory Treg is actually just developing from naive Treg through an activation process. But of course, we know that inducible Treg can also contribute to the Treg pool. And so in principle, we could have an input from any of these different stages of differentiation of the conventional T cell pool. So we were very interested to try and see if we could address try and understand whether there was a particular bias for the source of effector memory Treg in these mice. And so the system that one approach that we'd used with the conventional T cells was actually to try and better understand the experimental data that we observe in the busulfine chimeras, the dynamics of reconstitution and the cell numbers using mathematical modeling to try and understand how different populations might be linked to one another. And so this is the first approach that we took. So this is work that was undertaken by Sankit Rane, who's in the audience today. And so we used a very simple mathematical model where we consider that the generation of these cells is a sort of two-stage process. It resembles an activation process, so there should be a sort of a fast cycling compartment of cells, which then transitions into a sort of slower, more quiescent population. So just really a very simple kind of activation type of setting. And then in terms of the modeling, we use the data from our busulfine chimeras, where we looked at the dynamics of the reconstitution and cell numbers, and then we ask which of the different precursor populations can best explain the data that we observe with the effector memory Treg. So so Sankit basically generated 4 different models which considered these 4 different sources, and then simply asked which model gives the best explanation for the data. And so what we found is that actually, if we for naive Treg as the source, as the precursor, as the sole precursor for effector memory, this appeared to give the best description of the data. And this is kind of an illustration of the kinds of fits that the mathematical model would predict. So it captures the cell numbers and the dynamics of the donor cell reconstitution very well, in spite of having different age cohorts of mice. And quite critically, it also captures the Ki67 levels that you find in donor and host cells, where actually the Ki67 is much higher in donor than host cells, which again kind of speaks to the sort of activation-like process. But if we look at the different models statistically, actually some of the other sources actually did a pretty good job in describing the data as well. So the mathematical model gave us a lean towards naive Treg as the precursor for effector memory Treg, but this is not sort of unequivocal support for that, and actually sort of other models did a pretty good job as well. So it gave us some sort of indication, but it wasn't unequivocal. So we really wanted to try and validate this in a sort of independent setting. So there we then turned to alternative fate mapping systems, mouse genetic systems, to try and address this question. So this is now the work of Sanmoy Patak, who is a postdoc in my lab and is actually an alumni of the IISc. And so he came into the lab in particular to try and understand the ontogeny of Treg using this FOXP3 Cree fate reporter mouse. So in this system we have, it's a FOXP3 inducible Cree. So it's expressed only in FOXP3 expressing cells. And then if you treat the mice with tamoxifen, it'll switch on this tomato reporter. So any cell that happens to be expressing FOXP3 at the time of the tamoxifen treatment will become red. So that doesn't tell us where those regulatory T cells have come from, but we reason that if we, with careful observation about how those regulatory T cells disappear, this actually can give us some information, some insights into their ontogeny, because we know that there's dynamic replacement throughout the life of the mouse. So indeed, if we just look at the sort of time course here, so one very important thing is if we focus on the tomato positive cells, then actually, once at least in the steady state in these healthy mice, cells that are regulatory T cells generally remain regulatory T cells throughout the life of the mouse, and we don't see much evidence of reversion to a conventional state, for instance, and that supports work that Rudensky published some years ago. But what is clear is that the number of tomato positive cells does decline with time, and that's because we know that there is de novo generation of Treg, which after the tamoxifen treatment will be tomato negative. So as I say, we reason that actually there should be, if we can look at the dynamics of that tomato negative Treg development, this should give us some insights into the mechanisms of development. So this is just considering the 2 different scenarios. So if this effector memory Tregs are predominantly coming from a conventional source, then what we expect is that the tomato negative cells should rise with a relatively quick dynamic. So they're basically coming in from cold precursor population which don't have tomato expression. So that the new infusion should dilute the existing tomato cells pretty rapidly. On the other hand, if the Treg actually derived from a naive, pre-existing naive Treg population, which is already red itself, then the dynamics of loss of tomato expressing cells and the increase in tomato negative cells should actually be pretty slow, because actually there's a natural delay in cells having to infuse first into the naive Treg compartment. So in sort of validation of this approach, we first looked at the dynamics of tomato expressing cells in the naive Treg compartment. And what we can see very nicely is that there's actually a pretty rapid infusion of tomato negative cells into the naive Treg compartment. And this spline here actually is taken from our busulfan chimeras, which is the spline of donor cell infusion into the naive Treg compartment. So the two systems actually show very similar dynamics and kinetics of the generation of new Treg from the thymus. So then the question for us then is, what does this look like in the effector memory Treg? And what we see here is that if the spline here shows what we would expect, this is the expected new effector memory Treg generation that you see in the steady state. But if you look at the frequency of tomato negative cells within that compartment, actually it's pretty static, and we don't see a sort of rapid increase in those cells at all. So that really suggested to us that the precursors of these effector memory Treg, which are being generated throughout the life course, actually do derive from a pre-existing tomato positive cell, which is most likely the naive Treg pool. So this is suggestive of, as I say, a sort of linear development from naive into effector memory Treg. So the other question that occurred to us is that if these effector memory Treg are being generated throughout the life course, what are the antigens which are actually driving their generation and development? So we know that the mucosal and microbiota can drive inducible Tregs in the gut, and so it's a reasonable thing to suppose that this could also be driving these circulating effector memory Treg as well. So we address this in two different ways. So first of all, we examined the numbers of naive and memory Treg in germ-free mice which don't have any microbiota. And what we see here sort of very clearly is that actually the numbers of naive and effector memory Treg in either SPF or germ-free mice are actually very similar. So if you take all the microbiota away, that doesn't seem to affect the generation of naive Treg, which you'd anticipate because they're thymically derived, but critically the effector memory Treg are present in completely normal numbers, and that's regardless of whether you look at sort of young or old mice. And looking at Ki-67, we can also see that their dynamics of turnover are very similar as well. So it didn't appear that the microbiota were affecting the development of these cells, at least in the steady state. So the other experiment that we're able to do rather sort of serendipitously was to ask the sort of opposite question: what happens if we make the mice much dirtier? And this kind of happened in a sort of inverse way that my lab originally established at the NIMR in Mill Hill in North London, and my lab relocated about 10 years ago to UCL. And during this time, Thea Hogan, a postdoc in the lab, was undertaking these busulfan chimera experiments, and she was conducting them both at Mill Hill, and then she continued the experiments at UCL. And we quite quickly realized that actually the behavior of the chimeras was actually very different between the two different sites. And to cut a sort of long story short, it turns out that the facility that we had at Mill Hill was actually relatively dirty, which is no slur on the technicians or anything there, but we had open cages and water, tap water to feed the mice. And actually when we moved to UCL, we had IVCs and we had irradiated food and diet. So it's quite a big change in their environment. And this was reflected very much in the makeup of the conventional T-cell compartments. So if we look in the dirty and the clean at the ratio of memory to naive cells, we could see that actually whether we look at CM or EM conventional T-cells, there's an expanded ratio of memory to effector. So these dirty mice actually have many more conventional memory T-cells. And so this was actually quite a good setting then to look at the regulatory T cells and see whether this sort of increased sort of microbiota exposure could be influencing the Treg development. And so we looked at this in two ways in the chimeras. We can first look at the dynamics of the donor infusion between the clean and the dirty. And we can see that for naive Tregs, where they're obviously all coming out of the thymus, there's not really much impact of microbiota there. But for the memory Tregs, this donor infusion dynamics was actually very similar, regardless of the health status. And this is really nicely demonstrated when you look at the absolute number of donor cells which are generated in these chimeras at different times after transplant. And for these, for the memory Treg, actually, for clean and dirty, you are generating the absolute same number of cells in those two different environments. So we really didn't see any evidence that for a role for sort of microbiota or foreign antigens in driving the generation of these effector memory Treg. So this kind of strongly suggested that we are looking at a sort of self-antigen driven process in driving these Tregs, so that there's some subset of naive Treg which are selected on self in the thymus. They will encounter self-antigens in the periphery, and this will allow them to become activated and generate and feed into this effector memory Treg pool. So then the question for us is, what is the impact of this on our sort of understanding of Treg in the tumor microenvironment? And so over the years, there's been a considerable amount of effort trying to discriminate naturally occurring thymic Treg from inducible Treg using sort of a variety of different markers, so Helios or NRP1. But it's become evident in recent years that these are really not very reliable markers to distinguish these two different sources of Treg, and actually, some of the data that some of the conclusions about the role of inducible Tregs in tumor microenvironment came from these studies relying on these kinds of markers. So we thought it'd be interesting to revisit this question using the fate reporter systems that we had available to track the behavior of the different Treg populations. So we did this very simply using the FOXP3 Cree reporter mice. So we could treat the mice with tamoxifen. So we label all the Tregs. We give them a little bit of time, a couple of weeks to settle down. And then we can inject a sort of variety of different tumors into the mice. And then 2 or 3 weeks later, we recover the tumors and we just look at the phenotype of the T cells which are infiltrating those tumors. So this is just showing you the sort of typical tumor infiltrate, and we can see sort of very nicely we've got CD4 and 8 T cells on our tumors. There's a substantial population of regulatory T cells, and they all have this kind of activated effector memory phenotype. So then what's the phenotype of these cells in regard to tomato expression? So if we look at a variety of different markers, so if we look at sort of PD1 as a sort of activation marker and Ki67, we can see that the Treg coming into the tumor are sort of proliferative with a sort of activated phenotype. But if we look at the tomato reporter expression, it's really clear that all the Treg that are coming into the tumor seem to derive from pre-existing regulatory T cells from the circulation, because they have this very high level of tomato expression. We validated this to see that this wasn't just a sort of feature of one particular tumor model. We went to a couple of other tumor models and we see very, very similar results. So in these 3 different independent settings, we can see that the Tregs infiltrating the tumor really carry this high tomato level, suggesting they come from pre-existing Tregs. So then the final question that we sort of wanted to address was, again, this sort of question of specificity. What's driving this infusion of these Tregs into the tumor? And this, I think, that trying to understand whether it's a naive or a memory Treg, which might be the predominant source of these tumor infiltrating Tregs, is obviously, I think this is an important question because it does speak to issues of specificity. So if it's a naive Treg source, then this would sort of imply perhaps a role for tumor neoantigens in activating these naive Treg and drawing them into the tumor. But if it's actually a sort of memory Treg that's the predominant source, then this suggests that actually it's a sort of self-reactive Treg which is being drawn into the tumor. And I think these are sort of 2 sort of subtle but important distinctions in the specificity and ontogeny of the cells. So to try and address this, we sort of reverted to our busulfan chimera system, and just taking advantage of the fact that the different populations reconstitute with very different kinetics. So that the naive Treg reconstitute much quicker than the memory Treg. So the idea then was we could give tumor in a transplant very early on in the reconstitution where these dynamics are most distinct. And then we can analyze the phenotype of the Treg in the tumors and see what the donor host composition is, which population it sort of best sort of correlates with. So if we do this, this is just showing you the chimeras where, as I say, in the naive Treg, you can see a high level of donor chimerism at this time point, which is much lower in the memory Treg. And then when we look in the tumors of these mice, what's really sort of abundantly clear is that actually the donor infusion component of the Treg actual infiltrating the tumor is actually very low and matches really closely with what you see in the memory Treg pool. So I think for us, that's a very strong indication that these Treg are being pulled really quite specifically from the memory Treg compartment. So just to sort of conclude and sort of bring to a close, I think what this, as the title kind of alludes to, we think there's actually quite although there was a lot of sort of potential complexity in how the Treg, the circulating Treg compartment could be maintained. Actually, it turns out to be quite a sort of simple, straightforward story, that we see a very linear route of differentiation, that Treg generated in the thymus, some subset of those cells can become activated, and this constitutively feeds this kind of activated pool of memory Treg in the periphery, and that it's actually from this pool specifically that the tumors, at least initially, are drawing the Treg infiltrate into the tumor. And so this kind of represents a sort of broadly self-reactive primed Treg that is actively being pulled into the tumor. And we think that this represents perhaps a first line of defense by the tumor against the adaptive immune system. So just to kind of conclude, as I say, and acknowledge the people, so acknowledge the funders, and especially the people who did it. So Sanmoy and Sankat, who did a fantastic job, and Thea as well, who did a heroic job of performing all these busulfan chimeras in different labs, in different environments. So I'll stop there and take any questions. Thank you.
Thank you, Professor Benedict, for taking us through T regulatory phenotypes in health and malignant disease through your in vivo mouse models. Now I invite audiences, like 2 or 3 quick questions from the speaker. Anyone in the audience?
Thank you. It was a very lucid and wonderful presentation, and taking us through the functionality of how the naive Tregs develop into effector Tregs. So there is another population of inducible Tregs. So when you find that these naive Tregs are the ones which migrate to the tumors, so is there any difference in the functionality between the effector, the inducible Tregs and these naive Tregs which develop into effector Tregs?
Yeah, that's a great question, and that's kind of on one of our list to do is how important is it what the source of the Treg, whether it's is it just a matter of specificity that they come from different driven by different antigens, but they can end up at the same sort of differentiation state, or whether there's really important functional differences. So we're very interested to try and address that.
Yes. Also, on the other side of the spectrum, if you take dysregulation in the Tregs, which leads to autoimmunity. So which of these subsets are responsible, whether it is the inducible Tregs, which are not as functional, or whether it is the memory-derived Treg cells?
Yeah, I mean that's a really great question as well. And I think probably the Tregs which regulate autoimmunity. I mean, I think if you look in tissues, you can see that there's tissue resident T cells in a variety of tissues, and I think you can see regulatory T cells in tissues as well. So it's kind of a whole nother question whether those derive from the naive Tregs or not, or whether actually they're an inducible component as we see in the gut. Yeah, so that would be very interesting to look, and we should look in the fate reporters actually and see if we can isolate Tregs from those tissues, which are probably the most likely kind of regulators for the tissue-specific autoimmunity.
So these naive Tregs, were they working in antigen-dependent fashion or through cognate interaction, or it was antigen-independent separation?
Yeah, so that, again, that's a really good question that I don't have the answer to, but that, again, it's something that really occurs to us that, generally when people have done suppression assays or they've done, suppression in vivo or in vitro, they treat the Treg compartment as just a sort of homogeneous pool, and you take those out. So actually, it's really important to know whether, as you say, there's clearly different mechanisms of effector mechanisms for Treg. Some of those may be highly antigen dependent and some may not be. To me, it doesn't make sense that naive Tregs would have no function at all in regulating responses. And certainly I think of Tregs that have CTLA4, they can certainly have an impact on the APC, but maybe for some more mature Treg functions, such as sort of cytokine mediated suppression, that may require a sort of elicited antigen-specific stimulation.
May I ask you this question, Dr. Benedict? So as a Benedict solution for me, I am an autoimmune patient. Do I need to drink autoclaved water now for better T cells in my body? That's a good question. I mean, normally I would say it's probably better to have a little bit of dirt, but that's coming from a very clean kind of environment. So then I don't know. You want a little bit of dirt, but you don't want to be constantly unwell. Yeah.
Benedict, this question, this experiment that you showed on clean and dirty was very interesting to me, particularly when we are talking on climate change, pollution, for example, in the PM 2.5 going very high, NO2 and all that, and these people become very much susceptible to many diseases there. Do you envisage the same story as you showed us on clean and dirty here as well, that the Treg becomes really dysfunctional?
Yeah, I mean that's an interesting thought. I think, the surprising thing in some ways is that we didn't see as much impact on the Treg as we might have anticipated from the microbiota. So actually, it's more perhaps a balance that because you're getting this specific effect on conventional cells, that the upset in the balance is coming from the conventional side rather than the regulatory side. But it's really important, I think, to understand how these environmental signals are impacting the different compartments.
I think we would just take one last question from that gentleman.
Very nice talk. I had one question. A few years back, a couple of groups had shown that during early life, you have a specific population of Tregs which are coming in the periphery, and they are more involved in suppression of immune response against tumor antigens. So do you think that this imprinting is happening in the thymus for the populations you're showing in the periphery?
Our data would argue for a kind of peripheral imprinting, and just because that's where the effector memory cells are getting their stimulation. But having said that, it's possible that this is clearly happening in the neonate as well. So although there's many more naive Treg, they are becoming activated, and although the naive Treg get generated faster and to a greater extent, actually, when you look at the up curve in the neonatal mice, actually there's a big expansion of the effector memory Treg from early on as well. So they're probably in a highly activated state on a sort of per cell basis, I would say.
Thanks.
Thank you. Now we close the session.
You get my picture, right?
I'm just going to announce that.
Oh, yeah.
Our heartfelt thanks to Professor Benedict Seddon and the chair for this session. I kindly request both the chairs and the speaker to come up for a picture.
Thank you.
We have reached the final session of our event, where we have 3 parallel sessions happening simultaneously in different halls. For those of you staying in the main hall, we have an exciting session lined up titled as Comparative and Evolutionary Immunology. For those attending the parallel sessions, you may now proceed to Hall A or Hall B. Please refer to the event program for specific details on the topics in each hall. The main session will continue here in the main hall itself. I would request the people who want to attend the parallel sessions to kindly move to Hall A or Hall B. I would like to invite our session chairs, Professor Renny Borges and Dr. Rupesh Srivastav, to take over and lead the discussions for this session.
Good evening, everybody. And it's okay. Oh, sorry. I'm so sorry.
Thank you all for staying till, so late, and we won't delay in getting the session on. If I know that we are running quite late, so could I request the three speakers to try and maybe finish within 15 minutes if possible?
I will try. I will do it. Yes.
So, of course, on behalf of my co-chair, Dr. Rupesh Srivastava, whom you've already heard earlier, I'm very happy to invite Tina Mukherjee, who is from INSTEM in Bangalore, to make her presentation.
Great. Thank you so much. And without much ado, I will just jump into the topic. So I'm a fly biologist. Our lab works on Drosophila development, and something that our lab has been particularly interested in understanding is how metabolism controls development. And just a brief preview into why metabolism controls development of blood cells. Because if you see, this is hematopoiesis that is seen across the two different species, mouse and Drosophila. And if you would appreciate that development of blood cells, so everything that is red here are blood cells that are developing across different stages, in which is embryo, then comes the larvae, and then the pupa and the adult. And what is very striking is that they are different stages in physiology. So embryo is a very closed unit, which is not feeding. Then comes the larvae, when it is actually actively feeding and growing. So the embryo is not growing in size, but just developing, when blood cells are also developing in this context. Then comes the larvae, which is actually growing in size and actually is also actively feeding. So there is growth and feeding and blood development that happens in this phase of the life cycle of the fly. And then comes the larval stage. From the larval stages comes the pupa and the adult. When the growth has stopped, blood development is maintained, or homeostasis is achieved in this context, and the animal is feeding ad libitum when it needs. So the thing which strikes us is that how do blood cells develop in these diverse contexts where the physiology is so distinct, but it has to maintain its development and homeostasis? So this is a central question that the lab explores. And what we have found out in our ongoing explorations, that the blood cells, even though they express markers that we constantly see, when we start looking at their transcriptional landscape, they're quite distinct. So if you see here, this is an embryonic blood cell versus a larval blood cell. So immune cells that have been obtained from early embryos versus immune cells that have been obtained from the larval stages of development, they are quite distinct. While the metabolic, and what is distinctly observed is that their metabolic states are not the same. So what we find is that their embryonic hemocytes are glycolytic and very lipogenic, and whereas the larval hemocytes are not glycolytic at all, they're not lipogenic at all, they're rather very oxidative and also very lipolytic. So they're very typical metabolic states in which these two immune cells or the immune cells from the embryonic or the larval stages are defined. And what we want to understand, what do these state changes mean to the blood cells and any function that they would contribute to. And one of the things that I would like to point out is that this is developmental changes and not seen, which is not an immune context. We understand immune cells from the standpoint of immunity. I don't need to explain it to this audience. The whole meeting is about how immune cells are contributing to immune changes. But this is a very developmental context that we are trying to explore, that in homeostasis, in development, what would these transitions mean as the blood blood cells develop from one stage of the animal to the other. And what we identified is that, so this is a third insta Drosophila lavi, and what I'm trying to show here are all the green dots, which are blood cells, and they're this distinct pool of blood cells. The ones in here are all circulatory in nature, whereas this is the hematopoietic organ, which is the bone marrow equivalent, where the definitive wave of hematopoiesis happens. And this is not the relevant, or I mean, this is not relevant to today's talk, but what is very important for today's talk are these circulating immune cells. And what we find is that these circulating immune cells have, when we look at their internal metabolic states, now that the transcriptome has revealed their transitions, what we are wanting to see is how do they look? How do these circulating immune cells look in these metabolic contexts? And what we find is that so these are red. This is when animal is reared on a regular diet. And what we find is that these macrophages, we call them macrophage-like because they're highly phagocytic. So they're actively taking up beads. Every red dot is a bead here, so they're very phagocytic. And the green dots here that you're seeing here are actually the lipid droplets. So this is the state in which we identify or find a majority of the macrophages in a third insta state when their animal are living on a regular diet. What is very striking is that when we change the diet of the animal we start giving them high sugar diet, and within 4 hours of high sugar diet, we start seeing that their phagocytic activity has gone down and they've become very lipogenic. So the question is, the animal is experiencing a high sugar diet or a dietary change. Why should these immune cells respond to this dietary state so rapidly, and what does this mean to is there anything that this means to the animal in the context of development? Because the larvae has to develop into an adult fly. I'm not interested in asking whether the animal will be immune competent or immune compromised, but I just want to know, in homeostasis, what would these changes mean? And this is what the lab set out to ask. And what we as a first line of experiment we did is that we ablated immune cells in the Drosophila larvae. And so these are all the green cells that you have seen in the past picture and also seen here. So these are all the macrophages, or we call them macrophage-like cells in Drosophila. And this is a condition where we have ablated all the macrophages, or majority of the macrophages. And what we find is that when these animals are left to develop, they develop as smaller adults. And this is a wild-type adult fly. And this is, this is a wild-type adult fly, and this is a macrophage-depleted adult fly, which is smaller in size. This was not expected. We followed these animals all the way from larvae to adult stages. What we find is that the larval development or the size of the larvae are not affected, but when these animals are allowed to grow into the adult, they are smaller in size. Now, what is even more striking is that when I take these animals, subject them to a sugar stress, we're using a sugar as a dietary stress, there is a further compromise of the growth that is normally, I mean, if whatever the growth the animal achieves, these macrophage depleted ones are further retarded in growth. Now, this is a quantification, but what was interesting is that when we had a genetic combination where we actually increased the macrophage activity, or we increased the number of macrophages in the animal, and not just the increase in macrophage number, but also increased the activity of the macrophages, we found that it could restore the growth difference that was seen on the high sugar diet. So high sugar diet induced growth defect was restored when the macrophage activity was increased. And this is very specific changes introduced into the macrophage compartment and not anywhere in the animal. So this led us to conclude that there was something about the immune metabolic state or immune active state that was actually controlling animal size or animal growth. And this led us to ask, what is it and how is it being conducted? And that is what the talk is about. This was unexpected because we have never assumed immune cells would be regulating growth of the animals, scaling of the animal sizes. So to understand what is it about the immune cells that is leading to the growth, we did a screen, and like all fly biologists, we did a unbiased RNA, unbiased genetic screen, where we took different knockdown conditions for various genes available in the library that is there at NCBS, and we crossed them, we took these lines and crossed them to a driver line, and the idea was that, and we reared them on high sugar diet because this is a sensitized platform, to see what was the effect on growth, whether they were big like the PVR activated or they were small like the HID Reaper condition, or was there any condition that, so whether they were restored for the size, whether they were big or they were small. And when we did this, we identified a subset of candidates, and the candidate of today's interest is this particular one, which is SEMA. So what is SEMA? SEMA is actually orthologous to hypoxia-inducible factor HIF alpha, and essentially what caught our attention is that this is a transcription factor. So when this transcription factor is downregulated in the blood cells, we could see a plus, which means a growth recovery. So we're interested in understanding transcriptional changes in the immune compartment because that would be upstream, and any consequence on the above things which are metabolic are perhaps downstream. So we took this transcription factor and we started exploring it. And what is interesting is that when the animal shifts from a regular food to a high sugar diet, you see a growth retardation, but also what we see is the stabilization. So normally the red here defines the HIF expression normally seen in these blood cells. And what we find is that when the animal shifts to a high sugar diet, there is increased in HIF stabilization. So HIF, what we identified in the screen, was also seen to be going up in the high sugar context. And so what is HIF? HIF is the transcription factor that leads to hypoxia response. It binds to its beta partner, HIF-1 beta, activates downstream target genes, which are HIF response, carrying the HIF response element, and the one that is very well explored is the lactate dehydrogenase, which is the canonical target gene. So what we set out to do is that we now quantified. So this is the growth defect that is seen on smaller size. When we have a HIF RNAi, SEMA is the HIF component or orthologous component, you can see that there is a growth recovery. And also the downstream target gene, which is LDH RNAi, even if we remove that from the blood cells, we are able to see the growth recovery. So what this tells us that on high sugar diet, SEMA stabilization and induction of a glycolytic shift is what is leading to this growth retardation. And when we are inhibiting these changes, we are able to allow growth recovery. And what we find here is striking is that this is independent of the HIF-1 beta partner tango. This may be a non-canonical function of HIF binding through something else. Now, while this was on high sugar, we wanted to know if this is a general convention that HIF or the metabolic changes in the blood cells would be normally relevant for development in regular conditions as well. So we took regular food and did the same manipulations in the blood cells. And what we find is that when we upregulate, so what high sugar does is upregulates HIF in the blood cells. So we genetically manipulated the blood cells to overexpress HIF in the blood cells on a regular diet. And what we find is that they are smaller in size, not just by manipulating HIF, but other parameters which would inhibit HIF degradation. So other ways of stabilizing HIF in the blood cells, we're able to see the same consequence that the animals are smaller in size. And when we are able to downregulate HIF in the blood cells, the animal actually becomes larger in size, not just the HIF component itself, but also the LDH RNAi, which is the downstream target. So which means that even in homeostasis, this transcription factor has a consequence on the animal and how big it is going to develop. So the question is now, what is the HIF trajectory in blood cells when it is in normal conditions? So now what is the development trajectory of Drosophila? So from an embryo, it has the larval stages where it transitions to different instars, and then pupates, and then adult, and from the pupa emerges an adult fly. So we looked at HIF expression, and what is very interesting is that HIF expression gradually with development goes down, which is not seen when we have a high sugar diet. So when we have a high sugar diet, this downward trajectory is not seen. So the importance of this downward trajectory of HIF is perhaps relevant for the animal to grow. And if we what the student has done is actually has further downregulated HIF much earlier. So what the student has done is that actually downregulated HIF at this point of time, and he sees that if I further downregulate HIF earlier in development, he can actually see a much larger fly size. So what developmentally happens, if I further exaggerate the downregulation, I can further scale up the growth of the animal. So which means that there is a very strict regulation on HIF expression in the early stages of the larval blood cells, early in development, to control the scaling of the animal size. Now, how is this change in early larval life contributing to the adult size? So what is controlling HIF going down, what controls HIF to go down in early stages of development is what we asked next. And one thing that we hypothesize is that there are different kinds of diet that we are subjecting the animal to. We are subjecting the animal to high sugar stresses and other kinds of diet. And from the screen, there was one candidate that we found out, which was SlimFast. So SlimFast is an amino acid transporter. And what we found is that when this amino acid transporter is downregulated in the blood cells, it was a smaller size. So which means that sensing of sensing of amino acids by the blood cells was also necessary for scaling of animal size. So, and when this was in high sugar, also in regular food, when we downregulated the amino acid transporter, we could see that the growth was compromised. So, which is just the opposite of HIF, that you need this transporter mediated sensing of amino acids to for the animal to grow. And we asked what is the extent of HIF level in these conditions, and we find that in blood cells that cannot sense amino acids, the HIF levels go up. So which means that when an animal is subjected to a high sugar diet or regular food conditions, when they're not able to sense enough amino acids, HIF levels go up, and that is perhaps the reason why the animal is smaller in size. Now, what we have done is downregulated, which is not here. I mean, this corroborates with the results on high sugar diet, where high sugar also stabilizes HIF, and not being able to sense amino acids also stabilizes HIF. So the sensing of nutrient balance between sugar or amino acids is what is subjecting the stability of HIF in these blood cells. If it senses amino acids, the HIF levels are down. If it doesn't sense amino acids or it senses high sugar, HIF expression goes up, and that sets up the trajectory of the animal to a growth retardation. So what is the growth retardation meant by? And how is this impacted by the macrophages? Now, what I want to bring out here is that as I mentioned, that there is the larval stage of development. What we find is that when we are manipulating HIF, the larvae are not changing their size. So in this stage, when the blood cells are being manipulated, it doesn't affect the size of this particular stage. It is only in the adult when they are eclosing as smaller or larger adults. Now, how Drosophila, I mean, we call about Drosophila as born twice, once as a larvae and then from a pupa as an adult. Now, these adult structures actually come from very specialized structures called imaginal discs or epithelial pockets, epithelial cells that are set aside in the larvae, and they're just barely defined and developing. Now, it is only in the pupal stages that these imaginal structures are going to grow and develop and give rise to the adult fly. We looked at these imaginal structures in the HIF conditions, and what we find is that, and I'm talking about one particular imaginal structure, which is the wing disc, and what you can appreciate here is that this is a control wing disc from a third instar larvae, and this is a condition which has low HIF where the animals are big, and you can see that the wing disc is also much larger. Now, a condition which has high HIF, the wing disc is considerably smaller. And what we find here is that the proliferation, we have just done a pH 3 to mark mitotic cells. There's not much difference in the mitotic capacity of these discs. So there is a growth defect which is not explained by any dramatic change in pH 3 levels. So the question,
Tina, you're going to have to.
Yes. I'm almost there. So what we find is that when we cut the long story short, what we are able to actually find is that there is a defect in translation. So I'll just jump here. What we find is that conditions which have a smaller disc, they are not capacitated to conduct protein translation. So this is a puromycin labeling, and you can see that active protein synthesis is much reduced in these discs. And when asked for why is it reduced, what we know now is that there is a defect in rRNA biogenesis. So there is a defect in ribosome biogenesis. Not that they don't have ribosomal RNAs to be made. They are making ribosomal RNAs, but those precursor ribosomal RNAs are not being spliced or they are not being spliced into these functional ones, which are going to be incorporated into the ribosomes. So, but this is not a defect that is seen across the animal. I just put the fat body here. This is a larval tissue which contributes to larval nutrient regulation. And you can see here that that kind of defect, which we see in the wing disc, where the precursor is there, but not the smaller versions, the spliced ones, they are comparable here, but not comparable here. There is a downregulation of these. And putting the story together, how are we actually thinking about it, is that the way the RNA biogenesis happens is that there is a precursor one, which is then methylated and then spliced. We looked at methylation, and what we find is that there is a defect in methylation regulation of SAM metabolism in these discs, and clearly that there is, even though, so there is an increased conversion of SAM to SAM is being broken down very rapidly in these wing discs. And what is happening is that in these wing discs is that because SAM is being broken down very rapidly, the availability of SAM for methylation function, like methylation of the DNA, methylation of the RNAs, is not happening. And also there is perhaps excessive adenosine. So our idea is that perhaps SAM availability is limited for the methylation functions that we are looking at, which would be necessary for the rRNA biogenesis. So what the student has done is actually fed methionine and seen if they can recover. So when we feed methionine to these animals, we're able to recover the growth defect. So coming down to the conclusion is that there is a developmental regulation that blood cells undergo. And as they're developing themselves, they're also sensing the nutrient state in which the animal is. So as the animal starts to feed, it senses the balance of amino acids or balance of the nutrient, and then dictates the amount of metabolic regulation or translational capacity through regulating SAM homeostasis in these imaginal structures. Because along with blood cells, these are the structures which are going to be developing. And in conditions where they are, with this regulation is lost, like in high sugar or amino acid depleted condition, there is a dysregulation of SAM metabolism and the animals emerge as smaller flies. And one last...
You're going to have to wrap up.
Yeah, I just want to just bring it up here that while we're talking about a fly-specific thing, is this a fly-specific thing or not? We have started doing some depletion experiments in the macrophage, and we find that very early in development, if the macrophages are depleted from the mouse, as they are born, they are normal size, but as they grow into smaller adults. So this tells us that there is a parallel between what we are looking at in the flies and across the mammalian context as well. So this is something that we are carrying forward. And the work that I talk about today is majority done by Sajjad and Anushri, the screen, and Rahul has been looking at the methylation of RNAs, and it's a collaborative project with Angela and Pierre. So thanking all the funding, and thank you so much for the opportunity to present our work. Thank you so much.
I think only one question, and a short one.
A nice talk, Tina. My question is, the model that you showed about sugar, HIF, amino acid transporters, how specific is it to your immune cells versus the other cells?
They're very specific. So the way we are manipulating it is done only in the immune cells. So I didn't show a lot of things, but the other cells are not, I mean, they show an impact, but the genetic manipulations are done very specifically in the immune cells.
But wouldn't high sugar affect other cells?
Yes. So the fact is that high sugar is affecting other cells, but the effect on those cells is not compromising the growth. The one that is able to compromise the growth of the specifically of the imaginal disc is blood related.
Thank you very much.
Thank you.
So thank you, Dr. Tina. So our next speaker is Dr. Guru Shankaran from Department of Zoology, Central School of Biological Sciences, Central University of Kerala. And his talk is going to be on ecoimmunology in global amphibian crisis, study on anurin peptides. So I will ask Dr. Guru to kindly go ahead with his talk. And we request you to kindly cut it short to 15 minutes.
No, I will take only 10 minutes.
That's great.
This is general talk. IMMUNOCON.
Is there a problem with the presentation? Okay.
Working. Can I use this one only? Okay. Yeah, sorry. Good evening to all. First of all, I know I am coming from Malabar region of Kerala. It's near to Mangaluru. This is, very traditional place. It's a Saptabhasa Sangama Bhumi, 7 language speaking place in Kerala. Okay, I invite you all for any other programs. Today, I'm going to speak on ecoimmunology. First of all, I should thank Professor Deepankar Nandi, when I mentioned about this talk, topic of the title, he was appreciated and he mentioned that you can speak because it's not immunology. Just I am going to connect ecology with immunology, as Deepankar clearly assigned 2 chairs. One is Professor Rini is an ecologist and Rupesh is an immunologist. This is the one combination I want to make. It's a eco-immunology. Okay, this is the one immunology. From morning we are speaking about all threats and all the immune response and all these things. In that all immunologists all developed some of the immunotherapeutics and vaccines, all these things in laboratory models. They're all identical models. That is the one Clara, as earlier speakers clearly mentioned about the variations in immune response. That is the one what will happen in the field. That is the only that concept I want to bring here. That's the one. If you take here, that is in case of individual level or species level or population level or even community level, how this immune variation is there, how it is going to be contributing and it's developing some of defending their pathogens. That is the one I'm going to speak. I'm going to convey to you all. That's all. This is the one is a first paper. This paper, the prevalence and intensity of schistosoma, whether it is a ecology or immunology. That was the question. This was the paper passed in 1973. It was published. After that, there was no publication regarding eco-immunology. This is in 1996. These 2 scientists, Sheldon and Verold, they have coined this terminology, ecological immunology. After that, there is no concept of eco-immunology. Now we are having concept that is an emerging discipline that seeks to understand the causes and consequences of variation in immunity in the context of host environment. In the host environment, how it is going to be varies. The immune response is going to be varies within the species or among the species. In that, why it is so important means it is having a lot of implications in agriculture, biodiversity conservation, and public health. For that, understanding this variation, the immunity within the species or among the species, it will help to manage some strategies, how to defend, how to minimize the particular disease and other things. For that reason, we have started working on this. In India, is a mega diverse, is a fantastic country. We are having 4 hotspots in India. Number 1 is Western Ghats, Eastern Himalaya, Indo-Burma, and Sundarland. In that, major one is that Western Ghats. This is the Western Ghat. You can see this is the one. It's the Western Ghats. We are having globally 325 threatened species are there. For that UNESCO has declared that it is one of the World Heritage Sites in 2012. In that Western Ghat, I started working from 2001 onwards on amphibians. These amphibians declines and extinctions are happening in India and also in global level. In that you can see this is the one amphibians they evolved at the time of Devonian period when that these big animals were there in that time. These are the amphibians. The amphibians are phyclothermic organism. All these things. That's I don't explain all these things. These are all very important in ecosystem. There are the insectivores. There are the resource for various peptides and all these things. I'm not going to explain all these things. This is the one we are having 50 million species, but till now we are described only 1.7 million species. In that, just 10% of species are described. In that, amphibians are having this many, 8,787 species amphibians. In that, we are having 3 orders. One is Anura, is nothing but frogs and toads. They are the dominant one. Then another is Gymnophiona, is a legless amphibians. They look like snakes. And another is Chordata, they are all salamanders and newts. But in India, we are having 454 species. It's a Chordata is having 2 species, only 2 species in India. Anura is a 411 species, Gymnophiona 41 species. Okay, these are the various. You can see that beauty of this frogs and toads in the Western Ghats and also in India. We are having 16 families in India. These are the 16 families. In that, if you take India, the endemism is
is, if you take found nowhere else in the world, out of 7 families, 3 families are in our India. This, if you see that amphibians are around 60% of endemic to the India. If you take birds and mammals and reptiles and flora. Okay, these are the one, is a threatened. You can see that how many are threatened in amphibians threatened in India and global. Some of them are already vanishing. So you can see this is the one is a poison frog is extinct, is a golden toad is also extinct, and this brooding frog is also extinct. Some of them are already critically endangered one. This already vanishing. In same time, same type of nature in case of India. You can see if this is the one, it will tell the most threatened vertebrates in the world. Particularly 41% of amphibians are in under threatened conditions compared to birds or even mammals. If you see, this is the one, you can see it's a 41% in amphibians, 12% in birds, and mammals is 26%. Highest one is 71% in cycads. These are the one is a IUCN threatened categories in 2004 and 2023. If you compare this one, you can see the threatened, how much particular amphibians are under threatened categories. Okay, these are the causes. Particularly, you can see that a loss of habitat and habitat alteration. Then another is pollution. Then another is emerging infectious disease. These are the 3 you can see. If you see this, the first one is a 99% of habitat loss in India and also in the world. It's the same. If you see this is the one you can see in your Western Ghats. It's a 99% habitat loss. Just I am not going to explain anything. My pictures is going to say everything. This is a pollution. It's just I am working on pollution, particularly pesticides from 2001 onwards. You can see the effect of pesticides on amphibians. Amphibians are all, they're all habitat for parasites and microbes. Particularly, you can see this is the one many microbes are, affecting their population, particularly their survivability, particularly fungal or viruses and bacterial and other type of microbes. This is the one emerging disease in amphibians is chytridiomycosis. You can see the death of many mass mortality of the amphibians. That's why 20th century is considered as it's a worst disease, a wildlife disease of chytridiomycosis is one of the worst wildlife disease in 20th century. It's nothing but it's an amphibian plague. These are the one you can see this is a mass mortality of amphibians. This is the one caused by a chytrid fungus. This is the life cycle of chytrid fungus. You can see this type of chytrid fungus is also reported in our Western Ghats in 2011 is a first report. Then again second report in 2013. Then again another one report in 2018. Okay, these are the one we have observed morphological abnormalities in amphibians in Western Ghats. Then we reported this in case of paddy fields and coffee plantation using wherever the pesticide is applied, that particular number is more compared to the pristine ecosystem like forest or water bodies. This is the one, particularly amphibian immune system is completely different from innate and adaptive immune system compared to mammals. But we are not understand this immune system. But even tadpole and adults also they are differ from their immune system. Just I am not going to speak. Major one is this is the one they are going to produce a particular antimicrobial peptide. Their skin is going to secrete this type of antimicrobial peptides. All these peptides is already, we discovered, we described, we are characterized, and they are used for some commercial purposes. Particularly, urmin is described in Hydrophilix bohesthera frog. That is the one is used for inhibiting influenza virus. These are the many type of peptides is already described. They're having different type of activities. This is the one we worked about. This one tigrinin 1. This is the one anti-tumor activity of tigrinin 1 in case of particularly in A54 cell line. Then another one is a temporin SHF. Another one peptide we are worked out. This is the one is having antimicrobial and anti-tumor activities of this particularly cancerous cells and particular microbes. Whether do environmental stresses contribute to immunosuppression and amphibian declines? For that also we worked. We have selected this frog. This frog is commonly available in particular forest and sacred grove, paddy fields, and particularly endosulfan spread area. We collected this one. We have observed that the stress hormone is increases and that immune cells also is going to be is affected. Particularly, you can see that the corticosteroid hormone in case of endosulfan spread area is increases, but in case of these lymphocytes is going to reduces. Okay, same time we have collected the skin secretion. Just we worked about that secretion level. You can see that the pristine ecosystem is secretion is more compared to endosulfan and agriculture field area. And also you can see that their minimal inhibitory concentration effect, you can see that endosulfan spread area is taking more MIC value than other pristine ecosystem. Their abundance also, their number is going to reduce in case of endosulfan and agriculture field area. From this, I conclude that the microbes can play vital roles in host health. Emerging infectious diseases are threatening wildlife. Amphibian responses to the environment are mediated through the HP axis. Environmental stress and disease may interact to compromise survival. Ecological stressors will impact the immune system. Thank you.
Thank you, Dr. Guru. It was actually a wonderful talk. And I would say that this ecological immunology is already a part of your One Health approach, which is now gaining importance because without ecology, environmental science is having each and everything within it. So this is just a part of it. So it is already a very important field. And it is part of the One Health approach in which we are taking actually the animals, plants, and the human beings. So all these are basically linked. Thank you. So we will have just one quick question. If anyone is having any question.
Thank you. We know a lot about the fish immune system and then up higher birds, everything. How much do we know about the amphibian immune system?
That is already I mentioned, madam. Particularly in tadpoles, there is no MSC.
I see.
But in case of adults, MSCs, that's why they are all more affected this one. Particularly pesticides and habitat loss, no? It is going to cause a lot of drastic effects. Since tadpoles are not having MSC 1 and 2. But in case of adults, once they metamorphose, the adults are going to have MSC 1 only. And also another one is IgM is not going to be synthesized in adults.
Thank you so much.
Sorry, I am rushed to that because of time.
Okay, thank you. Thank you very much. Thank you. Okay, so we now come to the last talk in this session, and this is by Karthik Sunagar, and he will talk on the evolutionary ecology of Indian snake venoms and next generation solutions for snake bite. Karthik is a colleague of mine at the Center for Ecological Sciences in IISc, and we're really looking forward to his talk.
Sure. Yeah, thanks. Good evening, everyone. I know it's way too late for a scientific talk. But what I thought I'll do is because you might not be familiar with snake venoms and snake bites, I'll give you very brief introduction to both of these terms, and then I'll explain how we are using advanced immunological technologies to solve this century-old problem. So just to give you a background, so I had the evolutionary genomics lab at CES IISc, and we are mostly interested in understanding how ecology and evolution shape snake venoms. But major emphasis is also placed on discovering these broadly effective snakebite therapeutics, because you'll see that snakebite is a major problem in this part of the world. But we don't work on snakes alone. We work on all kinds of venomous and poisonous animals from India and elsewhere. But coming to snakebite, kills over 58,000 people in India every single year, and 3 times that number suffer permanent immutable injuries. And over 50% of all snakebite-related deaths that happen globally occur in India, and that's why it's a major problem. But it's one of the most highly neglected of the neglected tropical diseases. So there are very few people trying to solve this problem, at least for India and sub-Saharan Africa. So snake bites, as you may have heard, is treated using antivenoms, which is produced by hyperimmunizing equines, horses, ponies, and mules with target snake venoms, and then collecting the resultant antibodies, and that's packaged as antivenom. We have already known that different populations of the same species of snake can have very different venoms across its distribution. But ignoring all of that, we still produce an antivenom by collecting venoms from snakes from just one point location in southern part of the country, and this antivenom is used for treating bites from all kinds of snakes across India. And until very recently, it was also used for treating snake bites in neighboring countries as well as in sub-Saharan Africa, which made absolutely no sense. Before coming to India, I thought at least these antivenoms would be very effective in southern India because of the close proximity to the source population. But it turns out that even here in Karnataka, the antivenoms are not that effective because we are ignoring the evolutionary ecology of these snakes. Now, there are 4 major problems of snake bites or the current antivenoms. The first problem, as I showed you, was the limited specificity. The second problem is that this is a century-old technology that we are using. So almost 90% of antibodies that you find in a vial are redundant from a clinical perspective. They are targeting other antigens that you find in snake venoms. So what happens is, it has very low dose efficacy, meaning that you would require many vials for treating snake bites. And in some regions, a single vial can cost more than $100, which is just unaffordable to those people. And you might require more than 50 vials for treating snake bites in some regions. And it also adds to the cost. So they are extremely expensive. And they are unsafe because these are equine antibodies that we are pumping into humans for treating snake bite. And we know that in a lot of cases, they result in adverse reactions, including anaphylaxis, which can kill the patient faster than the snake venom. So they are very unsafe too. So we need to address all of these 4 problems of antivenoms. The current antivenoms are only made against 4 species of snakes: the spectacled cobra, Russell's viper, common crate, and saw-scaled viper. And antivenoms don't exist for any other medically important snake species. So what we have been doing over the last 6-7 years is we go to the wild and catch these snakes, all the medically important snakes, including the big 4, and we collect the venoms, we bring them back to the lab, and then we look at their activities and so on and so forth. To keep you awake, I have sprinkled photographs of snakes that I have taken in and around the campus, as well as in other parts of. So the first major problem of, or the attribute of venoms is that they vary dramatically. So I'll show you, so this is a study that we did a few years ago. We looked at some populations of spectacled cobra across India. We now have complete profiles of these snakes from all across India. And if you just focus on peak number 4, which is what contains this thing called neurotoxic 3-finger toxin. So this is what kills people when they are bitten by cobras. You can see that some populations have really large amounts of this neurotoxin, whereas, for example, Punjab. But if you go to the neighboring state, Rajasthan, you see that the amount of this toxin dramatically reduces, and it is now replaced by a toxin that causes necrosis. So if you go and see people who are bitten by cobras in Rajasthan, you'll see a lot of necrosis in their limbs. And so mass spec basically shows the difference, the exact compositional difference between these populations. And we now have profiles of venom profiles of all the medically important snakes from all corners of India. Venoms can I showed that venoms change across larger distances, right? But if we go out now and capture cobras from this campus, and trust me that there are many of them on the campus, so watch if you go out for a walk at night. So if you catch multiple individuals and look at the venoms, you don't expect to see variation, right? But when we did this in West Bengal and looked at this other species of cobra called a monocled cobra or a monocellate cobra, we captured individuals from a single location and looked at the venoms. So the venom profiles are represented by these donut charts. You can see that no two donut charts look similar to each other, right? They all have very different components, although they are from the same location. So much so that the antivenoms would work against some individuals, but not against others. So the outcome of treatment could even come down to, the individual snake that has bitten you. So that's the extent of venom variation. Venoms can also change as these animals age. So if you look at this video, look at the tail of this just born Russell's viper. You see that it's wiggling it like a worm, and it does so in order to attract lizard and amphibian prey towards them. So that's the strategy. I don't know if you can see the video. Yeah, you can see the tail, right? So they do that as soon as they're born. That's how they attract, lure. This is called caudal luring. So they lure the prey towards them, and that's how they get their meal. So we knew that these snakes probably feed on different things when they are small. So what we did is we captured cobras and Russell's vipers, and we housed them. So over 200 individual snakes, in captivity right from the day of their birth until they reached adulthood, and we profiled the venoms across a couple of years. And again, just highlighting the major findings, you see that venom becomes more complex as these snakes age. But you might think that the adult venom, because it has so many different components, it might be more toxic than the newborn baby. But it turns out that the baby secretes a venom that is almost 2 times as toxic as its mother. So as soon as they're born, they are capable of defending themselves, and they are also very specific against lizards and amphibians, which I haven't shown. So the consequence of all of this ecological and evolutionary variations in snake venoms is that antivenoms don't really work very well across India. And in some cases like Rajasthan against cobras, all mice that are injected with venom die. So the way we test this is we inject the venom into mice and then try and see if these antivenoms are capable of rescuing mice from the toxic symptoms and whether they're capable of saving these test populations. And you can see that for some snakes, they completely fail. No matter how much of antibody or antivenom you give, all mice injected with venom die. So coming to the solutions for snake bite. So the first thing that actually goes against everything that I've said is we are trying to make a universal antivenom, an antivenom that works across continents. And that's because India is not the only place that suffers from snake bite. Sub-Saharan Africa and India collectively experience almost 90% of snake bites in the world. And you definitely need better solutions because current treatment strategies are very ineffective. So in this approach, what we attempted to do is, if you look at the venoms of some of these snakes like elapids, so medically important snakes, you can classify them into elapids and viperids and so on, but these two families collectively kill and maim a lot of people. So I'll talk about elapids first. So if you look at the venoms, almost 90% of the venom is composed of 3-finger toxins in some cases. So we said, what if you knock out this one toxin with an antibody that targets the 3-finger toxin? Would you be able to produce a solution that could work across geographic barriers? And to do this, we have taken multiple approaches. So we are immunizing cows, camels, baboons, with snake venoms from India and Sub-Saharan Africa. Cows in Bikaner in Rajasthan. Baboons and camels in Bikaner, Rajasthan. Cows, of course, I can't immunize them in India. It's being done in Kenya because I don't want to get lynched. So we have been immunizing these animals with specific fractions of toxins and then raising an antibody response. And then what we do is we make these yeast display libraries. So basically the yeast cells will now express these antibodies on the surface, and you can now use any antigen of your interest, be it a snake venom or a COVID virus, and then you pull out antibodies that are specific to that particular antigen, and then you keep doing this in the form of an iterative sorting, and you'll get very specific antibodies at the end of the campaign. And that's one approach. But in this approach, what we did is we built a synthetic human library. So the yeast cells, instead of these antibodies against snake venoms, they would express huge number of antibodies on their surface, huge diversity of antibodies on the surface. They could have antibodies that don't even exist in nature. And then we now screened, using 3-finger toxins and pulled out some of those antibodies and affinity matured them. I'm not showing any of that data here, but I'll just show you what we found. So one of the affinity matured antibody was very effective in neutralizing venoms of these elapid snakes from different continents. So this is a crate, one of the most toxic snakes from Southeast Asia. You can see that when you inject mice with single component of this particular snake venom, all of them die within 4 hours of injection. But when you pre-incubate it with this particular antibody, you can see that even post 24 hours, these mice survive. The antibody is also capable of rescuing them, meaning that you can inject the antibody a lot later, post injection of the venom. And this one particular antibody, because it was targeting 3-finger toxins, it works against the species of snake from Southeast Asia. The monocellate cobra that I told you earlier, where conventional antivenoms don't work, this particular antibody rescues all those mice as well as black mamba, which is again another most medically important snake from sub-Saharan Africa. So now we have these antibodies that work across continents, and they are extremely broadly neutralizing and solve all the major problems of antivenoms that I iterated earlier. And again, because they are human, you will not get any side effects. Hopefully, and then it's also an ethical way of producing antivenoms, because you don't have to immunize animals and harm them. So this was published, earlier in this year. That snake was photographed not far from where we are now. So the second and last example that I want to give you is of this particular snake that you see in the picture. It's a species of snake called Russell's viper, photographed in Kerala. And this snake kills more people than any other species of snake in the world. It kills and maims a lot of people. So if you now produce an antibody that works against this snake, you would literally solve the problem of snake bites in India. And so to do this, we collected certain fractions that contain medically important toxins from the snake, and we immunized camels in Bikaner, Rajasthan, as I told you. And then we harvested the RNA and turned that into a yeast display library. And this library was used now to discover antibodies against this particular toxin called snake venom metalloproteinase. And skipping all the details, so there are 3 posters being presented by my students, and they would be able to give you all the technical details. But I'll just skip to the interesting parts of this campaign. So one of the antibody that we discovered, which we named 10X, which is very confusing, and as you have seen in the demonstration. So I'll show you what happens when you inject mice with Russell's viper venom, right? So these mice, and this was 2 minutes, this was captured 2 minutes after injecting the venom. So you can see that one of them already collapsed. And a lot of times the moment you inject and by the time you put down the syringe, those mice vomit blood and die. So that's the potency of some of these venoms. But see what happens when you treat those mice with this particular antibody. This was 24 hours post-injection, and you can see that they're all pretty active, right? So so these are camelid antibodies, which are expressed in a chimeric form with the human FC and then the arms from the camelid, this thing. So you can even express them as nanobodies in future. And we are now exploring whether these antibodies just rescue from fatality, or whether they're also capable of rescuing mice from necrotic effects that you see with snake venoms. And we tested this antibody all across India. I'm just showing some populations here. It works everywhere except in one region, the eastern region. And unfortunately, and interestingly, most of the solutions that we have found so far don't work in West Bengal. So if you are planning to get bitten by a snake, don't go to West Bengal. Okay, you can get bitten elsewhere. The solutions might work. And we figured out that it's because, so this is the first SVMP specific antibody in the world. So we don't know how it works. So we are trying to solve the structure and figure out exactly what it is targeting. And this is the first antibody against vipers as well. So hopefully it'll, after affinity maturation and all, hopefully the effectiveness will increase. But here in this region, the Russell's vipers there also produce another component called phospholipase E2. So a combination of 2 antibodies might work in future. So I think that's my time. So I thank all the members in the lab who have done. that's photoshopped. Although we do take pictures like that, but I can't show that in public. But yeah, thanks so much for your time and attention.
So it was a wonderful talk, Dr. Karthik. And just some quick questions. Do we have any questions or?
It's too late for questions.
Okay. Dr. Javed has one question. Yeah.
Sorry, say that again. Oh,
So
you're
this
saying
is,
that because it's a camelid antibody, will there be when you inject it into humans? So yeah, I mean, clinical trials have to, they have to take place before it can be administered into humans. But you can also engineer and humanize these antibodies. So hopefully, that will solve that problem.
So this is not a question, this is advice. So you use Photoshop in your last slide. Don't use your Photoshop in your publications.
No, I have pictures of live snakes as well, as I said, but I can't really put them in my.
So one
just important thing, sir. सुबह से सोच रहा हूँ कि मैं डॉक्टर नंदी को कुछ बोलूँ या ये डॉक्टर साहब को कुछ बोलूँ. उन्होंने बोला कि वो एनवायरमेंट फ्रेंडली कॉन्फ्रेंस कर रहे हैं, तो दैट वाज इम्पोर्टेंट पॉइंट. और आप बोल रहे हैं कि अब एथिकल में आप एनिमल्स को नहीं परेशान करेंगे, जबकि आपने एनिमल्स ही यूज किए. So there are certain claims which we make. Okay, we really have to be very, very careful when we make these claims.
So 200,000 people die.
That
from,
immaterial. That's
from...
immaterial, actually.
No, it is. It is
so
important because...
animal use of animal in medical sciences is going to continue for a long, long time for various experiments and everything. The best thing you have to claim is that your antibody is working.
No,
more than ethics or anything. That's my personal opinion. And
I respectfully
that
disagree because now that you have these antibodies, you can recombinantly express them like we have been doing in mammalian cell lines, and that would prevent immunization of animals. So there are tens of thousands of horses and ponies and mules that are getting immunized right now. So all of that will stop. So that was my point.
So I have a question. Can I?
I just wanted to say thank you so much. This was a very interesting talk. I wanted to understand whether you or your students, how scared are you when you handle such snakes?
So students don't, only a couple of students handle snakes who are experienced. But yeah, I mean, we are all kind of grown up with snakes, so I think.
I have a question. Can I ask?
So can we do these questions later on during the dinner session? Yeah, so we can finish it up because we have a GBM, I think. Yeah, after this. So thank you, Dr. Karthik. Thanks a lot. And with this, we conclude this session.
Thank you.
Our sincere thanks to all the speakers and the chair. We kindly request all the speakers and the chair to come up on the dais for a group picture.
Actually, I think all these three talks were actually off the track for today's, all of the sessions. And we were thinking they could be boring and before the dinner, it could be sleepy, but actually they have opened our eyes. Yeah.
Lovely. Thank you so much.
Thank you to the speakers and the chair. Now we will conclude today's session with a general body meeting. The general body meeting is exclusively for the society members. We kindly request the remaining audience to make their way towards the dining area. For the Indian Immunology Society members, the meeting will take place in the main hall, followed by dinner. Also, I would request all the participants who have presented the poster today to kindly remove their poster from the boards, and any kind of misplacement will not be responsible for the volunteer.